keynote address here. Prof. Wandari will introduce the speaker soon. Right after the keynote speaker at 9:50 a.m., we would have a parallel session in the PhD lecture room. When you exit this hall, straight down the corridor, that's where the PhD lecture room is located. And over there, we would have 3 plenary talks and then fellow sessions. The plenary talks will be from Professor Emmanuel Tego, Professor Fong Pham, and then Dr. Sybil Obobi. So those interested in those talks should look at the titles of their presentations in the program, and you can make your way there to listen to them. Then right after the parallel session and then the first session here, would all converge here for the session on vaccine and immune-based biotherapeutics. So everybody should take note of those arrangements. We'll have the keynote address here. Those interested in the parallel session should move to the PhD lecture room, and then we reconverge here for the final session before the lunch break. Thank you very much. So right now, I'd like to call on Prof. Wandari to introduce the keynote speaker for the final day of the WAGBIP Research Conference. Thank you.
Good morning. I hope you have enjoyed the first 2 days and you have a little bit of energy left to push through the final day. Don't worry, there will be the famous party in the evening at which you'd recover all your energy. All right. So this morning we have the third of our keynote speakers. Are you guys ready? So his profile is projected, but really the reason why he's here is because he's young, he's African, he's Ghanaian, he's doing very well, he's in the diaspora, and he's interested in what is happening back home. So all those check the boxes of a WAGBIP, person. Okay. So when we heard about what he was doing, we invited him here to give him this platform to talk about his work first of all, but also to connect with us here and our young fellows here, and hopefully we can develop some partnerships that would eventually lead to him coming back home. At WAGBIP, we have a mission to bring as many talented Africans as possible back onto the continent. So we are always engaging talented Africans who are out there whose research interests or whose research expertise aligns with ours. I was saying yesterday to one of our colleagues who was here for the first time that at WAGBIP we are building platforms and we are building capacity, and then you do whatever you want to do. We don't, even though our name has infectious diseases in it, we don't stick to only infectious diseases. We provide the platforms, and young people can do whatever they want to do. You can do cancer, you can do heart disease, you can do genetic diversity of parasites, you can do genetics of human beings, whatever. So we just provide the platforms, we provide the facilities for young people to develop their talent and to explore what is interesting for them. So in that vein, we are quite well aligned with David because he uses tools that we also use, even though he's more looking at non-communicable diseases. So, are you ready? Is okay? All right.
Sure.
Yeah. Okay, so David is an assistant professor in the Department of Comparative Medicine and a member of the Yale Center for Molecular and Systems Metabolism. He's also a primary faculty at Yale's inaugural Human Genome Sciences graduate program. His research focuses on understanding how genetic variation shapes complex disease risk, with an emphasis on pleiotropy, hematopoiesis, and neuroimmune-mediated mechanisms. His research integrates population genetics, single-cell multi-omics, functional genomics, and experimental modeling to uncover shared biological pathways linking autoimmunity, cardiometabolism, and neurocognitive traits. He's a recipient of multiple research awards, including the NIH, NIDDK New Investigator Gateway Award for Collaborative T1D Research, and multiple Breakthrough T1D Awards. His work bridges fundamental genetic discovery with translational insights into disease mechanisms and therapeutic targets, while advancing the importance of diversity in genomic research. So, David, you're welcome.
Hello. So I'm going to use the microphone. So I'm like a lecturer. I like to walk around when I'm talking. Yeah, so I'm really, really impressed and very grateful for this opportunity to see that. I said it, I like to lecture rather than. So I'm really grateful for the opportunity to be here to share the work that I do at Yale with you. Most of you don't know this guy. His name is Satuga. You can call him, not Andrew. That was my classmate in Notre Dame. He really made my journey here very, very easy, and I feel like I'm at home. And one of the things that stood out to me yesterday was the undergraduates when they were doing their presentations. I remember when I was an undergrad at Tech, I had zero experience in doing experiments or presenting data. So I had to defend my project work, and I could not interpret data. So I just said, out of frustration, I said, in fact, the results are haphazard. So people laughed at me so hard. Up to this date, I still haven't forgotten. But yesterday, I saw really, really great presentations from the undergraduate students, and I'm really motivated to stay in touch with WorkBip and to know what they are doing and to see how I can also contribute to the work that they do here. So, let me try this one then. Hello? Okay. Okay. All right. Sorry about this. All right. So I'm going to be talking to you today about genetic pleiotropy. Like, where do I face it? Is there a specific item that I need to face?
Those guys to the point that
Okay. Yeah, I'm just okay. Okay. So I'm going to ask for extra time because my time has been used up. So this is my lab. I started my lab at Yale as an assistant professor in 2023. And right now I have Zara was a postdoc. She was the very first person that recruited to my lab, and then after 2 years, because she was doing really well, we promoted her to associate research scientist. And then I just onboarded 2 new postdocs, Peng Yu and Leona. And then there are other trainees, Michelle, she's a PhD student, Scale, then the rest, those are, I have these 2 undergrads who are really smart kids. Andy right there, he actually went to high school in Ghana here, and now he's doing his bachelor's at Yale. And then down here you have Jolade. Jolade's father is from Nigeria. She was the very first person to work with me, and a very exceptional. She got into MD PhD programs in all the US universities, Harvard, Columbia, all of them. But the mom stays in California, so she chose to go to UCSF. And then Presla there. Presla also went to high school in Ghana here. When I got the faculty position, she was the first research assistant. She now goes to Harvard Medical School. And the 2 of them are the first people I mentored. My first 2 publications in my lab, they are the first authors in my lab. I'm saying this because I place so much emphasis on mentorship, and I'm glad to be here to see other students who are interested in education. So just a little bit of background to the kind of work that I do. So, you know, we are different because we have SNPs or genetic variants in us. So if I want to understand why people have type 1 diabetes and some do not have, I can genotype them. I'll take people with type 1 diabetes and people without type 1 diabetes, genotype them and see the frequencies of SNPs that are unique to those with type 1 diabetes. And if these SNP differences are statistically significant, then you can say this SNP is associated with type 1 diabetes. Each of us have about 500,000 to 1 million SNPs. So let's say there's a reference human genome. If I genotype you now and then align it with that reference, I'm likely to find about 500 to 1 million SNPs. So because I'm testing up to that number of SNPs, we normally use a threshold of 5 times 10 to the power minus 8 as being genome-wide significant, because we've tested multiple SNPs up to 1 million. But there are some SNPs that may not meet this cutoff point, but they are still relevant to human disease. And rarely do you find SNPs residing in genes or the exon, so that you can just know that the SNP is causing the disease by affecting this gene. So just like human traits, the molecules that you express, like your genes, RNA expression levels, the protein abundance in your plasma, their levels are also regulated by genetic variation. If I express 100,000 transcripts of gene A, maybe you are making less of that. And that is also due to SNPs. So we can also map SNPs that regulate these molecular traits like gene expression levels. And if it is the gene expression levels, we call that expression QTL. And these SNPs that regulate gene expression, they are cell type dependent and context dependent. You may have a variant that influences the expression levels of a gene only when you get an infection. So those ones are like response equatorial. So you can have a single SNP that is associated with a disease and also influences the expression levels of a gene. So if you want to understand the mechanism through which a SNP causes a disease, and you find that this SNP co-localizes with an equatorial, it's a giveaway that maybe the SNP is causing the disease by influencing the expression levels of that gene in that cell type. That's the reason why I'm showing this. Okay, and because I study genetic pleiotropy, where a single variant can influence multiple traits, what is pleiotropy, and how do you detect pleiotropy? So the first diagram is like you can do a genetic correlation. Let's say I do a genome-wide studies for type 1 diabetes, and then a genome-wide association studies for cardiovascular disease, and I want to find whether they share genetic basis. So I can overlay them. I can do a correlation across the genome to see which regions they share genetic variants. So I can do a regression to find whether they have a negative or a positive correlation. And within pleiotropy, there could be different types. Like if you take every region, you inherit your DNA from your parents in blocks. So those blocks, they would always be inherited together. And in each block, you have like many SNPs. So some of the SNPs are just targeting other SNPs. They don't really cause disease. So you can find that two traits can share a signal in a locus, but then they have different SNPs that are causing those traits. In that case, you would say that's like a horizontal pleiotropy, or you can even have like two different SNPs in that region causing the two traits, not the same SNPs. That's like a horizontal pleiotropy. But in the case where you can have a single SNP, it causes trait A, and then trait A then causes trait B. That one is vertical pleiotropy. And vertical pleiotropy is very important in drug discovery. Most of you probably know what is called Mendelian randomization. It's based on vertical pleiotropy. If I want to know whether high cholesterol reduces heart risk, I simply take variants that are associated with high cholesterol levels, and then group people into people with heart disease and people without heart disease, and see whether those with the SNPs that are associated with cholesterol have more of those SNPs. It's just like a randomized control trial. So those differences are very important. And then if I want to find whether, I'm a little blind, if I want to find whether these two GWAS traits share the same SNP, you can do a co-localization. So there are statistical tools to test whether two SNPs co-localize, they share the same causal signal. So you can do the co-localization between two traits, let's say type 1 diabetes and type 1 diabetes, to find whether they share a single SNP. Or you can do co-localization between a trait, let's say type 1 diabetes, and gene expression in, let's say, T cells, to find whether the signal that causes type 1 diabetes is a SNP that causes gene regulation in the T cells. And these are other, I'm not going to spend so much time, these are other ways to detect pleiotropy. So the bottom line here is that why do people with certain diseases tend to develop other diseases more commonly? And we think that it is probably due to pleiotropy, where you inherit the risk for multiple traits. So rather than studying traits one at a time, we try to understand how they are related. Yeah, this is why we need to study pleiotropy. Sometimes you take a drug and then you experience side effects. And that is because of pleiotropy. You are targeting a gene, but the gene is important for another trait. And because you're blocking that pathway, you get the side effect. You can also use pleiotropy to predict who is likely to benefit from an intervention, let's say clinical trials and all of those things. And we can also use that to understand the biological mechanisms of disease comorbidity. So pleiotropy is an emerging area that is the future of precision medicine. Rather, I like to approach diseases like holistically. Like if you have a disease, it's like a systemic disease, rather than say someone has type 1 diabetes because they have lost their insulin beta cells. You spend decades trying to restore those beta cells, but the SNPs that underlie the disease also have effects on other tissues. So we try to understand all these multi-system effects of those SNPs. So like I said, we have a hypothesis that hematopoiesis is like the central conduit for pleiotropy. And we drive our hypothesis from ancient wisdom. Like even our forebears, if you are good at something, they would say it's in your blood. Let's say you excel in academics. People will say, oh, it's in your blood. Why don't they say it's in your brain? So they knew that the blood is the bearer of genes. So we think that these variants that have multi-organ effects, they have effects on the blood. So throughout your life, you have about 200,000 hematopoietic stem cells in your bone marrow. So these stem cells continue to renew themselves and differentiate into all your myeloid and lymphoid cells. So imagine you have a SNP that affects the hematopoietic stem cells. And this SNP skews the stem cells towards the myeloid lineage than the lymphoid lineage. It means if I'm making 70 billion neutrophils every second, you probably are making 3 times that. So it is not at the extreme end of things. So you are not going to see disease right away. But over the course of life, these cumulated differences, they result in disease. So by the time you are 50 and over, this low-grade myeloid skewing produces inflammation that can lead to disease. And so this is kind of controversial. When you talk about cognitive function and you associate it with genetics, people begin to worry because, especially in the US, because of the history of eugenetics, where people were hired, they would test your IQ before they give you jobs. And Yale in particular, there was geneticists who were so much involved in this eugenics. And even right now, people choose their embryos. You can genotype the embryo and predict their potential IQ. So I want to give birth to very intelligent children. I genotype them, know whether the child is going to. People do that nowadays. So, but the reason why I study cognitive trait is not to use it to categorize people. It is because cognitive function has pleiotropic effects. If you have higher cognitive abilities, you are likely to have higher educational attainment. You are likely to make good decisions. You are likely to have better health outcomes. And each of these are heritable. Your personality trait, they are heritable. And cognition seems to influence all of these things. So cognition is like a typical example of pleiotropy. And I'll show you, this was a study that came out early this year from David Reich's group. And this guy has actually influenced some of the work that I do. He studies evolutionary genetics. So what they did was they took ancient DNA as old as 30,000 years ago, 15,000 years ago, up to present time. And then they look at SNPs in ancient DNA. So they map their change in frequency over the past 12 millennia. Like the last 12,000 years, the SNPs that have changed in frequency, that is consistent with selection, not by chance, not drift, or maybe migration. The change in frequencies of these SNPs are due to selection. So they found that SNPs that are associated with higher cognitive performance, their frequencies have been increasing over the last 12 millennia, whereas SNPs that are associated with conditions like your weight, your psychiatric disease risk, and those other ones, they are going down. So this is to show how important cognitive function is, because it regulates so many systems. So here we wanted to find out whether cognitive function affects hematopoiesis. And like I showed you earlier, there are so many hematopoietic traits: your lymphocyte count, neutrophil count, your leukocyte telomere length. So first, to know whether cognitive function correlates with hematopoiesis, we did genome-wide genetic correlation between these cognitive traits and multiple hematopoietic traits. So you can see if you take something like chronotype, you know there are some people who are morning person, evening person, very little correlation with hematopoietic traits. Whereas if you take something like height, the same thing, height only correlates with like just two hematopoietic traits. But if you take something like educational attainment, executive function, intelligence, you see that they correlate with a lot of hematopoietic traits. Typically, we study cognitive function in the context of brain neural networks, but new studies suggest that the immune system contributes to cognitive function. And then to further understand how cognition affects hematopoiesis, we took the genome-wide association studies for these cognitive traits and then took single-cell attack sequencing data. So single-cell attack basically maps regions that are open, where the chromatin is accessible, because most of the SNPs that are associated with these complex traits, they are non-coding. They regulate gene expression in specific cell types. So when you overlay the cognitive traits GWAS with single-cell ATAC-seq data in hematopoietic cells, from the stem cells to the mature cells, you do see enrichment in some of the hematopoietic progenitors. So this demonstrates that cognitive function affects hematopoiesis. Yeah, so now I'm going to use two diseases as examples. If you take patients with type 1 diabetes or people with cardiovascular disease, they tend to have cognitive deficits or accelerated cognitive decline. And I'm particularly interested in type 1 diabetes because it's mostly diagnosed in children. And most of these children, they have cognitive deficits. If you are older and you have cognitive deficits, the age becomes a confounder. People don't know whether it's the aging that is driving it or the disease. So here we have two good comparators, type 1 diabetes and then coronary artery disease. So both diseases, patients with these conditions, they do manifest deficits in these cognitive domains. Okay, it worked. Okay, so what you are seeing here is another genome-wide correlation. They have put the full meanings of the abbreviations over there. So the message here is that the cognitive traits, they have an inverse genome-wide genetic correlation with type 1 diabetes. And then the polygenic overlap, that's a different statistical tool called MEXA. You can use that one to quantify the number of variants that are shared between two traits. So the Venn diagram, the intersection is where the variants are shared. So if you want to know the number of variants that are shared, you multiply the number by 1,000. That will give you the number of SNPs that are shared between the two traits. And then the RG, that's the correlation. You see that it's negative. And then when you do a Mendelian randomization, where you take whether having type 1 diabetes causes cognitive deficits, you do see that there's no difference. On the other hand, if you take type 1 diabetes as the outcome, you do have, if you have higher cognitive abilities, it reduces your risk for type 1 diabetes. And then if you do a conjunctional FDR to identify specific SNPs that are shared between the two traits, you can see here like different cognitive traits. So the conjunctional FDR is like if I tell you that there's a thief in New York, go and find them. And then I tell another person there's a thief, go and find them. The one I told them that the thief is in New York is likely to find them first. So if you want to find SNPs that are shared between two traits, this tool allows you to find a SNP that is associated with the second trait on condition that it is associated with the other trait. So it's a very powerful tool. You can see so many SNPs that are shared between type 1 diabetes and these cognitive traits. Okay, so here, these are just a few examples where we are overlaying the GWAS for the cognitive traits with equatorial in brain cells and immune cells. So you can see that there are SNPs that are expressed in both brain and immune cells that are associated with both type 1 diabetes and cognitive traits. and then the coronary artery disease part. We did a similar thing. You can see the inverse genome-wide correlation between the coronary artery disease and the cognitive traits. And then when you do the mixer, you can see so many variants that are shared between the coronary artery disease and the cognitive traits. This one, we included the total cortical surface area of your brain. You can see the negative correlation. That was you can see coronary artery disease behaving like a neuropsychiatric trait. Even we did so many brain regions, you do see the negative correlation. People who experience things like heart attack, they normally tend to develop cognitive decline. And then clinicians interpret it as the disruption to blood flow to the brain that is causing the cognitive decline. But we took a different approach. What if these are all complex traits that have so many variants, so the probability that they share variants is higher. So you can see that there might be people who are already vulnerable to cognitive decline before they develop myocardial infarction. And here, just like in the type 1 diabetes, you can see the conjunctional FDR identifying so many SNPs that are shared between CAD and the cognitive traits. And this is the beautiful part. So the guy who did this is actually a first-year undergraduate student. So what he did was he took the GWAS for the cognitive traits and then overlaid them with chromatin accessibility in immune cells. And then he picked only the SNPs that are enriched in immune cells to develop a polygenic risk score. Then he goes into the All of Us. The All of Us is a US study where they have recruited over 700,000 people. They've been following them for so many years. Their genotype, their health records are available. So he develops this polygenic risk score based on the enrichment of the cognitive traits in immune cells to use that to see if he can predict who is likely to develop myocardial infarction, like heart attack. So he followed them for 30 years. So at baseline, nobody has myocardial infarction, but they are grouped into decile based on your score on the cognitive trait enrichment in immune cells. So you can see here, if you are in the 90th percentile, like you are in the top of the cognitive abilities, versus if you are in the bottom, you see the hazard ratios. Those who are in the first decile, like they score very less on the cognitive traits versus those who score higher. You see the massive differences. And this is where medicine is going. There are diseases that clinicians often misdiagnose. So if you combine symptoms with these polygenic risk scores, you are likely to be able to accurately predict, diagnose the disease properly. Or if you have a new drug and you want to test whether it can prevent disease, you can use the polygenic risk score to predict who is likely to develop myocardial infarction, and then you intervene with those therapies. I was chatting with somebody yesterday about how people look lean and then they develop type 2 diabetes. So people just assume that if you're obese, then you'll develop type 2 diabetes. It doesn't work like that. And you can't use symptoms to diagnose this, but these polygenic risk scores, they are fixed from birth, from conception. They don't change over time. They are not confounded by the environment. So they are able to give you an accurate prediction of somebody's risk for a disease. Okay, so I think I have a lot to talk about, so I'm just going to skip this part. So the message, so we actually have a perspective that will soon be published. We are trying to reframe how to interpret cognitive function. Historically, we interpret cognitive function from the perspective of neural networks in the brain, but there's a bidirectional communication between the brain and the immune system. So we are writing an article on neuroimmune circuits that regulate cognition. So if you inherit the risk for, say, type 1 diabetes, the risk is not just affecting immune cells, it is disrupting these neuroimmune circuits. So you develop type 1 diabetes and you're also vulnerable to these cognitive deficits. The second part is the part that I like the most, because it is, I came up with this one because it aligns with the theme of this talk. Because, so, you know, humans used to be, from a genetic perspective, we all came out of Africa. Like the movement out of Africa went to Europe and all of those things. So those who left Africa, they faced bottlenecks. So they were kind of perked. Like you needed to have some SNPs to overcome some of the harsh conditions that they met when they left Whereas those who remain in Africa, we didn't meet some of those bottlenecks. So we maintained the diversity of our genomes. Whereas those who left, there was serious purging. So they are more homogeneous than us. The argument I'm trying to make is to demonstrate how important it is for Africans to be included in genomic studies, because we bring this diversity of genomes. It takes somebody from Nigeria, somebody from Ghana. When I did my 23andMe, nobody knew where I was from. The results came, they said, you are 85% Ghanaian. Isn't that amazing? So here, about 12,000 years ago, human beings started domesticating crops, keeping animals at home. And it first started in Asia and then Europe, whereas Africans were still doing hunter-gathering. And so with this change in diet came metabolic pressure. People had food readily available. They needed to digest the carbohydrates that they were taking. So if you take people in Asia and Europe, they have developed some changes in their genomes that are absent in Africans. Most of you probably know by the time you are 40 years, you have lactose intolerance. So that is because there's a SNP that activates the lactase gene that allows you to metabolize lactose. But because these people domesticated animals early, they were taking a lot of milk, that SNP activated their lactase gene so they can metabolize milk. Whereas very few Africans express the lactase gene. So I have a lot of friends who are lactose intolerant, and it's because their lactase gene is inactive. And then the same thing with some of the genes that are involved in lipid metabolism. They have undergone selection in Asians and Europeans, and even the AMY gene. This gene is produced in your saliva that allows you to digest starch. So these are well-established genes that have undergone positive selection, because when humans adopted and domesticated crops, they needed to have these mutations to be able to survive. So we identified a gap. What's the essence? If you break down starch into sugar, but you can't metabolize the sugar, and we know that insulin is very important for the metabolism of sugar. So we decided to test whether the insulin gene has also undergone selection in the past 12 millennia. Like, by that I mean 12,000 years ago. So we looked at, there are about 30,000 ancient DNA from, there's a group at Harvard, they have been able to create ancient DNA that are up to 30,000, spanning 20,000 years to present. So we looked at those ancient DNA sequences and looked at SNPs in the insulin gene to see whether their frequency changes is consistent with selection. So we found this particular SNP that has been increasing in frequency over the past 12,000 years. And the regional plot in B is just demonstrating that it is pure selection. It's not just drift or by chance. And then we find that this SNP is already known to be associated with type 1 diabetes risk. So here you can see that the same SNP that is associated with increased risk of type 1 diabetes is the same SNP that has undergone the positive selection. So that was kind of surprising to us. Why would a SNP that causes autoimmune disease undergo positive selection? So this SNP in the insulin gene, there are 3 of them. They are in near perfect LD, so they are normally used interchangeably. Before then, we already knew that Africans, like the insulin gene, it has a variable number of tandem repeats in the 5' region. So depending on how many repeats you have, you are classified as class 1 or class 3. And the class 1 is a short repeat. That one is actually with higher risk of type 1 diabetes. Africans don't have a lot of the class 1. So historically, the reason why this SNP causes type 1, they think that it reduces the expression of insulin in the thymus. So the T cells don't get educated to insulin. So when the T cells come into the circulation and they meet the beta cells that produce insulin, they see them as foreign and attack. But there are emerging evidence that these SNPs are actually active in the beta cells themselves that produce insulin. So I tend to agree with the second model, where the SNP, people think the beta cells are passive in type 1 diabetes. They are active participants. They contribute to their own demise. So we think that the SNP is increasing insulin production in the beta cells. So although it is associated with type 1 diabetes, it may have metabolic effects especially if it was positively selected around the same time that humans adopted agriculture and were eating carbohydrates, it will allow you to produce more insulin. So postprandially, you are able to reduce those glucose excursions. And insulin is very important for fetal growth and development. So we went into the UK Biobank to test for the association of this SNP with metabolic traits and the different types of type 1 diabetes. So you can see the SNP, after the HLA region, this SNP actually has the second strongest effect size on type 1 diabetes. So you can see both late onset diabetes in adults and then the typical type 1 diabetes, their risk is increased, whereas that for type 2 diabetes is reduced. And then if you look at the metabolic traits, like random glucose and A1C, which is typically used by clinicians to measure how well you control your glucose, and then IGF-1, you see that all of those ones they have these opposing effects, like the metabolic traits, they are enhanced. And then you have the age at onset of type 1 diabetes is delayed, which is consistent with when you talk about evolutionary genetics, there's something called antagonistic pleiotropy, where SNPs that help you when you are a child, they are positively selected. Imagine a SNP has effects only in old age, let's say Alzheimer's disease. But by the time I develop Alzheimer's disease, I've already produced my children. So they'll continue to propagate those SNPs. Whereas if I have a SNP that affects me when I'm a child, I will not live to bear children. So that SNP over time will go away from the population. So this makes perfect sense. It increases type 1 diabetes, all right, but the age at onset is delayed. And we know recently more people are getting diagnosed with type 1 diabetes in 30, 40 years, unlike what we used to think that type 1 diabetes is a disease of children. Yeah, so this is just a schematic of the next experiment that we did. We were lucky to belong to some consortia that have transcriptomic data in human islets, single cell transcriptomic data in islets. So we wanted to know how these SNPs affect insulin secretion and insulin secretion and expression of immune markers in the beta cells. So here, this one is showing the allelic specific expression. So the SNP, if you have it, you normally have 2 copies of each gene. So you can see that, let's say you are heterozygous for the SNP. So your RNA that is coming from the wild type is even more than the RNA that is coming from the DNA that has the SNP. That was quite surprising. But then, if you look at insulin secretion markers, the red ones are increased insulin. Like if you stimulate the beta cells or the islets with glucose, how much insulin they produce in response. The SNP increases the insulin production, although at the RNA level it's slowly expressed. And then here too, ER stress is one of the early markers of type 1 diabetes. So when we look at the effect of the SNP on ER stress markers, you can see here, like all the typical ER stress markers, the SNP increases their expression levels. And then we also quantify immune visibility markers, like genes that are involved in antigen presentation and all of those things. And then we do a correlation between the expression levels of the ER stress markers and these immune visibility markers. So you can see that if you express more of the ER stress markers, your expression levels of these immune visibility markers too are increased. And then you can see the bottom here, this is just showing the dose-dependent effect of the SNP on the expression levels of the insulin RNA, dose-dependently, or the ER stress markers. And then, so I have a friend, he's been going around the world excavating people, including archaic humans. So like modern humans move out of Africa. Before then, there were other archaic humans, the Denisovans in Asia, and then the Neanderthals in Europe. But those ones went into extinction, but their DNA remains are still there. So we wanted to know whether this SNP is modern. So we looked at chimpanzees, and were able to get about 6 high coverage DNA sequences from these archaic humans. You can see that the SNP is modern. It's not present in those ancient humans. It's only unique to modern humans. And then if you look at the distribution of the SNP across the globe, you can see Africa in blue. On average, 18% of Africans have it. Meanwhile, it's almost near saturation in East Asia. And then East Asians actually moved to Latin America. They are mixed Americans. It was people from, there was a bridge called the Bridge of Beringa. Is the ones in East Asia, they moved there. So you can see that the high frequency in mixed Americans, it means that before they left that place, they already had the SNP. And then even within Africans, you can see that those in the Caribbean and those in America, African Americans, they have almost 30% frequency. Like the SNP is present in like 30%, whereas if you take people like the Esan people in Nigeria or Yoruba and other places, the frequency of the SNP is relatively low. The point here is that geographic location can influence allele frequencies, and that can also determine your risk to disease. Yeah, so the insulin pathway it has three main ligands, the IGF-2. This one too, the IGF-2, normally when you are a fetus, you express a lot of it, but when you grow up, it becomes silenced. It's very important for growth. And you normally express it from your paternal genes. So we were like, why would it end at insulin? It has to be an old axis, the insulin IGF pathway. So we do the same thing for other genes that are involved in the insulin signaling pathway. So you can see IGF-2, one SNP that is undergoing positive selection, the other one is going in the opposite direction. And you can see that the one that is positively selected, it reduces the expression levels of the IGF-2. We're lucky that SNP is an EQTL, just regulating the expression levels of IGF-2, which makes sense. Because if you express IGF-2, you probably will be like a giant, like you overgrow. Then IGF-1 begins to be expressed when you pass the fetal stage. So you see that children who have growth deficiency, they normally give them IGF-1 to enhance their growth. Yeah, there are interesting things about this particular gene, because if you are a mother with type 1 diabetes, you are less likely to pass it to your child. Whereas if you have a dad with type 1 diabetes, you're likely to pass it on to your child. And interestingly, these IGF SNPs are normally expressed in the paternal gene. So this is a new opportunity for us to understand how this might contribute to the disproportionate transmission of type 1 diabetes to the progeny. So same thing, the signal that has undergone positive selection is the same signal that is associated with type 1 diabetes. I'll show you the association data. We did the same association for metabolic traits and then type 1 diabetes and type 2 diabetes. The pattern follows. I'm just showing the SNP that is positively selected. So in the top is the positively selected one, then the bottom is the negative selected one. So they have this antagonistic, like opposing effects on type 1 diabetes, type 2 diabetes, and then the metabolic traits. But this one also includes red blood cell traits. It enhances hemoglobin concentration and most of the red blood cell indices, which we know are important for oxygen delivery, fetal development, and all of those things. So it's like the shift in diet has influenced the insulin signaling axis. And this one too follows the same frequencies. The SNP is lower in Africans, whereas it's more prevalent in other populations. And the timing coincides with the adoption of agriculture. This kind of study is not easy to demonstrate in the lab that, yes, it's the adoption of agriculture, but like everything looks perfect. The timing, when the SNP started increasing in frequency and everything follows the same order. So so the reason why I like this part was because it emphasizes how it is important to study genomics in every context. Like there are some SNPs that are not present in some populations, and that can predispose you to certain disease compared to other people in other geographic locations. Yeah, in terms of one of the things that I've been very passionate about is why don't we have a biobank? Like, let's say there's a biobank affiliated with the University of Ghana Medical Center. People consent. We take their saliva. It does. These days, genotyping is very cheap. The chips are there. Just genotype them. Then we create something like a touch screen. The questionnaire is there. Have you ever been diagnosed with this? How many hours do you sleep a day? They just put in there. And then people just key in their medical records. We don't disclose your name. We just give you an ID. And then it gives opportunity for guys who are interested in bioinformatics. They manage it. And then scientists can also use those data to see how genotypes this wouldn't cost up to a cathedral. It will. Yeah, it wouldn't cost. We can't rely on the other genomes like UK Biobank. If you take the UK Biobank, 95% of them are British. All of us, they try to include, but like I showed you earlier, African Americans, they are becoming more and more different from those in Africa here. And most of the genomic studies, when they say Africans, they are always referring to African Americans. It's not we those here. So I think that it's good to have things like, it will create a lot of job opportunities. I pay $11,000 to access the UK Biobank data. I cannot download them. I do the computing by cloud. And then every minute they charge me in addition to paying to have the access. And African genomes are a hot cake in genomics research. You write a grant, you don't make it inclusive, reviewers are going to say it's not diverse. So you normally anticipate that and say, wherever possible, we will. So having an African biobank where we genotype them, we take their medical health records, we will generate income from it, because all over the world, people want to include African genomes. If you understand how GWAS works, the more diverse you are, the more you are able to discover new genetic variants. So I would like to end here and to thank you guys again for the opportunity, and I hope I'll become an affiliate member of. Actually, my undergrads, they wanted to come to Ghana, so I told them maybe next time write an abstract submit for the WACBIP conference, and then so hopefully we'll show up the whole lab for the future lab.
Excellent.
Thank you.
Excellent, right? Amazing stuff. I hope the media are not here. The cathedral comment, tomorrow you may see it in the headlines, that scientists at WACBIP takes a shot at the MPP government for building a cathedral instead of building a human genome database. Anyway, I have good news for you. One of our colleagues, Yao Bediako, who started Yamachi, he's thinking like you, so you guys should have a conversation. He's interested in that money you are talking about. So we are waiting for our 1% when he hits the jackpot. Anyway, colleagues, I think that this has been excellent. There's so much knowledge that David has delivered here, and I'm so happy for the young people here, the students, to learn so much from what he has said. And I think we have to arrange for you to give some lectures here on human gene variations. Really, really interesting. Yeah. So anyway, let's take a few questions and then we can move quickly to the next session. Yes, there's a hand at the back.
Yeah, thank you very much, Prof. David. I have some few questions. The first question is, when you identify the pleiotropic genetic signals across different diseases, how do you distinguish a biologically meaningful shared mechanism from a purely statistical overlap? And how do you choose the first experiment to validate these relationships? And then I want to also ask, is there any relationship between a type 1 diabetes and then someone's eager to eat more sugar? Because I'm coming from a background where I have my grandparents, they were 10, all of them were having type diabetes, and 9 of them is gone. So I want to know sometimes my mom will just advise us not to eat more sugar, but the zeal for eating the sugar is always there. So I want to know, maybe is there any relationship between my appetite for sugar and then the way I choose my food related to this genetic background that a person might have?
Okay. Thank you. Yeah, you can respond to that.
Well, I'm glad you talked about your zeal to eat sugar. And that's the more reason why I try to emphasize cognition. Like something like executive function is basically measuring your ability to not be impulsive, not to give in to your urges. And so I will encourage you to do more reading and learning. When you learn, you activate those same cognitive pathways and you learn to. But to answer your question about eating sugar, I think there's a myth to it. If you already have diabetes, whether it's type 1 or type 2, it's not a good thing to eat more sugar because already you can't metabolize the sugar. But I don't see anything wrong with eating regular sugar. But in life, you should do everything in moderation. Don't do it excessively. But just putting sugar in your coffee, if you don't have diabetes or if you're not pre-diabetic, there should be nothing wrong. Your pancreas is functioning well. But there are a lot of people walking around, they are pre-diabetic. If you look at their A1C, it's like 5.8. In that case, you already know that you have a problem metabolizing sugar. So you wouldn't want to eat more sugar. But where you don't have that kind of condition. So there's a myth out there that if you eat sugar, you develop diabetes. It doesn't work. It doesn't work like that. Then your other question about how do you understand the mechanism of a pleiotropic variant. So my lab, as we speak right now, we do have we've created isogenic cell lines for some of the SNPs that we found to be associated with multiple traits. For instance, like one of the SNPs I showed you here, where it is expressed in brain and immune cells and it associated with type 1 diabetes and some of the cognitive traits. We were able to use CRISPR to induce the same point mutation. We have the wild type, the heterozygous, and then the homozygous isogenic cell lines. And we now make a mechanism and say, okay, this gene traditionally is involved in mRNA translation. So maybe the SNP is influencing the efficiency or the fidelity of translation. So we insert these cell lines with constructs that can quantify these readouts, like translation, fidelity, and those things. And we choose the cell lines based on their relevance to the disease. So we will see. Those are some of the functional validators. And we use mouse models. We have a mouse that spontaneously develops type 1 diabetes. It has some of the SNPs that humans have. The clinical features are similar to humans. So we have another study where we have generated a knockout mouse based on these genetic findings. And I'm personally interested. I'm more like, you see me taking courses in from academia to industry. So when I do any projects, I always ask from the beginning, what's the exit plan? So for all these things that we find, we always have a way to functionally validate it, because if you want to go to venture capitalists and then convince them to invest in your idea, you need to have convincing data. So when we find a pleiotropic variant, we study it, we induce it with CRISPR-Cas9 in cells that we think are relevant to the disease, and then see where the SNP actually exerts its effects.
Yeah, David.
Yeah.
I'm here.
Oh, okay.
Yeah, I mean, that was absolutely brilliant, and thank you. And the observation that diet actually shapes these SNPs really strongly correlates with what microbiome experts are saying, how the microbiome composition literally influences all these traits. So definitely. But the question that I have, how do you think these microbial organisms that live within our systems are influencing how these SNPs develop? Do you think there could be epigenetic, or what's your thinking about how these microbes are influencing our genetic changes? Thank you.
Yeah, thank you for the question. And that's the more reason why, imagine our children, 1,000 years to come, they would wish that they have access to our genomes so that they know where they are coming from and where they are going. These SNPs, they keep changing in frequency because of our lifestyles. The SNPs that give you a survival benefit. So there are a lot of people who die when they feed their children. They haven't given birth yet. There are because we don't have access to their genomes, we don't know why they do not live to procreate before they pass. So in a way, those who have some SNPs that allow them to procreate, their SNPs keep on increasing in frequency. So the gut you talked about is a really, really interesting area. The gut definitely shapes a lot of things in your physiology. So I wouldn't be stopped. No one has studied that, but I think the gut most likely can influence how these SNPs change in frequency because of the foods you eat, which can in turn influence your gut microbiota. So it's like a cycle. Whatever you do, something must change. The system has to be in equilibrium. So if you are putting things in your gut that is changing your microbiota, then you are creating some pressure. So you don't see the effect immediately, but we have people dying earlier because their gut microbiota is not compatible with longevity. So those who do not do that and live, their SNPs continue to increase. So those are the mechanisms that you can have. These days we don't experience the serious bottlenecks that our forebears experience, but these things like our lifestyle and those things, so yes, the guts can influence the changes in frequencies of these SNPs through these things.
Alfred.
Okay, I have the mic, so.
Who has the mic?
Yes,
Oh.
I do. Thank you. Yeah, so I'm Stephen Amma from UCC. I really like your talk because I'm also interested in the neuroimmune axis. So during my PhD, we observed that while the literature shows that first episode schizophrenia drug-naive patients seem to have microglia activated, for example. And with my PhD, where we observed that some microRNA was able to mediate the inflammation and then neuronal axis, as well as my microRNA of interest, although it is, elicited by inflammation, it seems to have an anti-inflammatory effect. And then you also have downregulation of some of the NMDA receptors, which have been linked to psychosis. So I really like what you're doing, and I'm wondering whether you're also exploring some of the non-coding genome, microRNA, circular RNA, because I think some of them could clearly establish some of those links so that hopefully targeting them as potential druggable agents could also enhance outcome in this field. So hopefully we may talk a little more to see how some of this could be explored further. Thank you.
Right. So that's the comment, right?
Yeah.
So let's take Alfred.
Yeah, I'm not sure this is working. Mine, I just more like comment and at the same time question. Now, yeah, it was interesting to see the trajectory of those SNPs in the populations you've analyzed. So, given the absence of African data in most of these human genetics resources that are available, I wonder how much African data was in that trajectory. The second part of that is, as you said, rightly said, farming started much later in Africa, and you could see that some of these SNP, they got selected for just when farming started, or domestication of animals also started, which happened much later in Africa. With the heterogeneity of that starting in Africa, and actually some populations in Africa are not still farmers, they're still gatherers. I wonder whether you could actually separate the trajectory of selection of these in these populations, or whether there's already evidence that some of these populations that are remained farmers and gatherers have not had this selection happening in their populations, and they definitely are not at risk of some of these disease traits that we are currently seeing in our populations. Yeah. Lastly, tied to Jerry's question, is the impact of our exposome to infectious pathogens and their relationship cognitive effects, our cognitive traits. There's been a lot of talk about malaria and cognitive traits in Africa. Do you see that, is it possible that there could be some relationships as well between our exposome to infectious diseases and some of these cognitive disorders, and that can there be SNP markers in the genome that could be picked up with the same approaches, pleiotropic approaches that you're actually using at this moment? Yeah.
Very good. I like your first comment. So my friend Diando he's an assistant professor in the genetics department. He's originally from Gabon, and I brought him on board. So he actually did the archaic human genome analysis. He had the same thing, like, within Africa, we have people who adopted agriculture much later. Even there's a place in Tanzania also where we still have people who are doing gathering. So he was like, what if we look into these subgroups in Africa to see the frequencies of the SNP. But I'm a very aggressive person. I want this work to be published. I'm like, we have enough evidence. Maybe we can do that later. But that was a very good point. And I believe if we do that, you can even see the SNPs that I showed here is from the All of Us and then the 1,000 Genomes Project, which included Africans, but not so large. And for the All of Us, when I say Africans, based on their genotype, you can tell whether they are Yoruba or those things. So we included those ones, not a big sample size compared to the other ones. And about the infectious pathogens. So type 1 diabetes or autoimmune diseases in general, they arose from infections. So the HLA and most of those genes, they are involved in response to infections. So you see that if you go to Europe, there was a plague. It has a name. I've forgotten the name. It influenced the selection in the HLA region. So you see some of these HLA polymorphisms, they are common in Europeans. Type 1 diabetes, when I was undergrad, I didn't even know what type 1 diabetes was. But in the US, every now and then, I've worked with at least 4 college students who all have type 1 diabetes. Even in Africans, when you find someone with type 1 diabetes, it's not so severe like in Europeans. Or if you go to Northern Europe, it's even worse. So the HLA polymorphism, they allow you to launch a very robust immune response against infections. But this robust immune response comes at a cost of autoimmunity. You are likely to attack your own cells. So yes, most of the variants that are associated with autoimmune diseases, they have undergone selection in response to pressure from infections. Yeah, so we thought that's what is novel about our work. People haven't really associated with metabolic pressure with autoimmunity. This is probably one of a few to tie metabolic adaptation to autoimmune, but most of the autoimmune diseases, they have been associated with infections. Infectious disease and autoimmunity, they are the same. They are basically very similar. Yes, so like the gut microbiome can influence selection through how you respond to infections and all of those things.
There's still hands up. Meanwhile, we are over time. So quick one, 30-second questions.
Yeah, so this will be quick. I may have missed it, but can you elaborate how you quantify or stratify the cognitive traits, especially something like the cognitive versus non-cognitive education attainments and others? Yeah,
so educational attainment is just based on years of schooling. So if you finish GSS, maybe we'll give you 10. If you finish high school, 12 or 15. If you finish college, 17. If you could finish university, 20. That's the only readout. And then we just genotype you and see which SNPs are associated with the years of schooling. And we know that cognitive abilities contribute to educational attainment. So we do a second GWAS by subtraction. So there's an equation, there's a tool called genomic structural equation modeling. So you subtract, so let's say I do a GWAS, I give you tasks that involve learning, and then I see your IQ. Then I subtract the SNPs that are associated with the IQ from all the SNPs that are associated with educational attainment. So whatever is left is a non-cognitive part of the educational attainment. And then the cognitive performance that are subtracted becomes the, and it's very interesting, I'll tell you, when we do Mendelian randomization, non-cognitive educational attainment or executive function, if you have higher executive function abilities or higher non-cognitive educational attainment, it significantly reduces your risk for heart disease. On the other hand, it doesn't have an effect on type 1 diabetes. But if you take the pure cognitive traits like fluid intelligence, you'd see that they rather have a marked effect on type 1 diabetes. Why it makes sense? Heart disease is mostly about life choices. You give in to smoking, eating fatty foods, you don't take good care of yourself. So if you have poor executive function, you do those things. Whereas type 1 diabetes develops in childhood and coincides with neurodevelopment. So that's the thing I like about using these genetic variants as proxies for these traits to understand how they relate. But that's how we measure the cognitive versus non-cognitive education attainment.
Okay, last question, Yao, and then we have to go.
So thank you, really fascinating talk. One is a comment. So over the last several months, we've just done about 200 whole genomes from African people. So it'd be great to talk to you about how we could begin to look for some of these SNPs. The other question is, you mentioned the value of African genomes. Obviously, we need to make the case that African genomes are useful to global drug discovery. And I'm wondering, so far what you've described are SNPs that cause disease primarily in Europeans, and it seems like in Africa we have lower frequencies of those SNPs. Are you looking at maybe the reverse, where there are maybe protective variants in Africa, a bit like PCSK9? Because that then really makes the strong commercial case that there's something in Africa that can, because the truth is the money is there, they care about themselves, right? Without putting it too bluntly. So if there were protective variants in African populations, that could then be used for future drug development. Do you have any examples or do you have any thoughts about those?
Well, not necessarily protective variants that are unique to Africans per se, but even the differences in their frequencies alone is protective. And you can imagine East Asians, 94% having a SNP, and less than 20% of Africans having the SNP. But there are so many SNPs that are, we actually have another SNP, it's called, it's in the transcription factor, the Icarus. Those who study immune T cell, the Icarus transcription factor. This SNP is present in like 0.005% of Africans, whereas it's present in like 20% of admixed Americans. So this SNP increases their risk for acute lymphoblastic leukemia. And I live in Boston and like New England. And I do work with hematologists. I actually started from liking hematology before I moved into this area. So I talked to some of the hematologists. Most of the patients they see are immigrants from Latin America, like most of the patients who have the acute lymphoblastic leukemia. Whereas this SNP is literally absent in Africans and Europeans. So there are several, and this transcription factor there are drugs, they are called immune modulatory something. They are example like lenalidomide, iberdomide, and those things. They target these transcription factors. So I mean, these are opportunities for drug, but like to just say a SNP is present, or maybe something, I can give an example like, there's a SNP in the chromosome 9 that is associated with heart disease in Europeans. Africans can have it, but they don't develop heart disease. Even the ApoE variants, there's evidence that Africans may have the variant, but the penetrance is very little, and they don't. So, and that is because genomics is very complex. Things like epistasis, gene-gene interactions, we have polymorphism in other areas. Yeah.
Okay. Right. So the question that I have for him for coffee break is, why is it that the people with high IQ, they are usually weird? So the weirdness, how is that, inherited together with the cognitive function? So this is for coffee break. So we'll talk about that at coffee break. So let's thank David very much. Peter, take over.
Good morning once again. So we'll now continue with the pathogen biology, genomics and bioinformatics session here in the conference hall. And this will be chaired by Reverend Dr. Winfred Seth Bewonyo and Dr. Gloria Megache. And then concurrently, a parallel session will be held in the PhD lecture room with Dr. Collins Moranga and Dr. Christelle Choba as the session chairs. So those interested should kindly make their way. We'll all reconvene here at 12:45 for the second session of day 3, which would be on vaccines and immune-based biotherapeutics. So ushers are outside to usher those who wish to attend the parallel session to the PhD lecture room. And can we have our session chairs here to begin day 3, session 2? Thank you very much. Prof. Kwashi, do you have anything else to add?
Good morning. So there's, if some of us can go to the parallel session, which is directly opposite this room, and then we come back here after the parallel session. As I mentioned, we are trying this out and seeing how it's going to go, whether we are going to scale it in future. Let me just let me just there's a second. So it will be the first parallel session, tracing microbial footprints in non-communicable diseases. So it will be still heavily focused along the same discussion as the keynote. And then this session in here will be, let me just make sure. Wait, sorry. This session in here will be pathogen biology, genomics, and bioinformatics.
Okay. So Jasna, who is presenting on, I think he will.
Okay.
I can't handle this. The topic, I'm sure, will come.
Yes.
Hey, where are they?
Oh, yeah, there we go. All right. Good morning, everyone. I think, yes, I'll introduce my topic, which is mechanisms of emergence and spread of Artemisia. Microphone, yes. So, yes, I thought I would just introduce mechanisms of emergence of Artemisia multidrug resistant malaria parasite. So, just to introduce myself, that I'm a Wellcome Trust Early Career Fellow. This presentation is just to give you an overview of what the project entails and what we've been doing as a preliminary work to sort of be able to get the project going. So, how do I?
Point to the back.
The back.
The back. Where the guys are.
Oh, okay.
You want the forward arrow? Forward
Yeah.
arrow.
Yeah. Oh, there we go. All right. So we all know about malaria, so I'm not going to talk a lot about this, but it's just to highlight that drug resistance has been an issue in Africa, and Prof. Alfred has talked a lot about this, so as I said, I'm not going to. But it's just to highlight that we started with chloroquine and sulfadoxine-pyrimethamine as the first-line treatments. The parasite developed resistance, and then that ushered into the combination therapy, which Artemisinin combination therapies. We now have also resistance to the partner drugs during that period. And in the sort of third period that I've sort of highlighted as emergence and spread, we have the parasite has developed resistance to almost all the drugs, including Artemisinin, which has been which was first emerged in Rwanda and is now spread in most of East Africa. We are lucky to not to have it in West Africa, so that's good news. So basically you now have a parasite population that are resistant to all the drugs. And so you're asking the question, what is causing this resistance? How has this parasite emerged, evolved over this period of time now that we've changed the drugs? One of the things that we want to, if you look at the characteristics of this emergence, you can see that it coincides with a declining transmission of the parasite. So in areas where the parasite transmission has really declined, you do have the multi-drug resistance emerging. In typical areas like Thailand, where is the epicenter of this drug resistance, and also in East Africa. And so what we want to understand is that what are the mechanisms that are driving this resistance? So I'll just give a highlight here, maybe for Ghana. All right, I think I missed this slide. All right, good. So I'll just talk about that these drug-resistant markers are very important for monitoring efficacy to anti-malaria drugs. And so it's very, very important that we do validate them. We continually validate them in different transmission settings. There are some markers that were validated in Southeast Asia. They might not be necessarily useful for West Africa or even particularly Ghana. And so it's very important that we do validate them. And I've highlighted here some of the drug-resistant markers that the classical drug-resistant markers that are being used to monitor the resistance in the parasite population. These are the classical resistant markers, but a lot of speakers have thought there are now a lot of background resistance, resistant to a lot of these drugs. And so one of the things that we want to understand is what are the background resistance genes that are involved in driving these multi-drug resistant markers. And what we want to do is we've been doing some surveillance in Ghana, so this is not just my work, but this is just a group of the malaria researchers in Ghana who have been doing a lot of the surveillance. So in Ghana, we have 3 transmission zones, and I've highlighted them here: low transmission, very high transmission in the middle, and then also in the Savannah region, which is basically seasonal transmission. And here we've highlighted here a lot of the K30 mutations that have been detected across these transmission zones. But the beautiful thing is that if you look at this transmission zone, you can see that so Michael Forey and co have done a lot of ex vivo resistance work, and they've shown that across this transmission zone, there's a sort of heterogeneous resistance to some of the drugs that we are using. So this is just ex vivo assay. What we want to understand is what is also happening at these mutations conferring resistance within the parasite backgrounds that we have in Ghana. And so my project for the next 5 years is to... So yes, so with the Work and Trust project, so I'm very thankful, Ben is here, so thank you very much for investing in this project. One of the things that we really want to be able to build is building capacity in sequencing in Africa. And one of the things that this project is really bringing to WAGBIB and to the University of Ghana is to be able to bring in packed biosequences. That's really the next 3rd generation sequences that we can be able to assemble the parasite genome. So we've been using short fragments to really assemble the genome. There's been a lot of AT rich in the parasite genome, and so it's very difficult to really assemble the genome. Packed bio nanopore provide a really advantage to be able to do that. One of the things that we want to do is to be able to build microhaplotypes to be able to barcode some of these parasites according to their drug-resistant gene and see how these microhaplotypes are sweeping through the population. So basically, you see these microhaplotypes. What we want to do is to be able to also be able to track their transmission. Where are these mutations emerging from? So for instance, some of the K30 mutations that I've highlighted, where are they emerging from? Are they indigenous or are they imported cases? And so we want to also be able to, traditionally, I think we've been able to, how are these mutations, how do we select this mutation and be able to study them? Are these mutations that I presented on the map, are they all conferring resistance? We probably don't know. So one of the things that we're doing is to be able to really do a hardcore population genetic to be able to see which genes are really under selection. And those are the candidate genes we're going to put in to be able to do CRISPR screens to see how they confer resistance. So we have a point where basically we can create a dress code, basically drug-resistant code, and then we can validate this, whether these drug-resistant codes are confer resistant. And so this is something that we have to design first for Ghana and be able to see also how it works for Africa. So basically we'll be doing a lot of genotyping and phenotyping in this project. So, as I said, there's a lot of resistant genes that have been associated, but they've not really been validated. And one of the things we really want to do is to be able to sequence them and see which ones are associated with artemisinin resistance. So here, I've just highlighted some of the work that we've just started doing with Prof. Nancy. Basically, Quisitando is doing this work where we are multiplexing a lot of these resistant genes and being able to sequence them using the PacBio sequencing. All right. So yeah, also just to introduce some of the work that we've been doing in Australia, I'll be able to develop, so I'm really interested in putting some of these assays into point-of-care diagnostics. One of the things we've been doing is to be able to detect mutations. So not just detect parastemia, so we can detect parastemia, yes, and that correlates with QPCR, but we also want to detect mutations. So can we take a point-of-care device to the hospital and tell that this patient does not just have malaria, but has a mutation to the artemisinin? So we've been able to do that. My students have been able to do that. We are yet to put this assay integrated into a point-of-care device to really be able to offer complete diagnosis at a point of care. All right, another preliminary study that we've been doing is that, so I was able to take some parasites from microflora. We started culturing that they are doing well. So these are clinical isolates that we've adapted into culture. They are really doing well. And one of the things that we are looking at is being able to understand the parasite drug resistance within this parasite. So if you look at this parasite, this is the reference strain, so basically 3D7, which is sensitive to most of the drugs anyway. And then this is the Southeast Asia Lancome 3.2, which is multi-drug resistant to almost all the drugs. You can see the Ghanaian lands are resistant to at least two anti-malaria drugs, which is beta sulfadosine pyrimethamine. And then also, most of them are also resistant to have copy number duplication in plasmepsin 2, which make them resistant to piperacin. So this genotype, does this correlate with the phenotype? So that's one of the questions that have been asked. So our master's students and a research assistant have been also doing a lot of drug screening to be able to see whether these phenotypes, this genotype correlates with the phenotypes. Here you do see that I showed that the pyrimethamine resistance, you can see, so that's what you see in a DHFR marker. You can also see, we are beginning to see resistance to lumefantrine. And this is a bit dicey because we think there are a lot of genes that are at play. So this is just a preliminary. But we also see resistance to mefloquine. So these are some of the things that we've started doing to be able to build a sort of biobank that we can be able to characterize most of the parasites that we have in Ghana. Yes, so we are also interested in, as I said, gene editing this parasite, so we can easily edit 3D7. It's not so easy to edit the clinical isolates, very, very difficult, but we're getting some success. So we've been able to edit a lot of the mutations that are unique to Africa. We'll be able to edit them into 3D7, so those are ongoing. As I said, with the Ghanaian lines that we call the P300 lines, we've been able to do some transfections. Last we had a news that we didn't have success, but we are repeating it. So it's not easy to really edit a lot of these clinical isolates. We think we have success because we've developed a method to do that. So I just want to also just do a bit of advertisement in a way that, so for the 5-year vision, we'll be looking at really be able to train PhD students and also master students. So if you're interested in sort of joining my group, I'll be interested to talk to you and to see what opportunities we can offer. All right, I just want to say thank you to my collaborators, my mentors. I want to thank you to especially my students who are doing a lot of this work. And I want to thank Wellcome Trust for believing in my vision and putting a lot of generous funding to really support this project for the next 5 years. So thank you.
Right, thank you very much, Charles. Questions for Charles, please? Right, so there's a hand raised there.
Charles, thank you so much. That was great work and great plans. One thing you said is something that I think others have experienced. It's very difficult, apparently, to do editing, genome editing, on clinical isolates, even though they're growing continuously in culture. That's a bit of a barrier, isn't it, for experimental work? And I'm wondering if, A, on one hand, do you have any hypothesis why that would be the case, why it's difficult? And secondly, whether you can think, or you have any plans to systematically try to work out ways of overcoming that, which would open up the field for gene and genome editing.
Yeah. Thank you, David, for your question. I think you've put me on the spot in terms of what do I think about what is I think field isolates one of the we've had a bit of challenge culturing them. One of the things that we've been doing is to try to culture them in a way that mimics the sort of natural with the blood infections. In terms of what might be at play, why they are not really growing, I don't think I really have a good answer, so I don't want to speculate. One of the things that we want to do also is to be able to generate resistant lines to these parasites. So we've started generating some resistant lines to these 5 isolates that way. I think that might give us a way to really study what is causing the resistance in them and why they probably are not growing well. So we generated some resistance line. And I think your second question was?
Yeah, thanks, Charles. It's about, even if those clinical isolates are growing well and they're thriving in culture, somehow the actual mechanism of gene editing, the specific process of gene editing, seems to be difficult to achieve. Yeah. Do you have any ideas why that would be, and any approaches that you might plan or that you might do in collaboration with others to overcome that? Because it would open the door to some more experiments.
Yeah, I think so. I think that's what we're working on in Australia. And one of my collaborators, so Julian Rayner, is an expert in that, and we plan to really be able to do some of those things. So we have plans to do that. The other thing too is that some of these parasites, we're beginning to see that they have some K30 mutations, and we want to be able to phenotype them. And so we've started sort of doing some drug assays to see if, although they don't have the K30 mutation, we want to be able to see whether they do have resistance to artemisinin, because background mutations also contribute to that. So yes, we're doing a lot of work, and those are in our plans to be able to do that.
Thanks very much for such a wonderful presentation, Dr. Na. And in your presentation, you stated a development of a method to actually develop to detect mutations whilst malaria patients who come in there. So with this kind of developments or these kind of methods to be able to detect these kind of mutations, you will need a robust device or something that can be able to do that function effectively. So I wanted to ask, what metrics are you looking at in terms of creating a good device to be able to actually detect mutations? The sensitivity, accuracy, what are you actually looking at?
Yeah, that's a good question. So that's an industry question. And yes, so I think this is an early phase. So I do sort of, my background has also been in industry. So I spent 3 years in industry manufacturing point of care devices. So, and I've worked with colleagues here like Prof. Musi to be able to implement some of the devices that we manufacture. So we do have the platform, we do have the techniques, we've done it before. Most of the products that I develop, one of the products I developed is commercially available and is being used in Australia. So we do have the platform. We just have to integrate into the existing platform. And so, yeah, those are things that we are looking at.
Right.
Thanks, Charles. I just had a question about the lumefantrine. So what threshold or criteria did you use to define lumefantrine resistance?
So that's a good question. I'm not defining lumefantrine resistance. We do see resistance as way higher than 3V7. So these are preliminary data, so I don't want to go into it in terms of...
Could be problematic. And have these isolates been tested to see if they have high RSA values, survival rates against artemisinin? and if they are also responding to, because apparently when you're getting selection on artemisinin, you might be getting selection on some other blood drugs, and that fits very well with your topic. I mean, if there are 3 isolates out of 100 or 6 out of 200 or 7, that's already a lot of isolates considering the rate of amplification of these, and there's more malaria in the middle belt of the Ghana. So it's something to watch out for. So I don't know what you guys are doing in terms of phenotyping and genotyping to actually confirm this prevalence of 3.4%.
Yeah, thank you. I think that's a bit concerning too, because in terms of the, so it's definitely below the WHO threshold 5% to call for resistance. The other thing to these isolates or these samples were collected as dried blood spots, so we don't have the we didn't collect whole blood, so it's difficult we can't go back to culture them. These are just cross-sectional surveys that were done to pick them up. One of the things that I think Nancy and co, and think Lucas and co are also doing is to be able to monitor and be able to collect the blood and be able to look at. So these are things that are embedded in this program over the next 5 years. And I think that's the beauty about this program, is to be able to sit at the hub in Ghana and be able to build a platform for the CRISPR gene editing and also for the phenotyping. So over the next 5 years, we'll be building that platform here to do that. We have that platform in Australia, so it's basically translating that here.
Right, thank you very much. Further questions over coffee break, I believe you can grab Charles. Charles, thank you so much for the talk. Shall we appreciate him? Right. So our next speaker, Idrissa, will talk on waiting in the wings, resurgence of sylvatic dengue virus in Senegal within a hyper-endemic area. Context, sorry. Idrissa, please take over.
Good morning, everybody. It's a pleasure for me to come and share with you some data that we have regarding the surveillance of dengue viruses in Senegal. As a background, dengue is a big public health threat worldwide, and basically it is a virus, arenavirus, with 10 kb of length, and globally dengue is found all around the world except the Arctic, and usually the distribution of the viruses is mapping the distribution of the vector that is transmitting the virus, that is mainly mosquito of genus Aedes, and principally Aedes aegypti and Aedes albopictus in some temperate area. And according to the estimate, 4.5 billion persons are at risk of dengue infection, and among them they have 3,000 deaths in 2025. And basically the clinical picture of dengue infection is difficult to discriminate with a malaria infection. It starts with a fever associated with at least on 2 of the symptoms like arthralgia, myalgia, muscle pain, or retroorbital pain also. And for some cases, the infection can evolve to cause hemorrhagic form, and this can be exacerbated by the circulation of different variants of the viruses that is called serotypes. This is an antigenically different group of the viruses, and for dengue, they have 4 described serotypes of the viruses that are distributed according to a specific geographical pattern. And for the epidemiological level also, the viruses is circulating in 2 different cycles. This is what is called the sylvatic cycle, for which we have the circulation of the viruses between the primates and the mosquito that you can find in the forest. And from occasionally, the human can be infected by it when they move to the forest for some activities. For example, in Senegal, they have a description of the sylvatic cycle in the region of Kédougou that is bordering Guinea-Conakry and Mali. And also we have the urban cycle, and in the urban cycle we have the circulation of the viruses between the human, and this is driven by the mosquito that we found in the pre-domestic area, and principally Aedes aegypti. And at the genetic level, dengue viruses have 4 serotype, and each of the serotype you have different genotype, and recently they have a new classification that have a shape of the diversity to different major lineage and minor lineage, and also among the lineage we can have different clade, and this have an issue to complicate the development of an effective vaccine and the design of reliable diagnostic tools. And in Senegal, we have starting to detect the dengue infection in 1972, and as shown on the animated figure above, the first detection was the salvatic dengue in 1970, and after from 1 year to another, we have the detection of the other serotype, and the first documented urban dengue infection took place in 2009 with the first urban dengue epidemic that was caused by dengue virus 3. And this figure below summarizes the landscape of dengue virus circulation. Before 2009, most of the dengue infection that we have detected in the country was related to the salvatic cycle, and this was associated with few cases. Between 2009 and 2015, they have a low circulation of the viruses, and from 2015 to now, actually we are facing recurrent and yearly dengue outbreak in Senegal. And recently we have implemented the genomic surveillance of the virus in order to be able to discriminate and identify the variant of the viruses that are circulating at the level of the lineage. And to mitigate the burden of dengue infection, we are using a strategy that is called sentinel strategy, like the one that is used by MICETS, for which that they have one animal that is at the elevated level among the population, and then he is surveilling the surround environment, and in case of danger, he is providing the signal for the other people in order to be able to protect themselves. And this strategy started in Senegal in 2012, and it was only the circulation of the respiratory viruses that were surveilling. And from the data that we are generating from 1 year to another, we found that they have few infection that was associated to respiratory infection, and the patient have the presentation of febrile illnesses, and that was not related to malaria, because as I explained yesterday, we are performing the systematic screening of malaria in case of fever. And in 2015, we implemented the surveillance of arboviruses, and actually at the day of today, we are targeting systematically 8 arboviruses, and among them we have dengue, Zika, chikungunya, West Nile, and others. And in 2017, we have implemented the genomic surveillance of the dengue variant, for which we are trying to track in real time what is the pattern of distribution of the variant of viruses circulating in the country. And recently, we are starting to understand and try to see what is the impact of the found genetic diversity on countermeasures and also on diagnostics. And this figure summarizes the structure of our Sentinel surveillance program. Senegal have 14 administrative regions, and for each of the region we have the Sentinel site, and the aim of the Sentinel site is to capture any inhabitual health event. For example, in one region, if we have an increased number of fever, it is a signal to collect the sample and ship to the laboratory where we are performing the molecular and the serological diagnostics. One thing that need to be taken in consideration, it is when we are looking for arboviruses, it is better to combine molecular and serological testing, because sometimes the window of circulation of the viruses in the blood is too short, and then by combining the both assay, we can have an overview of the circulation of the virus. And then recently, we have layering this surveillance by adding molecular testing in order to know what is the serotype circulating. And among the dengue positive sample, we have subsample, few isolate according to the distribution, spatial and temporal distribution, and for which we perform extensive genomic characterization. And then according to the data that we have generated since the implementation of the surveillance program, you found that from 1 year to another, they have an increase of the number of dengue infection that is detected around the country. At the start of our surveillance program, only one region, the region of Chess, was detecting dengue infection, and till now we have the circulation of the viruses in the all 14 regions of the country. And then at the serotype level, this figure summarizes what is the dynamic of the circulating dengue serotype from one year to another. And here we found clearly that they have a shift of the dominant serotype. This can be explained by the fact that if you are infected by one serotype, you have immunity against a specific serotype, and if this serotype come back to the community, they have a difficulty to propagate properly, but the other serotype can move and cause the epidemic recorded at the specific moment. And also at the genotype level, all the 3 serotypes that we have detecting dengue 1, dengue 2, and dengue 3 are belonging to one specific genotype, except for dengue 2, for which we have both the circulation of the urban genotype and also the circulation of the Salvatic genotype. And at the major and the minor lineage level, they are also belonging in the same major and minor lineage, but they are clustering in different clade, meaning that we have a continuous introduction in the movement of the viruses around the country. And then recently, in 2025, we have recorded the highest number of dengue infection in the countries, where the hotspot was at the border between Senegal and Mauritania. And also this outbreak was matching with the epidemic of Rift Valley fevers that occurred in the northern part of the country, because Mauritania is known to be an endemic region for the circulation of Rift Valley fever. And according to our genetic analysis, we found that this particular epidemic associated to a higher number of cases was linked to the introduction of a new dengue lineage that come from Indian Ocean. And then this particular lineage harbored mutation on the NS1 and the envelope gene, and this mutation was described in the past to be associated to human invasion, the higher production of the NS1 protein, and also some fitness parameters for the viruses. And then also we wanted to understand what is the level of match among the circulating dengue viruses and the vaccine that are under development. And what we did, we sequenced the genome of the dengue virus 3 that we use as model, and then we extract the portion of the genome that are associated and integrated on the vaccine and compare them with 3 different vaccines. And we found that they have 22 and 23 amino acid changes that we found on the region that is included on the vaccine. And by running just MDS analysis, we found that the circulating strain in Senegal is quite far from the vaccine that are under development, and this meaning that the use of this vaccine on the countermeasure may not be effective, but this need to be confirmed by experimental assay. And also on the surveillance program, we are looking for some variant of the viruses that are resistant to antiviral. For dengue mitigation, they have few antiviral that are under development, and among them we have the molecule that is called mosnodenvir. This molecule have the capacity to block the interaction between 2 proteins that are key for the replication efficiency of the viruses. And recently, in the Pacific Island, they have few strains of the viruses that are emerged, and they harbor mutation on the NS2B protein, and this mutation reduce the efficiency of the molecule by 100-fold. And in the way of our genomic surveillance, we are performing the routine screening of the circulating strain in order to advocate for the use of this molecule for the dengue viruses mitigation. And then for this purpose, we have developing an in-house tool that allow the people, if they have this genome sequence, they can have the possibility to know in real time if the mutation, the strain that is circulating harboring the mutation associated to the resistance to the molecule. And then the good news, it is among all the screened strains from Senegal, any of them have the mutation associated to this antiviral, and this is the good news for the use of this molecule on the strategy for the mitigation of the virus's burden around the country. And then the next step, it is to understand and see what is the impact of this genetic diversity on the diagnostic. And I will start with this figure that summarizes the history of Achille. His mother wanted to protect him, and he dived on the Styx River. All the part of his body was protected except the heel. And on the field of the diagnostics of emerging disease using molecular tool, the HLL is the genetic diversity. In 2021, we have a clinical infection of dengue patient, and then the sample was collected and shipped to the reference lab for the diagnostic. When you look the difference between the date of the sample, the symptom onset and the date of sampling, the patient was in the acute phase and have all the clinical manifestation that suspect an arboviral infection. And the sample was collected and shipped at the reference lab for testing. By doing the generic dengue detection process, we found that the sample was positive for dengue with a CT value below 26, meaning that on the sample we have the viral RNA. But in our surprise, when we tried to serotype this variant of the virus, all the attempts to serotyping failed. And that we wanted to understand what is the reason of this failure. What we did, we amplified the conserved region of the genome of the viruses that is part of the polymerase, and then we perform genome sequencing using nanopore device, and then by running the blast we found that the strain that we have detected in 2021 is a strain belonging to the salvatic cycle that we did not detect in the country since 20 years. And from the blast result, what we did, we picked the closest hit with the full genome sequence and designed a set of primer in order to cover the full genome sequence of the viruses. And this allows us to characterize the full genome lengths of the viruses. And then the question that we asked us, why this strain was not able to be serotyping properly. And to answer this question, we map back the primers that is used on the diagnostic tool against the strain, and we found that the primers and the probe are exhibiting a higher number of mismatches on the binding site, and principally on the probe where we have 5 mismatches. And this explains why we was unable to assign this particular variant to a serotype. And then also we repeat the same experiment by mapping back the set of primer that people usually use for sequencing dengue viruses. And then we found that they have also a higher number of mismatches on the primers against the salvatic strain. And if you look for the urban strain, they have consistency around the primer regions. And then what we did, we compare what is the level of coverage of the genome that we will get by using the 3 set of primers, the 2 that was previously published for dengue virus 2, and the new one that we developed in our study. And we found that only our new set of primer was able to perform very well and have consistent coverage across the genome of the virus. And then also the question that we wanted to answer, since for detecting the virus genotype, people need to go up to sequencing, and the sequencing capabilities is not present worldwide. What we did, we designed a set of primer that are specific to the specific variant, and then we integrate it on the screening test that we are using in order to detect the viral serotype. And then we compare the performance of this new assays, both in singleplex and in multiplex. By realizing this study, we found that the assay is able to detect the viruses either both in singleplex and in multiplex with the same sensitivity. And then also for dengue, they have different kits that are available in the market. And from one country to another, people use this kit, and the others are using another one. And then we compare with the file that we are found in our work is just limited to one kit, or this is something that is shared by all the kits available in the market. And for this purpose, we tested 3 different kits, and all of them failed to assign this variant of the dengue to a serotype. And then by collaborating with the industry, now we have developing a new version of the kit that allow people to detect both the 4 variants of the urban dengue viruses and the salvatic strain by just running a PCR. This can be a reliable tool for the field of One Health approach, where we want to understand what is the pattern of the emergence of the viruses, both in human, in animal, and in our environment. And retrospectively, in 2021, we found that they have a cluster of dengue infection that took place in the region of Kédougou that was not investigated because this period was matching with the circulation of the COVID-19 pandemic, and all the effort was oriented to the response to COVID pandemic. What we did, we made a retrospective study on this cluster of dengue infection that we found in Kedugu, and we found that this particular epidemics of dengue infection associated to 52 cases was also caused by the variant of the viruses that are circulating in the salvific cycle. This is surprising to us because in the literature, they is shown that salvific dengue viruses don't have the capability or is not yet associated to any sustained epidemic in any part of the world. And then for this purpose, we try to understand what is the pattern of the circulation of this variant of the viruses. For this purpose, we sequenced the 4 full genome sequence of both the cases in Kedugu and the one that we detected previously in Kolda. And then we put them in the context of the available salvific dengue viruses strands. And then by making the time tree, we found that the cluster of the sequences that we found are dated back to 2004. This meaning that they are the cryptic circulation of the variant of the viruses and probably is maintained in the nature by an unknown reservoir that need to be explored. And also we found that this is not forming a monophyletic cluster, but they have 2 trajectory of evolution of the viruses, and maybe the circulation of the viruses in this part of the country is probably underestimated. And then from the genome sequence that we got, we're trying to assess either if they have correlation or link between the higher number of cases that we found related to the salvific viruses that is previously described to be associated to few cases, and any gain of adaptation. But beside instead of using the cell culture experiment, we have just using our genome and use some in silico approach by calculating the codon adaptation uses between the strain of the viruses and also some housekeeping gene that was described both in the urban vector or in the human. And we found that they have a weak level of correlation between the adaptation to human and to the mosquitoes, but they have a higher p-value. This means that beside the genetic factor, there are probably other factors that are associated for the emergence of the viruses. And then we found that also this particular variant of the viruses have a higher level of adaptation in human, but a lower adaptation in the urban vector of dengue that is widely distributed. And maybe this is the factor why we are not seeing a huge outbreak of this particular viruses in other part of the world. And then beside our detection of the emergence of salvific dengue viruses in 2020 and 2021, recently they have a team in Malaysia. I just wanted to let you know that this particular variant of the viruses is what described in 2 places in the world, in West Africa and in Malaysia. In West Africa, they have a report of the detection of the viruses in Senegal, in Cote d'Ivoire, Guinea, in Nigeria, and in Malaysia also they have a description of the re-emergence of the viruses. And beside our study, one study found that this particular variant of the viruses have also emerged in Malaysia in 2024. And then both for our study and the study in Malaysia, they identified this particular variant of the viruses because the generic testing for dengue working very well, but both of the study was unable to describe and assign the viruses to a specific genotype, serotype using molecular tools. And this meaning that the pattern of the emergence of salvific virus that we found in Senegal is not something restricted to Senegal, but it is something that can be global. Yes, this is what I wanted to share with you, and thank you for the assistance.
Right, thank you very much, Idrissa. Quick one. So from your presentation, I'll just take a quick one before I open the floor. Do you think the climate change or any land use changes could have affected the re-emergence of these strains? And then, affecting their adaptation as well as the transmission that you've talked about. Did you look at that?
Yeah, that's a good question. And actually, we are performing the global genomic analysis of the viruses, and now we are associated to climate data in order to see with some parameter like the temperature, the rainfall, also or the change of the landscape are associated to the re-emergence of the viruses. This is something that is actually under investigation.
Right.
Yeah.
Okay. Thank you. Right. The floor is open for questions, please. Right. Can you please pass the mic?
So, Thanks very much for the presentation. And we have seen several of these dengue viruses, how it's spreading in, I think, Senegal. And I wanted to know about, in this kind of mutation of these dengue viruses, maybe it could be that because of the treatments that they are being subjected to right currently. But then, aren't there fears that probably vaccines under development, once they are being rolled out, because of already the treatment that you are being given to these kind of dengue viruses, it can actually cause a vaccination resistance in these kind of situations? And when such things happen, what are you going to suggest that we can actually do to offset these kind of effects that are happening?
Yeah, the mutation is not related to the vaccine, but it is related to the antiviral. And actually, there are not any study of the extensive use of this antiviral in order to mitigate the dengue. But it is just the idea that we have behind it is before using to see what is the level of the sensitivity of local strain. And then if we are planning to integrate this antiviral on our dengue mitigation strategy, to be able to do the follow-up and then maybe change to another one that cannot have the mutation and be resistant to the variant of virus that we have in the country.
Thank you very much for your presentation. I was going to ask a follow-up question to what Gloria asked. You did mention in your presentation that between 2009 and 2015, there were very low circulation. I was just wondering what could have been responsible for that?
Yeah, I think actually in Senegal we have a good surveillance for human. And then for the animals, the surveillance is not something that is performed longitudinally. And then we believe that maybe they have a reservoir that is hosting the viruses that we are not investigating too much. And that's why we plan to move in the One Health approach, for which any place that I shown here that we have human surveillance, we will integrate the One Health approach in order to sample the animals and all the mosquitoes in the same area, and this can help us maybe to understand better what is the factor that are shaping the emergence of the viruses.
Yeah, I think you kind of preempted my question, because my question was about the non-human reservoir, or you kind of indicate hypothetically that there may be human-to-human transmission occurring more often in more recent outbreaks, and I'm wondering how you are inferring that when you are not sure that there is a gap in between. It's quite seasonal. It's also quite seasonal. So off-season, where do the viruses go? So they must be sitting somewhere, except you're saying that there's also a human reservoir of the virus rather than being the reservoir in a non-human host. So obviously, you've already indicated that it's going to be more severe. The second thing is that with Kédougou, the Kédougou area is like a hotspot. Yeah. It's not only a hotspot for dengue. It's a hotspot for TB, it's a hotspot for malaria, it's a hotspot for HIV, it's a hotspot for multiple things. So there's something about the area. I know there's gold mining around there. Yeah. So people coming from various places. So I don't know whether you're looking at the differences between local transmission and probably transmission that may have been coming from probably Guinea or from Mali or other parts of West Africa.
Yeah, I think also this is a limitation for all the people that are running genomic study. For example, we made recently one study where we are just go to the database and pull down all the genomes that are available for African countries, but they have actually under sequencing. This means that when you run some phylogeographic analysis, you see that the cluster of virus that you have, they have a long branch, meaning that you lost most of the recent evolutionary study, but evolutionary part. And then also one thing to consider, it is for dengue, as many of the other arboviruses, most of the cases are asymptomatic. This means the patient don't feel the need to go to hospital because he is asymptomatic. This is just the few part of the infected people that go to the hospital, and our surveillance program is based to these people that went to hospital. Maybe they can have human that are maintaining the viruses as asymptomatic, and also they have one part of the animals. And recently they have one study in Malaysia that found dengue infection on 3 shrews mammals. This means that beside the non-human primate, maybe they have other mammals around the part of of Kédougou or other part of Senegal that are hosting the viruses from one year to another.
So my question is just, I think there are 4 genotypes, right?
4 serotypes.
4 serotypes, yeah. So I'm just wondering, what's the severity of disease associated to this serotypes? And also, when you have an infection, say serotype 1, I think you said genotype, serotype 2 was the predominant one. So how does that affect your risk to a different genotype? Do you build some sort of cross-immunity?
Actually, in Senegal, this year, the dominant one is dengue 2, the new lineage that was introduction. The year before, the dominant is the dengue 3. It means from one year to another, the dominant is shifting. And then usually people say if you have infected by one serotype and subsequently infected by another, you can have a severe presentation. But they have not the clear pattern if it is 2 followed by 3 that cause the severe infection, or 1 followed by 2 that cause. It is different from one place to another, meaning that it is not maybe some different aspect together that are explain the severity. Just the example about this, it is the same genotypes of dengue is circulating between Senegal and Burkina Faso. And Burkina Faso have reported a higher number of deaths associated to dengue. And for Senegal, we don't have any. We have just a simple dengue infection. We have few dengue associated to hemorrhagic sign. And this aspect also is something of interest for us. This is something that we want to understand and explore. And also we wanted to know if one person is infected by one serotype, the antibody for this specific serotype are stable from which time, in order to have a model that can maybe explain what are the patterns that are explaining the explosive outbreak, beside only the viral factor.
Right. So I believe we can break now and go for our coffee break, since our glucose level is becoming lower. So we need to go and get some, energy, and then we come back, after which we'll have the fellows section. So shall we applaud our speakers for such wonderful presentation? And we'll be back in the next 20 minutes for our fellows session. So please enjoy your break.
Oh, okay. Let me see. How does it work? Left. Is it for this? Oh, this okay.
So you were doing like this?
No, you can present from here.
So the people having the slides like this?
Yeah, I think so.
I'll share the question with you. And we will revise so maybe when we're revising, it shall be a responsibility to somebody. Oh, okay, yeah. Yeah, so what should maybe should be some stuff, what's possible. Since we're not supposed to do work, we're supposed to do the master. And I think there will definitely be some parts that I hope to go and just do the parts. Because we build it on the and here's one more. Okay. Yeah, I was trying to get the file. It looks like, oh, I looked around, I don't see anything there. They don't look at Solomon Islands and Papua, and that's their first kind of stuff. So that's one of us. I think it's more of a mechanistic question. Yeah, I got some meditation. Yes, but she don't have this one. Check. Oh, this is one. Yes, but again, we're talking about how we do some sort of and of course it came out, we don't need money out, we only receive samples. We don't do that again. We've got money. They don't give money out. In fact, when you are taking the money, they give you that's stingy. They're very stingy. They will give you the money for but my university has been very far. Okay. All right.
Yo. Let's talk. I can't wait to see more of these.
All the best. I was following it's like 5 years, right? You can leave to other things.
The ancestors are calling. From the golden dust we rise. Simuka Africa. I remember the smell of rain on the dry savannah, where the baobab stands like a king over the land. Ethiopia's ancient coffee at this morning heat, the sound of children laughing barefoot on the dusty street. Nigeria's heartbeat pulsing through the Lagos night. South Africa's gold shining in the morning light. hyena's wild spirit, where the lions run free. The rhythm of the djembe calling back home to me. Zimbabwe, Zambia, Malawi, Mozambique, South Africa, Botswana, Namibia, Kenya, Tanzania, Uganda, Ethiopia, Rwanda, Ghana, Nigeria, Ghana, Senegal, Ivory Coast, DR Congo, Cameroon, Gabon, Egypt, Morocco, Sudan. Hey. One heart, one blood, one land. Nigeria, Ghana, Senegal, Ivory Coast, DR Congo, Cameroon, Gabon, Egypt, Morocco, Sudan. One heart, one blood, one land. Mama's prayer at dawn.
My name is Gordon Awandare. I am an associate professor of biochemistry at the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. I'm also the director of the West African Center for Cell Biology of Infectious Pathogens, which is a center of excellence for higher education. And we are a center that was founded through funding from the World Bank African Centers of Excellence program. And then we have been supported by the Deltas Africa program, which was funded by the Wellcome Trust and coordinated by ISA and the African Academy. So I trained in the US. I did my PhD at the University of Pittsburgh. Then after that, I did a postdoc at the Walter Reed Army Institute of Research in Silver Spring. And in 2010, I packed my bags and I came back home to Ghana to start my independent career here. And basically, the motivation for coming home was that I thought that the skills that I had were much more needed here than where I was. In the US, I was just one of millions of postdocs doing daily routine research. But I felt that if I came home here with the skills that I had, with the ideas that I had, we could set up a system where other students could have the opportunity of the training that I got in the US, but without having to go to the US. So that was really the driving force for me to come home. And when I came, I was fortunate enough to get the opportunity to do what I always wanted to do. The whole vision about the center was really derived out of the dreams of a few young and energetic scientists in the Department of Biochemistry and our collaborators from the American Society for Cell Biology. We used to run some workshops for African scientists on cell biology and molecular biology. And these workshops became so popular that every year we had a lot of students coming from across Africa to participate in these workshops. And the satisfaction that the students drew from these workshops gave us the impetus to try to provide this type of training on a more permanent basis. So that's where this whole idea of having a center came about, and we started talking about it. The problem was the funding. How do you make this type of thing happen? And then the World Bank came along with the African Center of Excellence, and we thought that it was a perfect vehicle for the type of work that we're trying to do. So I organized my colleagues here, and we put together a proposal to establish WACBIP, really. And the idea was to provide world-class training in cell and molecular biology to young scientists. But using this as a tool to do research into infectious diseases, diseases that are common in Africa. What distinguishes us from other research institutes is that we are in the business of using research as a vehicle to train, and not just doing research alone, but we're using it as a vehicle to train. So we have a degree program we have a graduate degree program, which awards master's and PhD degrees. And we also have a postdoc program. And we drive all this through the research. So we have a research program which supports various faculty to do various research projects. So we have a wide spectrum of research. So we started off doing infectious diseases mainly. So we're doing a lot of pathogen biology, looking at viruses, rotaviruses, HIV virus, looking at malaria parasites, trypanosomes, looking at mycobacteria. These are the bacteria that cause TB and borreliosis and things like that. So that's what we started with doing a lot of. Then with the Deltas project, we got the opportunity to start to introduce human genetics, but all this at the molecular level. Our focus is always doing work at the molecular level. So we started doing human genetics, trying to look at what are some of the genetic markers of the risk of disease, both infectious disease and non-communicable diseases. So we have people even looking at diseases as different from malaria, as autism, or hearing impairment. For me as the leader, it has given me the opportunity to affect both the careers of my colleagues as well as the students. So for the students, they get an education out of it. So far, we've given fellowships to 110 African students for master's, PhD, and postdoc. All these people, there's no guarantee that they would have had a master's or a PhD or a postdoc position without the opportunities that WRC gave. So 110 students, 3 years into our life, have already had fellowships through WRC, and they are working here in the building as well as in our partner institutions. And what that has done is that we're creating a pool of scientists who have high quality training, who have the exposure and the connections that they need to then proceed to have an independent career and then also impact people around their surroundings. On the side of the faculty, there are many colleagues here who have been able to access funding by taking on students and giving them research projects. So they are getting their research done through the students and the fellowships that come with the students. They're able to use those fellowship resources to buy reagents, to run their labs, and get their career also moving forward. In the bigger picture, we are reducing the brain drain, and in fact, we are achieving some brain repatriation because some of our postdocs trained outside, and they came home because they had the opportunity of a postdoc in WRC. They could have gone for a postdoc in the USA or in England or Germany or somewhere else, because these are brilliant young guys. Basically, the environment that we provide is, it sort of provides the assurance that when they come home, they can get the quality of science that they want to do, that they can have a good wage, a good salary that they can live on, and their careers can go on without all the problems that are usually associated with African science. So that the most important thing is that we are providing them with an environment where they have access to good equipment. They have access to world-class scientists from across the world who come and teach here and interact with them and co-supervise their work. They get funds for buying reagents. Each of them has a budget line. Master's students, PhD students, postdocs, each of them, as part of their fellowship, they have a budget line to buy reagents. So they are assured of the ability to conduct their research. And that is the driving force for most of them to come back. Most African scientists who train abroad, if they get that assurance, they usually want to come home. Everybody likes to be home. So if they get the assurance that there's a position for them, there's an opportunity for them to be able to earn a living whilst achieving their dreams of high quality science, most of those that I've talked to, they are willing to come home. So far, we have brought home four Ghanaian students or for African students, let me say, for African scientists who otherwise may have been somewhere else. They finished their PhD, they heard about WACBIP, they applied to WACBIP, and the WACBIP fellowship gave them a landing, sort of a landing spot for them to come and fit back into the system. So, it doesn't get better than that. This is how we stop the brain drain and even start to reverse it.
My name is Dr. Emmanuel Amlabu. I'm a postdoctoral fellow at WACBIP, University of Ghana. I'm from Nigeria.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, is one of the World Bank's African Centers of Excellence and was established in 2014. Led by faculty from the Department of Biochemistry, Cell and Molecular Biology and the Noguchi Memorial Institute for Medical Research at the University of Ghana, the center was chosen by the World Bank as a center of excellence for cell biology with a mandate to provide master's and PhD training.
WACBIP is a place to be as a young, upcoming scientist in Africa because it gives you the enabling environment to undertake collaborative, integrative, and progressive research directed towards advancing health and science education in Ghana or Africa at large.
WACBIP's research mission is to conduct cutting-edge research to guide the development of new approaches to disease diagnosis, prevention, and control. Having established a world-class scientific research environment with wide-ranging technology platforms, the center is now optimized to conduct high-quality research into an expanded range of infectious and non-communicable diseases.
My home country is Kenya, but I studied in Japan and I wanted to come back to Africa to continue the research in infectious diseases. I specialize in malaria research and we are trying to get a vaccine. So I am fortunate to get an opportunity in WACBIP to continue with my research and join fellow Africans.
WACBIP has positioned itself as a major hub for training young African scientists. Since 2014, the center has provided fellowships for master's, PhD, and postdoctoral trainings to more than 200 young scientists from more than 10 countries across the African continent. The center also provides opportunities for career development to industry professionals through short courses.
I'm second year PhD student at WACBIP. To me, I see WACBIP as a springboard. WACBIP provides basic infrastructure in terms of facilities and training for young African scientists who aspire to achieve great heights in science, with access to facilities that enable us to do good science here in Africa, and a rich research collaboration with a lot of faculty members from all around the world.
With a strong commitment to the principles of impactful scientific citizenship, WACBIP places high premium on public and community engagement with numerous initiatives that seek to bring our science closer to the people we do it for. These initiatives give us the opportunity not only to serve, but also to deliver potentially life-saving information on disease prevention and treatment.
But the project team has been able to conduct, watching that very fun where we're able to reach out to children in senior high school and for us to do which makes such an impact. It's something that I would never forget. It's something that I always will WACBIP has presented internationals to never be despite all I love WACBIP.
The center also has a mandate to conduct applied research into biology and pathogenesis of tropical diseases and increase research output and basis.
Shall we
Hello.
humbly be seated for us to take the next session, please?
Private sector leaders in the sub-region.
Do you want me to take this one? So,
right. So welcome back from the break. Okay, can I have your attention, please? Thank you. So we have our fellow session. We continue with Erica Akanko, who is giving a talk on genomic characterization of IZP resistance genes in West Africa, Plasmodium falciparum isolates. Erica. Shall we have Erica, please? Is it online or?
you are Erica. Okay.
Right, so you have 12 minutes, please, and stick to your time. Thank you.
Good morning, everyone. My name is Erica Kanko, and I'm presenting on the genomic and structural characterization of IZP resistant in Plasmodium falciparum African isolates. My presentation will follow the following outline. Okay, now, over the years, we've witnessed the emergence of drug resistance to almost every frontline anti-malaria, and this has created the need to develop new therapeutics with novel targets. One of such novel therapeutics is imidazole-piperazine, IZPs for short. So IZPs are a class of compounds with strong therapeutic potential and have shown to target multiple stages of the life cycle of the parasite. Essentially, its main leading compound, known as ganaplacid, is in phase 3 of clinical trials as of November 2025. And it disrupts protein trafficking via 3 key genes. That is PF-CAL, PF-ACT, and PF-UGT. These are memory transporters, and in their normal function, PF-CAL act to regulate what proteins reach the Golgi apparatus to be distributed to other parts of the parasite cell. The acetyl-CoA transporter, PF-ACT, also acts to regulate the importation of acetyl-CoA, which is needed by the endoplasmic reticulum for protein modification. Then we have the UDP galactose transporter, which is the PF-UGT, which also acts to import galactose that is needed for the overall survival of the parasite. However, there have been the emergence of mutations that confer resistance to these IZPs. When mutations occur in IZP, it reverses the function of the drug, so PF-CAL stabilizes new proteins that are produced and facilitates their delivery to the Golgi apparatus. In PF-ACT, it also regulates the importation of acetyl-CoA, and PF-UGT also regulates the importation of galactose, which overall lead to the survival of the parasite despite the presence of the drug. 2 main studies have established a baseline for my study. In the first study, which was conducted using Senegalese sample and important malaria cases, they identified 3 mutations on the PF-CAL gene and no mutation at all in PF-ACT and PF-UGT. Even these genes harbor these mutations. They were shown not to be associated with decreased susceptibility to any anti-malaria drug. In the other study, no mutation were found in PF-ACT and PF-UGT. This was done in Uganda using Ugandan isolates. However, they identify 1 key mutation, that is the I140V, on PF-CAL, which was associated with moderate decrease in susceptibility to ganaplacid, the drug in phase 3 of clinical trials. Okay, in my study, I aim to characterize IZP resistance in African Plasmodium isolate to establish a genomic surveillance guide even before the drugs are deployed into the African population. Specifically, I will identify single nucleotide polymorphisms, that is SNPs, in this memory transporters and predict their protein stability and provide a structure mechanism of ligand binding via molecular docking. My study followed 3 main methods. In the first part, we will collect data from malaria gene, perform some quality control, and perform bioinformatics analysis. In the second part, for every gene, we will retrieve missense variant and predict their effect on the stability of the structure. And then in the other half, we would predict the structure of the protein, validate the structure, and then mutate the structure for each mutation. And then we'll finally conduct molecular docking to see how they bind onto the drug, which is ganaplacid. This is what I have found from my study. Majority of our samples of 2,802 monoclonal samples came from Ghana, which followed by Mali and Gambia, and countries like Burkina Faso and Cote d'Ivoire representing the least representation. It also spanned 16 years, starting all the way from 1984 to 2018. Yes, PF-CAL had majority of the variant in our samples, with the non-synonymous being greater than the synonymous, showing that PF-CAL variants are widespread in Africa, and then also natural selection may be actively favoring changes in the protein structure, driving their adaptation. Now we want to understand which IZP mutations are circulating in the African population. We found 5 mutations in PF-CAL, of which K903E has the higher prevalence, standing at a percentage of 98.3, showing that it's almost reaching fixation in our population. In PF-ACT, we also identified 3 mutations, of which T459I has the higher prevalence. And then we also found the mutation that was reported by Krenz. Yeah, that was reported to be associated with moderate decrease to ganaplacid susceptibility. Now we want to understand which country is harboring these high-frequency mutations. For T459I, countries including Burkina Faso and Benin and Cote d'Ivoire has the higher prevalence. For I140V, we record Mauritania with the highest prevalence. For PF-CAL, the K903E was almost prevalent in most of the countries. Now I want to understand when these SNPs first emerged. For I140V, it started all the way in 1984 and has been consistent across the years. Between 2001 and 2007, there was no data set. And for the other 2, which were recorded at high frequency, also remained steady with a few dynamics in later years. Now we want to understand how these mutations affect the stability of the proteins in the long run. Using a tool called MU-Pro, we identify that all of the SNPs decreases the structure of the proteins. Now, I want to understand.
She's trying to.
Okay, now I want to understand if these mutations affect the binding of the ligand.
Erica, 2 minutes.
Stable, or it's just the static structure that you used?
Are you talking of the wild type or the mutant?
Both.
Okay.
And the wild type, I know has been experimentally resolved, but then the mutant type, after changing, how did you conclude that it's structurally unstable or it has instability?
Okay. Right. So we use the tool, which is the MU Pro, and it's made of machine learning models. So we have neural networks and support vector machine. When the tool generates the results, it provides you with a value which is known as the change in Gibbs free energy. And this value is between a ratio of -1 and 1. So all values that are less than 0 are unstable, and all that are greater than 1 are stable. Okay, so that was how yeah.
Okay, thank you. I would just recommend that if you can run CapFlex to change the conformational changes of it and see whether this static instability is actually persistent when you run a simulation analysis.
All right, thank you so much.
Our next speaker.
You're not appreciating him.
Oh, sorry. Thank you very much.
Thank you, Erica.
Erica.
Please take your seat.
Our next speaker is Mr. Dovey, who is Ignatius Nunana Dovey, who will speak on the topic Genomic Surveillance in Ghana Reveals Localized Emergence of Artemisinin, Partial Resistance, and Widespread Sulfadoxine Permethrin
resistance. Thank you.
Yeah, good afternoon, everyone. I'm Ignatius Nunana Dovey. I'm a research assistant here at WAGBIP, currently being supervised by Prof. Lucas Amenga Etego, and I'm presenting on genomic surveillance of drug-resistant malaria in Ghana. This study was conducted from 2019 to 2025. Yeah, this is the outline of my presentation. So the use of anti-malarial drugs has proved to be very useful in treating malaria. However, P. falciparum has developed resistance to almost all drugs or the anti-malarials in use. So we've seen multiple emergence of drug resistance, especially in sub-Saharan Africa. It began with chloroquine resistance, where as of chloroquine resistance began spreading to East Africa. It swept from Southeast Asia, and as of 1978, it has spread through the entire continent due to intense drug pressure. This led to the introduction of sulfadoxine pyrimethamine, which soon lost its efficacy. ACTs were introduced in the early 2000s to counter this, but as of 2021, artemisinin partial resistance has emerged independently in East Africa again. So now it is feared that resistance could spread across the entire continent like it did with chloroquine if proper measures are not put in place. Hence, the need to monitor drug-resistant trends in high endemic region like Ghana, it's very critical. And this leads to the aim of my project, to monitor drug resistance spread, to provide timely and actionable information to the elimination program to help support their decisions. So drug resistance is simply our effort to eliminate the parasites. So the more we try to use the anti-malarial, the more we put pressure on the parasite. There is drug pressure, which selects for which kills sensitive parasites while selecting for resistant ones, and this is being amplified by within host selection, and over time there is population sweep. So there are several ways of mitigating this. We have combination therapies, drug rotation, and using multiple first line therapies, which Ghana is doing has implemented some of these measures. Also is genetic surveillance, where we are able to detect early before it is too late. And this is our way, or my way, of contributing to stopping the spread of drug resistance. So we genotyped genetic markers associated with resistance to the anti-malarials here. So we genotyped resistant markers associated with the anti-malarials that were shown, and so we took blood from patients, or dry blood spots from patients who tested positive for P. falciparum malaria. We sampled across 32 sentinel sites across the country. So dry blood spots were taken, they were processed, and then they underwent selective whole genome sequencing for P. falciparum enrichment. We prepared libraries and then sequenced on the Illumina platform. Downstream analysis was performed, where we analyzed drug-resistant mutations, and our findings communicated back to the National Malaria Elimination Program. Yes, so overall we collected over 20,000 samples, as can be seen there. While sequencing is still ongoing, we have successfully genotyped 3,613 samples. As at now, this is a tremendous achievement by the group. So mutations in PF-KELCH13 are associated with resistance to artemisinin, and in our studies, we identified novel KELCH13 mutations like C580S, E605D. While these mutations are currently uncharacterized, when we look at those that have been characterized, we see the P5, those that have been characterized to be associated with artemisinin partial resistance. We see the P574L, which has been associated with artemisinin partial resistance in the north in 2019. However, subsequent monitoring after several years, we did not find this mutation again. But when we look at S522C, which was also identified sporadically in our data, it's been associated with delayed parasite clearance as well. We identified this across multiple time points at different locations in our data. And concurrently, studies from our partner sites in Togo and Nigeria are also identify this mutation, which is suggesting that this is a regional signal. S522C is a Pan-African SNP, and this has to be taken note of, because if this is selected, could be a major threat to effective treatments of malaria in Africa. So to the control programs here. So sulfadoxine pyrimethamine is used in intermittent preventive treatments in pregnant women, as well as a partner drug in combination with amodiaquine, which is being used for SMC for children in the north. And mutations in DHPS and DHFR are associated with resistance to sulfadoxine pyrimethamine. In our study, we observe high prevalence of the sulfadoxine pyrimethamine resistance, which can be seen in the purple, increasing steadily all the way to 90%. And this is due to high prevalence of the triple mutant in DHFR, which can be seen in the blue, increasing steadily all the way to 90%, as well as high prevalence of the DHPS mutation 437G, which can be identified in all the haplotypes that have the G. You see, they have high prevalence. So one thing to note here is that we classify the parasite by the number of mutations it accumulates in both genes, both DHFR and then DHPS. And the more we have high capacity mutations like the K540E, A613S, and A581G, the more resistant the parasite. And this can be summarized in the plot, the spatial plot here, which shows high quadruple mutation. That's the combination of the triple mutant and the 437G, and as well as low prevalence of the quintuple and sextuple mutation. These are due to low prevalence of the high capacity mutation. So in summary, despite high mid-level resistance, SP remain below the WHO 50% threshold for discontinuing targeted interventions like IPTP and SMC. Yeah, so amodiaquine and lumefantrine are key partner drugs in the ACT regimen we use here in Ghana. While there are currently no validated mutation or haplotypes that are associated with lumefantrine resistance, the NFD haplotype, the one in blue, has been associated with reduced lumefantrine susceptibility.
You have 2 more minutes.
Yeah, reduced susceptibility, and we can see it has high prevalence across, and these are poor or early warning signs of reduced lumefantrine susceptibility. Yeah, so we see, yeah, so chloroquine resistance has reduced substantially since it was since policy change in 2005, and however we see a residual of 2%, which is which shows that, and this residual may be due to ACT partner drug amodiaquine, which continues to select for the same K76 allele that the withdrawal was meant to eliminate. And this residual, yeah, we see this, the high frequency of the residual in Greater Accra. This may be due to the fact that the ACT partner drug amodiaquine is being used, is readily available in in Greater Accra because it's an urban center. So in summary, sporadic emergence of WHO validated KEL13 resistant marker P574L highlights the need for continuous monitoring of P. falciparum population for early detection of stable transmission of artemisinin resistance in Ghana. High prevalence of PF-MDR1 NFD haplotype suggests ongoing selection by artemether lumefantrine and may indicate reduced parasite susceptibility to lumefantrine. Also, widespread DHFR, DHPS quadruple mutations warrant urgent SP monitoring in interventions group, particularly for high frequency mutations that we saw earlier. So a lot of work has been put in developing real-time surveillance dashboard, which can be accessed, including chatbot that can be queried to know more about this drug resistant trend. This is very useful, especially to the control program, the elimination program, to provide time to update their treatment guidelines as early as possible as drug resistant mutations emerge. Yeah. Thank you very much. We acknowledge our funders, our collaborators for their, as well as research assistants and everyone who did this work for their tremendous effort they put in stopping the spread of drug resistance. Yeah, thank you.
Thanks
very much.
Thanks, Bobby. We have time for 2 questions. One at the back. And one here.
Yeah, thank you very much for the question. I want to know if there is any possibility of changing the drug prescription patterns in these regions, and also if we can actually connect these resistant patterns to maybe the environmental factors, especially climate, or some activities that are being done in some part of our country. We all know that we've accelerated our land use through Galamsey for like 10 years ahead. So are these resistant patterns connected with these environmental factors, and then the drug distribution and administration and prescription patterns?
Yes, to answer that, the parasites adapt differently according to, the environment they find themselves. So let's say the parasites in East Africa would the resistant patterns it develops would probably be different from what we observe here due to, the different conditions that we find ourselves. And also back to the first question, which is on, please can you remind me?
I wanted to know if the program is integrating these resistant patterns into prescription.
So currently we have the elimination program. So we have three first line treatment ACTs that we use for treating malaria, and other countries rotate them to ensure that one particular drug is not administered for long, so that it reduces the chances of they having resistance.
So you did tremendously well, and what I want from you is to help me understand what you meant by partial resistance.
So artemisinin partial resistance occurs when there is delayed parasite clearance. So for instance, when we take artemisinin or ACTs, let's say artesunate amodiaquine, it's two components. One is the artemisinin component and lumefantrine component. One is supposed to clear, one is fast acting, which clears it quickly, and so that the remaining ones is supposed to be cleared by the other component. But in the case where there is delayed parasite clearance, the sensitive ones will die, but the resistant one will take a longer time before it is being cleared away. In that case, we have partial resistance.
Okay, thank you very much for your wonderful presentation. So I have only two questions for you. So the first question is, please could you provide us some of the limitations of this work when it came to sample collection across the years? And secondly, for how many generations are you supposed to have for a particular parasite, for a particular mutation against a particular drug to be fixed? Those are the two questions. Thank you.
Okay, so with the limitations, if we look at the temporal trends that the map I showed, we saw a few samples collected, especially in 2020 and 2021, which were lower. So the sample collection was heterogeneous due to disruptions in COVID-19, for instance, and also seasonality also affects sample collection. There could be during seasonality. There is spike in malaria cases where and people come to the sentinel sites because they have malaria, and then we could get more samples. But in the dry seasons, in some cases, you would have a lower number of samples. Then your second question. You would have to remind me.
I think, we're running out of time, so.
So the second question is basically on the genes or the SNPs that you're seeing that confer some level of makes them.
Okay, the duration it takes
for a particular parasite to have a fixed mutation, which will help it confer resistance to a particular drug.
For that, I can't specifically, pinpoint, but due to different factors like parasite adaptation, immune pressure, even human, the host immune pressure, it could take like years for a resistant allele to become fixed in the population. But over the years, if you go online, you could tell, the duration in which, let's say, chloroquine became, fixed in the population, sulfadoxine became fixed. Yeah.
Okay, thanks very much.
Yeah.
Looks like we are running out of time. Thanks, Nunana. And our next speaker is Emmanuel Ativi. And Emmanuel will be speaking on genomic and resistant profiles of carbapenem and fluoroquinoline resistant E. coli and Klebsiella pneumonia, causing SSIS. I'm sure he will tell us what that is.
My name is Emmanuel Ativi, and I'll be presenting the findings from my MPhil thesis, genomic and resistant profiles of carbapenem and fluoroquinolone resistant Escherichia coli and Klebsiella pneumoniae from surgical site infections, SSIs for short. Okay. All right. Surgical site infections are among the most common causes of nosocomial infection, and in sub-Saharan Africa and low and resource limited countries, is the most common source of hospital-acquired infections. Now, surgical site infections are infections that occur at the site of incision within 30 days after the surgery, and in situations where implants are used, is up to a year. If infections occur within a year after an implant, it's regarded as surgical site infection. Now, a review of the infection incidence globally reveals a disproportionately high burden of the infection in sub-Saharan Africa of 7.2% against the global estimates of 2.5%. However, pooled estimates of the infection incidence in Ghana reveals a much higher burden of 13.3%. Now, surgical site infections are frequently linked with increased hospital stay, increased healthcare costs, as well as morbidity and mortality. Now, in addition, surgical site infections are linked with risk of antimicrobial resistance due to the frequent use of antimicrobials, mainly for as surgical prophylaxis. Now, during surgery, depending on the type of surgery that is being carried out, you are expected to, there are guidelines that prescribes the antibiotics that you are supposed to give as prophylaxis prior, during, and sometimes after the surgery. Now, this frequently links to antimicrobial resistance due to selective pressures from these prophylaxis. Now, antimicrobial resistance is frequently identified as a silent pandemic, and the World Health Organization report in 2025 on antimicrobial resistance reveals that in every 6 laboratory confirmed infections, one of them is by a resistant pathogen. However, this burden is much higher in Africa. And the same report also reviews an increasing burden of antimicrobial resistance in gram-negative bacteria. Now, a study in Ghana reviews that in surgical site infections, the burden of antimicrobial resistant, especially multi-drug resistant pathogens were increasing. However, fluoroquinolones and carbapenems are major antimicrobials used in treating these infections, where fluoroquinolones, including ciprofloxacin, levofloxacin, or fluoxacin, are used as first-line treatment options, and carbapenems are last resort antibiotics against these infections. Now, the World Health Organization classified these 2 antibiotics under the watch category of the aware classifications of antibiotics due to their risk of resistance, and that requires monitoring. However, studies in Ghana has revealed that fluoroquinolones, especially ciprofloxacin, is frequently being used as prophylaxis, which usually don't go in line with the guidelines for treatment. Now, the two classes of antibiotics are being categorized under the watch category, requiring that they are being monitored for resistance and then the spread in the locality. However, these classes of antibiotic, they could be resistant to them from various mechanisms. And unfortunately, there are few facilities in this country that do culture and sensitivity testing to identify resistant pathogens. Most treatments are done by empiric means, and the few that does antibiotic susceptibility testing on culture are unable to do genotypic tests to identify the various mechanisms that underlie these resistant pathogens. So this study therefore sought to characterize the antimicrobial resistant profiles and the genomic features of these carbapenem and fluoroquinolone resistant pathogens in Klebsiella pneumoniae and E. coli. So this study forms part of an ongoing project which since 2018 has recruited about 750 patients, and out of it 388 has been identified as 388 isolate has been recovered, of which are Klebsiella pneumoniae and Escherichia coli. So out of these 388, the species were confirmed by mass spectrometry, and antimicrobial susceptibility testing was performed on these 388 isolates. Out of it, those that were resistant to carbapenem and fluoroquinolones, specifically meropenem, ciprofloxacin, and ofloxacin, were subjected to Illumina whole genome sequencing. Then bioinformatics and data analysis were then performed from these the sequence with. Below is we identified the various resistant genes, plasmid replicons, sequence types, and mobile genetic elements, as well as virulence from RES finder, plasmid finder, MLST, mobile genetic element finder, and then virulence finder and clevolate. So the study recruited 750 patients, and they were from 2 sentinel sites, Kolebu and 37 military hospital, out of which 129, 259 K pneumonia and E. coli were isolated respectively. From this, 74 Klebsiella pneumonia was sequenced, and 111 E. coli were sequenced. Now, the burden of resistance in these two antibiotic classes reveals that E. coli and Klebsiella pneumoniae, the resistance rate were comparable. About 3/4 of the isolates were resistant to fluoroquinolones. And with the carbapenem, we realized a disproportionate burden between E. coli and Klebsiella, where Klebsiella pneumoniae have about 5 times the rate is seen in E. coli. Okay. Now, a brief look at the antibiotics being used at these facilities for treatment and prophylaxis. We note that ciprofloxacin was among the mostly used antibiotic, both for prophylaxis as well as for treatment. Now, a dive into the resistant profiles of the carbapenem-resistant isolates reveal a very interesting finding where just a few of the antibiotics had some of the isolates were susceptible, some of the isolates, just a few of some of the isolates were susceptible to a few of the antibiotics. So, for example, except for chloramphenicol, where we can see about 50% susceptibility, the rest, most of them were resistant. Similar patterns were seen in Klebsiella pneumoniae. We would understand some of these mechanisms why it's so going forward. No problem. All right. Similar patterns were seen for fluoroquinolone-resistant isolates, where also, except for chloramphenicol and meropenem, most of the antibiotics were resistant, and that tells us that we are really running out of options for treating some of these surgical site infections, as well as prophylactic options for these antibiotics. Now, the resistant patterns reveals the identifications of point mutations, missense point mutations in key components of the organisms, where target mutations in the fluoroquinolone-resistant target, the fluoroquinolone target, which is the gyro-A per se, the gyrase and topoisomerases, where the fluoroquinolones add on to cause the resistance. We could tell there were some resistant mutations, and almost all the topoisomerase resistance, which is the par C and par E, also had gyro-A mutations. That's point mutations in the codon 83 and 87. And that indicates high-level resistance. Similarly, we saw plasmid-mediated quinolone-resistant markers coupling with the chromosomal mutations, also together conferring high-level resistance. With Klebsiella pneumoniae, similar patterns were seen where both chromosomal and plasmid-mediated mechanisms were seen conferring high-level resistance. For carbapenems, interestingly, 6 out of the 16 carbapenems were having carbapenemesis. However, for 6 of them, they lacked carbapenesis. Now, out of these 6 that lacked carbapenemesis, 4 of them we identified had ESBL background and with OMPK36 purine loss. So mutation differentiates, several differentiates in this OMPK gene. And that has been noted to be associated also with carbapenem resistance. Now, about 9 of these 16 isolates had OMPK background as well, which is the purine loss, which frequently also leads to carbapenem resistance. Now, what does this tell us? Most of the diagnostic kit or rapid diagnostic kit for this carbapenem resistance mainly depend on carbapenemesis. And that means if we are going to base on these carbapenemase-based diagnostic kits, we might be losing out on a number of carbapenem-resistant pathogens.
You may just have to summarize.
So, now this slide shows an overview of whatever we did. So we have different sequence types, and there was high diversity. But you realize that the dominant sequence types had a plethora or a repertoire of resistant markers, disinfectant genes, valence markers, as well as plasmid replicons. What is this telling us? This is telling us that the various valence genes, the biocides we are seeing. Biocide that's resistant for chlorhexidine, and a disinfectant that we frequently use to clean surgical wounds, and the presence of biofilms, thermal tolerance, all are indicating adaptation to these hospitals, and we have to include some of these biocides stewardship in our stewardship programs. And we also saw carbapenem and high-level fluoroquinolones. And that means we really need to look out for other alternatives to treating some of these infections. And of note is the carbapenem resistance, that there were non-carbapenemase genes. And this is an opportunity for scientists and biotechs to look into alternatives into treating and identifying some of these pathogens. Thank you.
Thank you, Emmanuel. We have time only for one question, if that's a burning one. Okay.
Yeah, hello. Yeah. Yeah, thank you very much for the presentation. Please, did you compare the two hospitals, their resistant patterns? Maybe it might be a reflective of their practices and then the kind of products that they use in the surgical process.
Okay. Thank you very much for the question, and that's a very good question. Now, we did that comparison, and we noticed some differences. Now, some of the hospitals frequently use some antibiotic, especially ciprofloxacin, though that's not presented here. They frequently use ciprofloxacin, and that correlated with some level of resistance in these ciprofloxacin markers. So yeah, the practices of the hospitals were key, and that's why we are advocating for better stewardship programs. Thank you.
Thank you, Emmanuel. I think Emmanuel will be available for him to be grabbed during lunch. Thank you very much, Emmanuel. Right. So the last speaker for this session is in the person of Felix, who is talking on genomic characterization of Neisseria meningitidis among asymptomatic individuals in the northern region of Ghana. Felix, the floor is yours. Thank you.
IT side. So. Okay. Thank you very much. Good afternoon. I'm in the person of Felix Nenyode, and I'll be presenting on this particular topic. Okay, so Neisseria meningitidis is one of the organisms that normally causes meningitis and septicemia. Just as northern Ghana is also part of Ghana, and Ghana falls within the meningitis belt, which most of these meningitis cases occur. So as a result of that, there are 12 different serogroups of Neisseria meningitidis, but mostly 6 of them are those that cause invasive diseases. And because of these 6, vaccines have been developed against these particular 6, invasive organisms that cause the diseases. And since the introduction of these vaccines, Ghana, we are currently using meningococcal serogroup A vaccine, which mostly is given to children when they are born in the early ages to prevent, these invasive diseases. So, since the 1990s, this vaccine has been in progress or has been in use, and for its introduction, it has helped in so many ways. Since up to now, it has helped to reduce about 56% of the cases of invasive meningococcal diseases. And as you can see, there are some trends as it happens. From the 1990s, when meningococcal MEN-A was the major cause of invasive disease, and the vaccine for MEN-A was introduced, it led to the reduction in this particular pathogen causing in this disease. So MEN-W, which is serogroup W, became the major cause of invasive diseases in Ghana. Somewhere in 2016, an outbreak occurred caused by this meningococcal serogroup W. As a result of that, another mass vaccination was organized, and then it led to the reduction also in this particular, what's the name, serogroup. Somewhere in 2023, another serogroup also appeared and caused invasive disease. As all this is happening, carriage studies has been led to be able to, for us to help to know what exactly is happening in the reservoirs, because we know carriage mostly is the reservoir or the precursor for this invasive disease. So this particular study seeks to characterize carriage studies to be able to identify what is happening in those people who are asymptomatic, for us to be able to derive method for interventions and processes. Just as we were having over here discussion last week or yesterday, in most interventions are against 2030, and meningitis is also part. Invasive meningococcal disease is key, as well is part. So as a result of that, World Health Organization in their 73rd World Health Assembly also proposed the elimination of meningitis, which meningococcal disease is part of. So this leads to this particular project which was carried out. And the main objective of this project is to characterize Neisseria meningitidis among asymptomatic individuals in northern Ghana. The specific objectives are as on, you can see. The major activity, the study was conducted in two different sites in northern Ghana, the Upper West and Upper East. And the Upper West region, which was followed by an outbreak. It was post-outbreak study that was carried out. So after an outbreak had occurred, we followed up to pick pharyngeal samples and to be able to conduct this particular study. In Upper East, that one was part of a major study that is conducted to be able to assess vaccine's impact on this meningitis cases. So that one is a major study which this study falls under. So these two cross-sectional studies are where these samples were gotten from to be able to carry out this particular project. So from oropharyngeal swab to whole genome sequencing, swab samples were collected from individuals who consented for their samples to be picked and to participate in the study. Culture was done on the samples, and Moditov was used to identify the isolates. PCR using targeting the source gene was used on the samples that have been confirmed by Moditov, were submitted also for sequencing, and then bioinformatics analysis was carried out. FastQC was used to assess the quality of the reads. Also, speeds were used to assemble the genome. PAP-MLSD was used for characterization, and Ramsmel was used for phylogenetic tree analysis. So from the analysis, we had a little unfortunate incident, which, if I might call it. Instead of isolate, we were able to identify 13 out of 2,934, which leads or gives us a carriage prevalence of 0.44. For survey 2, which is the Upper West region, we had a carriage prevalence around 1.6, which is 17 out of 146 participants that were recruited. What happened is these samples that have been identified by Moditov and PCR targeting the source gene after whole genome sequencing characterization came out or showed to be a different species, which is Neisseria bergeri. So what, as scientists as we are, what we are supposed to do is to investigate what exactly is happening, whether it's a problem or it's a processing error. So we tried to investigate, and we got to realize that Indeed, this Neisseria is also a Neisseria species. It's carrying the same gene which the Neisseria meningitidis is carrying. In fact, 3 of the Neisseria species are carrying that same gene which the PCR gene is targeting. So it's definitely common. And if you look at the sequence section of where these primers are designed to target, all of them have those exact sequences, no even single nucleotide change, talk less amino acid change. Also, Moditov, which was also used in the process, definitely could misidentify during processes. So from this study, we were able to identify Neisseria bergeri as a new species from this particular study, and this was also the correct prevalence for Neisseria bergeri and Neisseria meningitidis from the 2 different studies. For the characteristics, because we are interested in Neisseria meningitidis, this particular data is about Neisseria meningitidis. These were the characteristics of the participants that were identified to be carriers of Neisseria meningitidis. Majority of them were females, and most of them fall within the ages of 10 to 14 for the survey 1, and for survey 2, those who were above 15 years, mostly because the sampling were not really proportional because of the targeted group that were sampled during the process. We also observed that those with vaccination status really had no, great impact, because the vaccine, remember, is for, currently what is in section is for serogroup A, not targeting the current or most, let's say, those who are currently circulating. So we went further to do serogrouping and PCR genotyping to be able to confirm what these isolates that were identified. So we got to identify that serogroup B and serogroup C were among the isolates that were identified, even though majority of the isolates were non-groupable based on serogroups. However, if we go further, we'll get to see the WGS analysis from the table. So for the isolates that were processed, we did also conduct the antimicrobial susceptibility test to be able to check the resistance level. And this data here is only specifically for the resistance isolates. So we are fortunate that ceftriaxone and chlorphenicol are still susceptible. Apart from that, almost all the other antibiotics that are used in treatment are showing some signs of a level of resistance. Just as you can see, those in dark, very dark one are those of survey 1, and the other one is those of survey 2. The light one is giving us an indication of those that are intermediately resistant to the specific antibiotics. And we can see clearly that penicillin G is having most of its isolates being intermediate resistant, which is really worrying and can cause a lot of issue in the future. We went also on to do sequence typing and to assess the clonal complexes in the analysis. And these were some of the clonal complexes. We looked at the 2 different sites, and the 2 different sites, one of them, which is the upper west region, had one single clonal complex which was in circulation. So all the isolates that were identified from that specific surveillance was one single clonal complex, which is clonal complex or ST175. However, survey 1, which is the upper east region, had at least 4 different clonal complexes, which shows diversity. And don't forget that for the survey 2, the isolates were collected from, or it was a post-meningitis outbreak. So this could suggest a possible that these isolates or were having hand in probably the outbreak that occurred. Also, however, these isolates are non-groupable. Mostly these non-groupables do not cause invasive diseases. So that is one thing also you should take note of. So we did a phylogenetic tree analysis, and this is, I believe maybe you might not be seeing it very well, but it's to just give a brief view of what you see, giving us 2 different or 3 different clusters, telling us that the isolates we have are clustering. This is basically from isolates from Ghana. So we have the Ghana isolates alone from those that we identified. So our isolates we can see are at the bottom side over there, this side, and those that were identified are those at the edges over there. So we looked again at a clear phylogenetic tree using other specimens that are isolates that are available on PUP MLSD to be able to assess, to see where our isolates fall. Even though some are non-groupable, we have 2 that are groupable that may cause invasive disease. So we try to look at some of these isolates. And in the grouping or in the image as you see on the board, it's indicating the 4 different clusters of clonal complexes that were identified, and our isolates or the Ghana isolates that were identified are those that are indicated with the black on top, indicating these specific isolates are. Also, we use I downloaded sequences from PUP MLSD to be able to compare these isolates with, and the countries that the isolates were coming from, their source they were coming from is what is also present over there. So you can see that the 2 serogroupable isolates that we identify fall into those that are really causing invasive diseases elsewhere, which probably suggests that there is a potential chance of these isolates causing invasive disease probably in the future or in time to come. So this is also just to help us to be able to understand the isolates that were
2 minutes more.
The isolates that were identified from the Neisseria beggarii, because these isolates, this is the first time they have been isolated, particularly in Ghana, even though from other sites in the world they have been identified. So the first cluster is an indication of those in orange are an indication of the Neisseria beggarii isolate. The second cluster is Neisseria polysaccharide. The third one is Neisseria lactamica, and the fourth one, those reds, are the Neisseria meningitidis isolate. And those highlighted ones are indicating the Ghana isolates for you to have a clear picture of what they are what's happening over here. So from this particular study, we've got to understand that carriage prevalence of Neisseria meningitidis is low. We've also gone to realize that isolates that have been identified can even confirmed by Moditov and PCR can still be misidentified. So it's very important that if you are doing carriage surveillance of Neisseria meningitidis, or if you are doing carriage of oropharyngeal samples, you probably have to go with whole genome sequencing to be able to get it accurate. And we also realized that resistance patterns were appearing in these isolates that are circulating, even though they are not causing invasive diseases, they are worth noting of, so that in case of invasive or outbreaks, there are ways to treat them becomes very much important. And we also realized that SOVE2 has one clonal complex which dominated throughout, which is somehow a little bit worrying, because even though it's not serogroupable, which cannot, or normally it's not causing diseases, but it has been identified in other places that these non-serogroupable also cause disease. So I think I want to thank these specific individuals for their support and guidance to be able to carry out this work. Thank you very much.
Right, thank you very much, Felix. I'll give a first shot. So, your asymptomatic carriage, did you check whether they were vaccinated or not?
Yes.
Have they received any form of vaccination?
Yes. So, if you can show the data earlier, everybody who was recruited, we assessed their vaccination status before
recruiting them,
yes.
Right? And you made mention of prevalence being low. So the carriage prevalence being low, was it across the whole age group or specific age groups?
So carriage, I think if you can go back to the table, It was basically across all age groups, not specific age group. Yeah.
Oh, okay. And
But mostly, we know that carriage, or mostly these Neisseria meningitidis are carried between the ages of early age and adolescence.
Yeah.
However, that's why I was saying that in survey 2, the participants that were recruited were secondary school children who are a little bit higher, and we also know that carriage is mostly high in those areas, those secondary school levels and dormitories and military camps as well.
Yeah, I mean secondary school level, but maybe you stratify them into age, maybe between early teen or adulthood or 50s or something. That's what I'm asking.
Yes, they were stratified in 1 to 4, 5 to 9,
okay,
11 to 15, but there were no significant difference.
Oh, okay.
Yeah.
And then, sorry, and then you did mention that you saw resistant strains, but specific for a particular drug, or just antimicrobial resistant strains that you found. Was it like carbapenem? Is there a specific one that they used to treat, or just no?
Yes.
I didn't get that, whether you differentiated the specific strains that are specific for a particular drug resistance.
So are you talking about the genome, the genetic?
Yeah, from your genomic studies, you
Yes.
did mention that there were some microbial strains.
Yes, so there were microbial strains. In fact, there were
strains, yes.
beta-lactamase was identified in 2 of the isolates. The phylogenetic tree that I showed is indicated on top over there.
Okay, it was very tiny, so I couldn't see.
Oh,
Thank you.
I apologize for the
Right.
image.
The floor is open for one. Okay, there's a question. There's a hand raised there, please. And then we'll bring the session to a close.
All right. Thank you very much. That was a wonderful presentation. I have 2 very quick questions. One of them is, what's the significance of the non-meningitidis strains you found? What was the clinical significance? And then my second question, did you consider other non-Neisseria causes of meningitis, like maybe strep pneumo and other things, since they also have carriage? Did you consider that? Thank you very much.
Yes, thank you for the question. And let me start from your second question. I think both of these studies are for those organisms. So it's not specifically only for Neisseria meningitidis. I have other colleagues who are working on strep pneumo, others are working on Haemophilus influenza, and even potentially some might also be working on the viruses as well. So it encompasses all sides. Can you repeat your first question?
What's the significance of the non-meningitis strains you found? You kept mentioning that what is the significance? How do they impact, or how will they impact disease progression, for instance?
Yeah, so I think they are normal flora. The Neisseria bergeri is a normal flora in the human human gut. So the significance of it as part of this, I don't really know about it yet, but maybe it's worth looking into. Yeah.
Right, thank you very much, Felix. And shall we applaud all our speakers for this section? Right, thank you. So on our next, we are done, but we're just introducing the next session, which is on vaccine and immune-based biotherapeutics. Is it? No, it's not. Is it lunchtime? No, you're coming, right? Yes. So we're calling Kamara and Pate to take over.
Sharing this session with another person, which apparently is not in the room. But I think I can take all alone. Oh, thank you for the support.
Welcome.
So, from my understanding of the program, I think we have one plenary And then we have poster, right? Tubotox. So I got the students should be aligned over there. So let's start with the plenary. So our plenary speaker is Kwashi Adu Bershi. And Kwashi is going to talk about biomaterials and the efficacy of immunotherapy. So Kwashi, please, floor is yours. And you have 15 minutes,
right.
and we have 5 minutes of questions.
Okay,
thank you.
I wanted to try this before I start. So it really is an honor to be here to share about my work on immunotherapies to enhance efficacy of biomaterial-based technologies to enhance the efficacy of immunotherapies. So I'm going to talk about 2 stories. A bulk of the story will be in oncology, but then if there is time, I'll kind of talk about some of the malaria work that we are doing. I want to motivate this discussion by showing you 2 scans. The first patient had multiple myeloma. The second one had lymphoma. So you can see the patches of dark. These are the cancer cells. Now, these patients underwent a form of therapy called adoptive T-cell therapy. And you can see that everything is cleared. Now, the reason I'm showing these scans is that it shows the power of these new and emerging therapies in terms of treating cancers that are otherwise very difficult. So what is adoptive T-cell therapy? Adoptive T-cell therapy involves taking T-cells from a patient, either from the blood or from the tumors. For those who are not immunologists, war scenario, think of T-cells as special forces, right? They go and they kill. So these T-cells go and then they kill viral infected cells, they also kill cancer cells. For this therapy, you can take them out of the patient, and then after taking them out of the patient, you can activate them to kind of give them some juice, and then you genetically modify them so that they're able to target a specific cancer cell of interest. From there, I knew this clicker would be a problem. Okay, from there you expand the T cells, and then you put them back into the patient. And it's generally done by IV infusion into the blood. Now, adoptive T cell therapy has worked really well for liquid tumors like lymphomas and myelomas, but then when it comes to solid tumors, it's not great. I think only 1, maybe 2 therapies have been approved for solid cancers. So there are limitations that need to be addressed. So some of these limitations to overcome when dealing with solid tumors really are multi-tiered. The first is that you think about a solid tumor being localized, right? Now you inject these cells into the blood. What do they have to do? They have to circulate before they find the tumor. So the fraction of cells finding the tumor at any given time might not be as high as you'd want. Now, when these T cells get into the tumor, there are other issues that they need to face. First is immune suppression. Solid tumors have very elaborate mechanisms for making sure that the T cells are not as active. And then cancers mutate. I think everyone here knows. T cells are antigen specific, meaning that they recognize one type of antigen on the cell. So if these cancer cells end up mutating so that the T cells no longer recognize them, then it doesn't work well. And so all these factors lead to a situation where the T cells have poor functional persistence. So that is where we come in as engineers. The central question the central goal is to enhance the functional persistence of the T cells that we are delivering, because it's an adoptive T cell therapy, whilst also trying to minimize the impact of cancer mutations. So for the T cells we deliver, can we enhance their functional persistence? And also for the cells that are mutating, can we find a way to target them? So we developed a technology called mouthful, Synergistic In Situ Vaccination Enhanced T Cell Depot. I'll just call them CIVETs. And they're trying to do 2 things. The first is that we are trying to enhance the local persistence of T cells we deliver to the tumor. And the second is that we are trying to target the cancer cells that mutate. right? And in doing that, you are trying to generate host T cells that are able to recognize the cancer cells. Because you're assuming that the cells we put in will not be able to recognize all the cancer cells that mutate. So we need a different source of T cells to be able to do that job. And in doing that, we are going to target, generate host T cells to doing that. And if you're able to do that correctly, our hypothesis is that we will result in long-term tumor control. So are we able to actually do this? We envisioned this system. So this is a biomaterial into which you can load your T cells, right? You have a tumor, so you can inject, this is how is an injectable biomaterial system, can inject this biomaterial right next to a tumor. Now this biomaterial has factors that will keep the T cells going, even in vivo. The T cells can leave the biomaterial and then they can attack the tumor and start killing them. Now what happens when cells die? They release antigen, right? And so we are thinking, can we harvest these antigens? So the purple cells are antigen presenting cells. We have material systems. This material system can actively recruit antigen presenting cells to the area of dying tumor cells. Then these antigen presenting cells, their main job is to literally pick up antigen and present them, right? So they present antigen, they traffic to the lymph nodes. I think someone is blocking me. All right. So they traffic to the lymph nodes where they prime host T cells, right? So I'm just describing the process of vaccination for those who don't know. They prime host T cells and then they generate host T cells that are able to target tumor cells. So here we've generated two cell populations, right? The first cell population is what we put in. The second T cell population is a population that we generated in vivo. And so by doing that, we are able to result in long-term tumor control. So how did we exactly do this? So let me introduce the material system to you, if I'm allowed to advance. Okay, so so the material system is what we call a cryogel. So the image to the left is how it looks like. They're kind of tubular length, so you can, depending on how long you'd want it to be, it can be very tunable that way. It's foldable, meaning that you can kind of inject it to a certain spot and then it will fold and conform to the area. These are macroporous. So the pores are many times larger than the size of a cell, meaning that it can serve as a home for the T cells to live and then expand and then traffic out to the tumor. Now I wanted to show you how the T cells generally look like. So if you look at the image to the right, these are like clusters of T cells that are at home in one of the pores of the cryogel. So you can imagine then that you put this right next to a tumor and then they're able to live and attack the tumor. One question we asked is if we can actually enhance the local persistence of delivered T cells using a material like this. So we took T cells that express an enzyme called luciferase. In the presence of a substrate, it produces light, meaning that you can see these T cells under the skin using a technique called IVIS. And so using IVIS, we either put these T cells directly under the skin, so this is what we call direct T cell injection, or we first put them in this civet technology. And what we found was that over time, we are able to get significantly enhanced T cell persistence after we used civet. So the first part was exciting because now, as in the first part of the equation, we're able to do that, able to enhance local persistence. Now, the second question that we wanted to ask is if we can generate host T cells that recognize cancer cells. For the sake of time, I won't go into exactly how we do this, but please trust me when I say that we are able to generate host CD8 T cells, host CD4 T cells that are able to recognize the cancer cells and can work with the T cells we put in to be able to result in long-term tumor control. Does this matter? And so we took mice, gave them tumors, aggressive melanoma tumors, and then we treated them with civet, and they wanted to know if this works. 13 out of 16 mice were able to completely control their tumors long term, suggesting that we are able to use at least this principle. This principle works. Now, since we've published, other groups have also been able to demonstrate that this technique of putting in T cells and generating host T cells actually works. Now, when I started this talk, I talked about how it is very important to look at cancer mutations, right? So how will CIVET actually work within the context of cancer mutations? For this model, we included cells that mutate, that have cancer cells that have mutated, so that the cells, the T cells that we are delivering will not recognize the mutated cancer cells. And the question is that the generated host T cells, can they recognize that mutated cancer cell so that the mice will get protection? And we saw that indeed that is the case. Yep, indeed that is the case. This wasn't like 80-90%, but 60% of the mice were able to control both the normal cells and the mutated cancer cells as well. So that was very impressive and generated a lot of excitement. And so what have we then talked about? We've talked about the development of a technology called CIVET that really enhances adoptive T cell therapy in solid tumors. And it does it in multiple ways. First is that because you put the T cells directly next to a tumor, you're able to get enhanced T cell infiltration. The second is that, I've not shown this data, but then we did quite a lot of work to phenotype the T cells, and we saw that the T cells delivered using this approach had decreased immune suppression. And later we can talk about what exactly we did to be able to figure that one out. And then the third is that we're able to get enhanced protection against cancer mutations. And so then what happens is an overall increased functional persistence. This is the oncology work. I know it was a lot, but with the 4 minutes that I have a timer, so I know how much time I have left. I would like to talk about our malaria work that we are doing using the same principles of biomaterials. So, all right, the malaria is a problem, right? So I don't need to convince anyone. But one thing I wanted to draw your attention to is that 75% of reported deaths occur in children under 5. So this is something that really has to be addressed. Some of you may know about the R21 vaccine. It is a virus-like protein. It's a VLP that displays the circumsporozoid antigen on the surface. So this TEM image really shows the sort of nanoparticulate nature of R21. I would like to show the motifs that are present in R21 and specifically draw attention to the NAMP repeat. So the NAMP repeat is important because NAMP targeting antibodies are highly correlated with protection. So ideally, that is what you want to generate. R21 is approved by WHO, however, it is not durable, right? Usually require 4 shots to get any meaningful protection. I live in Boston, and in the US, you cannot get anyone to come to get 4 shots. Well, maybe a few people. But so in Ghana, where the access to clinical care might not be as readily available, you really want to find a way to cut this down. So we asked ourselves this question, and the question, I need to go back one slide. The question was, how do we develop technologies that can amplify immune responses to malaria vaccines in a way that will make them more feasible in low middle income countries? And the idea was that antigen release kinetics shape responses to malaria vaccines. So think about it this way. If you are infected with a feeble infection, that clears very quickly. Your body does not need to spend the resources to mount a strong immune response because it's gone. But then if the infection is a bit more established, then you actually do spend the resources to mount good immune responses. In the same way, if you just take a bolus vaccine and we inject it and it's cleared very quickly, you don't really get as strong immune responses as you get if we take that same vaccine and then spread it a bit with time. And so we developed the technology to be able to do just that. These are, hopefully the video will play. Yep, it's playing. So these are polymer solutions that you can mix before injection. So you mix, it goes through a small gauge needle. At the site of injection, it stays there, right? And then over time, it will cross-link. The cross-links and then form a depot and a gel. So if your vaccine, your R21 is in there, it kind of releases slowly over time. And we're able to tune the release kinetics of our vaccine that way. So the first question is if we are actually able to do this. So we took the R21 vaccine. So we are working directly with Oxford, the group that developed this R21. So they gave us R21 vaccine. We took it, we modified it with a dye so that you can be able to see it under the skin, and then we injected it. And as bolus, it's gone very quickly. That is a blue dashed line. But with our CH technology, that's the name of the technology that we developed, we're able to tune the release. Right, not only are we able to slow the release, we're able to tune the extent to which we slow them down. And so that was really nice to be able to have that level of control. And then we asked ourselves the question, does this matter? So we looked at total IgG and various IgG subclasses, and saw that for total IgG, we have over 10x improvement when we go with this slow release. The same dose of R21, the same adjuvant, just released differently. And then if you look at the subclasses as well, we see a benefit. So for the sake of time, I'll not tell you all the studies that we did, but we looked at different routes of injection. So we looked at sub-Q versus IM. We looked at different R21 doses, going from 0.05 microgram to 5 microgram R21. And then we also looked at sex. So we looked at biological sex, female and male mice. And for all these observations were consistent, and you do see a benefit for slow release in that regard. So I will end by acknowledging the VS team at Harvard, an ecosystem of scientists, business development, and sponsored research. The faculty head is David Mooney. I also want to acknowledge the scientists and the RAs that I lead to drive this project. So I'll end here, and I'll be happy to take questions.
Yes, thank you very much. That was perfectly in time and very interesting topic. So do you have any question? We have room for a couple of questions.
Thank you. That's a very beautiful presentation. Your system and what you added to slowly release the antigens, can it be called an adjuvant?
So adjuvants do something different. Adjuvants are what enables the immune system to say that your antigen is a danger thing, right? So you actually need to recognize it. So this system itself does not impact the immune system. What it does is that it changes the way that the antigen and the adjuvant interact with the immune system. So we load both the R21 and the adjuvant into the system, and then deliver it. I don't know if I've answered your question.
So is the mechanism depot effect? Because one of the mechanisms of adjuvant action is depot effect. So what is happening, adjuvants. Can it be so how your system is working?
Yeah, that's a really good question.
Can the mechanism be described that way?
Yeah, so it is a depot effect by some adjuvants. So like adjuvants like alum, right, has that depot effect. But the adjuvant used for R21, matrix M, and some kind of other saponin adjuvants actually is not a depot effect. So it is a depot effect, but it is not like alum. So alum is also like Th2 driven. Here we are trying to use the antigen and the adjuvant that is on the market, and then have a biomaterial that itself does not affect the immune system, but then can tune the way that the things we deliver will interact with the immune system.
Thank you.
And we have, yes, at the back.
Okay, thank you so much. My question is about the T-cell immunotherapy you talk about. Yeah. In a case, let's assume that in the future where you are successfully develop this and it goes into the body of the patient and then it successfully clears the tumor. These cells, what happens to them? Do they keep staying in there? Do they have any property that will get them out of the body? Because T cells can sometimes be notorious. After they are done, they are in there and they may cause some other problems.
Actually, in the field, you actually want the T cells to be there because the T cells being there generally help you to fight against the tumors coming back, right? T cells have memory, And so they'll be able to say, this thing is not good, we need to fight it. So one thing I do want to mention is that the material itself will degrade over time. So it will vanish. The T cells, what we have observed is that they will actually, some of them will attack the tumor, some of them will actually go to the lymph nodes. And then they'll stay there as, let's say, a reservoir of cells. And we did a work where we re-challenged the mice and saw that there is a memory response, right? So with adaptive immunity, you actually do want to get a fraction of the cells being there. Otherwise, you might get the tumor coming back.
Yes, so I have a question, but before that, I have a comment. You mentioned that R21 is not long. I mean, the efficacy, it doesn't stay long. I don't think we currently have enough data. I'm standing to be corrected. So we don't have enough data for now to conclude on the long-term efficacy of R21. For RTSS, of course, we know because since from 2011, it has been running, right? But we know R21 is more, the efficacy is about 70%. In the long term, we need more time, maybe 10 years we can.
Yeah, so my point is that we have enough data to know that we need 3 or 4 shots, right? And that alone is a problem. I think if it was just 1 or 2 shots, that would be great.
But there are other vaccines like hepatitis B, you need also several shots.
That is also a problem.
After the last shot, we can have immunity for many, many years, right?
Yeah, so what we are trying to do is to get really, really, really good responses so you don't need 4 shots.
Okay, so my question regarding the T-cell stuff, how did you identify the antigens of cancer cells?
Yeah, that is a big problem, and there are whole teams working on this. For our work, we worked with defined cells that we know the antigen. So like we worked in mice. So for melanoma, there is an antigen called a PMEL antigen that was targeted. So this work was more downstream. This is saying that if you have the antigens, how do you make this work? But I know there are a lot of people, also some in my group, that is trying to look at ways of identifying antigens that are on cancer cells but not on normal cells.
Okay, and also just the last one, you mentioned lymph node, right? So yes. If you have a cancer model or tumor model, you also have normal draining lymph node and you have cancer draining lymph nodes. So which one are you interested in?
Yeah, that's a really good question as well. So there's been a bit of controversy, right? So generally, when someone has cancer, the surgeons will remove all the lymph nodes because they don't want it to serve as a site for metastasis. But then with the emergence of immunotherapies, people are beginning to question that, because at times you shouldn't remove all the lymph nodes because the lymph nodes itself can serve as a site for immune responses. In that case, we're using the cancer draining lymph node, but then we also see cells in circulation, also see cells in the spleen, so they are not kind of localized to the cancer draining lymph nodes. But I think the idea of removing all lymph nodes might be problematic, and surgeons should rethink that strategy.
Okay, thank you very much. That was really very excellent. So,
thank you.
We have to move on.
Now we have table talks, just 2 minutes for each person, and I'm going to put the microphone by the timer. Once you hear it, your time is up. So are you all at least here, gathered here, so the line will move quickly. We're going to start with Abdul Latif Kone, Shadow. Are you here? Okay, Samuel Ntiamwa Osei.
Good afternoon. We've heard so much about AMR from our fellows. AMR.
Is it me? Okay, no. Go ahead.
Okay, AMR is a problem. Millions are dying, and we are looking for different strategies to fight AMR. One of such strategies is employing the use of machine learning. I believe this study is intriguing, and you should visit my poster, number 135, on the 2nd floor, so you can learn more about it. Thank you.
Oh. Thank you very much. Arnold Abaka. I hear. Okay. Tabitha Koni. Tabitha. All right. Joy Kojo Futagbe.
Yeah. Good day, everyone. I have a question to start with. So, have you ever been in severe pains, or do you know anyone with sickle cell disease? Well, what if I tell you that the pain that you've experienced could just be possibly 1/10 of what a person with sickle cell disease goes through with each vaso-occlusive crisis? Imagine feeling pain so severe that it has been described as more than that of childbirth, a major surgery, or even a heart attack. Well, that's the reality of over 8 million people living globally with sickle cell disease. Yet, one question continues to quiz the minds of both researchers and clinicians, that why do two patients with the same sickle cell condition often experience remarkably different levels of pain? Well, the answers to this question may lie just beyond the sickle cell nature of the red blood cells, but rather in the body's own molecular pain sensors. And my name is Joy Nutepe, and I'll be presenting my findings today on the role of transit receptor potentials in sickle cell disease pain genotype, where we used in silico guided wet lab approaches to find out what the roles of these pain receptors are in the pain sickle cell patients feel from day to day. So if you want to hear more about this presentation, visit my poster at poster number 100 to listen to, so that we discuss more into details. Thank you.
Thank you, Joy. We have Adelaide Corios Humani.
Hi, good afternoon. I'm
Hi.
presenting our findings on the distribution of high-risk HPV genotypes among women in different settings within Ghana and Kenya. So as a background, high-risk HPV has been implicated in cervical cancer, which is also one of the commonly diagnosed cancers in women. So the WHO has also established that out of the top 20 countries with highest burdens of cervical cancer, 18 of them happen to be in Africa. So our results also analyze the HPV genotyping tests of 1,025 cervical samples collected from women at screening events and from clinical requests that we received. So we realized that the distribution varied across different settings and with different demographics. Also, with the younger age women that we tested, we realized there was higher risks of positivity as well as multiple and mixed HPV genotypes. So I'm inviting you to pass by our poster. These led us to a number of recommendations, which I would love to share with you. So if you want to know more about this, my poster is at poster number 49. Also, my colleagues will be sharing more on the work we do at Yamachi Biotech, and you can find them at posters number 110 and 154 to discuss more. Thank you.
Thanks, Adelaide. We have Elizabeth Bugase.
Hello.
Hello.
Please, I'm Elizabeth. Please, that's not my slide.
Oh. Can we have Elizabeth's slide?
Anyway, while we wait, I'm here to talk about...
I won't time you, so go ahead.
Okay. So I'm here to talk about a pathogen that has been hiding in plain sight in almost every hospital environment, right? And I know from the previous talks you've heard about AMR, and when you hear AMR, you hear of the popular pathogens like Klebsiella, E. coli. However, who has heard about Morganella morgani?
Okay.
Okay, that's just one out of the many here, right? Yes, but it's a cool name. You can name your daughter Morganella. Yes, so Morganella morgani is an emerging opportunistic pathogen, and like I said, it's being found in almost every hospital environment, but nobody is asking the obvious question: what's making it become dangerous? So we did that by asking the obvious question: what's making Morganella become multi-drug resistant? So, okay, we are here now. Yes, so we asked the obvious question, and the answer is very interesting. It lies in the mobulome, right? That's the mobile DNA that constantly moves in and out of the genome. So I entreat you all to visit my poster at poster number 81. I promise you it's going to be exciting. So come, let's unravel the mobulome and let's understand its pathogenicity. Thank you.
Thanks, Elizabeth. Poster 81, right? Next, we have Esther Donko.
Good afternoon, everyone. My name is Esther Donko, and I'm presenting on genetic diversity of virgins and its association with malaria transmission dynamics. Yeah, so as we all know, malaria caused by Plasmodium species remains a public health concern. And then Plasmodium falciparum in the form of merozoites invade the red blood cells to infect it. And then infected or damaged red blood cells are cleared by the spleen. However, these parasites have been able to modify the surface of the infected red blood cells such that they bind to the walls of the blood vessels and prevent clearance by the spleen. And the molecule that is responsible for this process is called the PFEMP1 protein. That is Plasmodium falciparum erythrocyte membrane protein 1, which is expressed on the surface of the infected red blood cells and then binds to the walls of the blood vessels and prevents clearance by the spleen, as I said earlier. Yeah, so these molecules are not produced randomly. They are encoded by a family of genes called the VAR genes. We have about 50 to 60 VAR genes in the Plasmodium genome, but only one can be expressed at a time, which enables the parasite to evade immune response. So the DBL alpha domain of the VAR gene is a domain of interest because it is present in almost all the 50 to 60 VAR genes, and it is relatively conserved, which makes it a good molecular marker for determining VAR gene diversity. And it's also serves as a proxy for each VAR gene. So by understanding VAR gene diversity, it's going to give us insights into malaria transmission and then population structure. So to know more about this work, kindly visit poster number 83. Thank you.
Kofi Junior Anaman.
Good afternoon, everyone.
Good afternoon.
My name is Kofi Anaman Jr., and I'm here to present on my work. I would like to ask some few questions.
Yes.
Please, can you hear me now?
Yes.
Okay. I would like to ask a few questions. Can life ever exist without disease? Can life ever exist without disease? Oh, some will say yes, others will say no, right? Yes, so you all have different opinions, that's fine. But what if I tell you that this disease that we always fight simply stem from this untreated wastewater from our hospital that releases highly adaptable Staph aureus into the environment? Will you believe that? Well, we are still thinking about it. That's fine. But my question is, one, how accurately are we identifying this Staph aureus from the wastewater from our hospital settings? How? Oh, no answer. Okay. So in my study, I realized that the phenotypic testing of this Staph aureus significantly overestimated the true number of Staph aureus, which called for the molecular confirmation using the multiplex PCR targeting the NUC gene, which serves as the molecular marker to identify the Staph aureus, and the MEC-A gene, which is a resistant gene in the Staph aureus. But do you know what surprised me most?
No.
Most of our key antibiotics, like the vancomycin, which serves as the last result of treatment of this Staph aureus, is now being highly resistant. Don't you think it's a problem? You don't think so? We can die, or you don't believe that? Yes. And so epidemic agent was not found in any of my isolates, which is fine because the pathogen is always evolving its resistance mechanism. This is why I will need you on poster number 59 for us to identify or speak or discuss why this phenotypic testing is not identifying the true Staph aureus and what other alternative mechanisms like MEC could mean for surveillance. Thank you.
Thank you. We have Welbeck Odame Jade. Welbeck, okay. Joseph Yaya.
Good afternoon, everyone.
Good afternoon.
We've heard a lot about malaria and a lot of stuff here. I've not heard anybody talk about trypanosomiasis. So we are neglected tropical diseases. Yeah. So my work centers around tissue tropism in naturally infected animals. And I'm also looking at the variable surface glycoprotein expression and then the transmission potential of this parasite in tissues of naturally infected animals. So the parasite is known to be a hemoparasite, but we are beginning to see that this parasite do not only reside in the blood, but has the capacity to also reside in some tissues, at least in the lab models. But you and I will agree that as scientists, anytime you try to experiment in the lab with cells or lab models, and you try to translate it in the real sense, everything becomes messy. In fact, in some cases, it's not translatable at all. But we decided to take the bull by the horn, and we went ahead to sample animals from tissues from cattle, sheep, and goats. And then we tried to see whether these parasites are in there or not. So what we find is that it's interesting, most of the time when we are talking about trypanosomiasis in naturally infected animals, the infection is usually low. In fact, in some cases, you can see it under the microscope. But to our greatest surprise, in A, we saw massive parastemia in some of these animals. And in fact, that animal was having over 45 million parasites per 1 ml of blood. That is quite surprising. So we went ahead to test whether these parasites are in those tissues that we sampled. So we were able to find this parasite when we amplified the alpha-tubulin gene, which we identify the parasite. We find that this parasite was present in liver, skin, kidney, spleen, and lungs. So this further adds to how complex infection in natural hosts can be. So we went ahead to check what is the infection rate when it comes to different tissues and across all the domestic animals that we sampled. So we found out that there was high infection rates in the sheep and goat that compare to cattle. So it tells us that when there is an infection, you may have variability in terms of which tissue is getting infected and also which animals get infected. So if you want to know more about this work, I'll kindly invite you to come to poster 99. Poster 99, please come for further enlightenment. Thank you.
Thanks, Joseph. Poster 99. Next, we have Selassie Fabla. Sabla. Why did I see F? Oh, there's an... Selassie F. Sabla.
Yeah.
Yeah. Please go ahead.
Yeah. My name is Selassie, and I'm talking about a poster that discusses an in silico approach to designing and developing diagnostics for drug-resistant bacterial infections. You've already heard a lot about bacteria and about AMR, but basically we targeted two microorganisms, Morganella morgani and E. coli, which are on the bacterial priority pathogens list as defined by WHO. So, yeah, there's rising resistance. There are millions of deaths associated with AMR every year, and that number is almost doubling over the next 50 years. So by the year 2050, we are projected to see about 8 million deaths. That's very scary. And part of the reason why the number is rising is because of the limitations with our current diagnostic systems. They are slow, they are expensive, and they are inaccessible, particularly in underserved areas in low to middle income countries, such as Ghana. So that leads to delayed treatment, and when people have treatment delayed, they find antibiotics from anywhere, and that also contributes to the spread of AMR. So it's a snake that ends up eating itself. So our solution is simple. We targeted virulence factors using a multi-omics pipeline we developed based on some papers and we added some stuff to it as well. And we developed a multi-epitope protein candidate for the diagnosis of E. coli and M. morgani associated infections. So if you want to find out more about the solution to AMR, please visit poster number 136. It's on the last floor. I'll try to make it hard to miss. If you come, I'll wave at you. Thank you.
Thanks, Selassie. Next, we have Glenn Kabule.
Hey, good afternoon, everyone. My name is Glenn, and I'll be presenting on my work with the genomic annotation of functional probiotic characteristics of lactic acid bacteria from fermented foods. Okay, so we know probiotics to be live microorganisms that when you take in the right amount will confer you with health benefits. You can usually find them in our normal foods like yogurt and sauerkraut. But these probiotics have the potential to benefit the human body by improving our digestion and help to fight also pathogens as well, right? So before they're able to do all these things, they must first be able to persist within our gut and survive the harsh conditions of our stomach and our gut. And so in order to do these things, you must first have the genes necessary to have these qualities. So my work just simply downloaded 5 strains from NCPI where I ran Quast, Proca, and Eggnog to assess the quality of these genome, annotate them, and also group them into functional classes that are pertaining to probiotic features. And from what we found that there were variations between the strains that we found, and overall for all of them, even though they all shared certain probiotic characteristics, some were more prevalent in one than the other, and they were pertaining to different probiotic functions. So if you are interested in my work, you can find me on the first floor, poster number 89. Thank you.
Thanks, Glenn. We have Olaya Mathilde Adioti. You want to do it?
Good afternoon, everyone. I'm Dr. Mathilde Olaya Adioti. Biostatistics, epidemiology is my area of study. Let's say it like that. It is very good to have a good result from lab, but it is more better to know and use convenient statistical analysis to give the maximum from our lab work and to have a sound and rational scientific paper. This work is an example on infectious diseases spread out dynamic modeling that we look for one approach. The approach show clearly more usefulness in modeling different infectious diseases, Ebola, Lassa, Zika, and so on and so forth, in modeling spread out dynamic, modeling viral load or kinetic exploration of drug and vaccine, and so on and so forth. The other side of things is that the data may even show, let's see, a space matter. So the position, the geographical position of some data may matter. And then including spatial effect in the model is important to improve the model efficiency and then to support a strong evidence-based decision making. So maybe poster 50 to be more informed about for particular data, what analysis may go more better for a sound scientific. Thank you.
Thank you. Next we have Francis Yenube Dut. Feel free to mention your name again, because I don't know what I did. Mention your name again.
All right, the name is Francis Yenube Dut.
Oh.
Yes, I'm presenting on the prevalence and determinants of hepatitis B, hepatitis C, and liver injury among HIV people on antiretroviral therapy in the Upper East region. Imagine treating someone perfectly for HIV just to lose them to a silent liver disease you never imagined. HIV and viral hepatitis, as we all know, when they coexist, they exacerbate liver damage and as well complicate heart management. So in our recent study in the Upper East region, we realized that hepatitis B was a dominant threat among the people living with HIV, and this drive active liver injury in nearly 1 in 6 people living with HIV. And the male sex, older age, as well as the hepatitis B co-infection were revealed to be the significant predictors of liver injury. And to fix this, routine hepatitis screening and liver function monitoring must be integrated into the HIV standard care system in Ghana. For more insight on our study and collaboration, visit us on poster number 87. Thank you.
Thanks, Francis. Next, we have Rhoda Dorcas Seni. Oh, Seni. Oh, lunchtime.
Good afternoon, everyone. I believe at this point we are all famished. My name is Rhoda Dorcas Seni from the Department of Biomedical Sciences, University of Cape Coast.
So okay, you go ahead.
This picture shows, or this image shows, one of Ghana's favorite delicacies, fufu with light soup and meat, specifically offal. From this image, we can see 3 different kinds of offal displayed on the food. We have the tripe, the liver, and the intestine. Now, these 3 types of offal, together with all internal edible organs in an animal, are all classified or called offals. Now, offals in Ghana, popularly known as yemuadie, serve as a nutrient-rich, affordable, and an economical alternative to muscle meat as protein source. However, due to the extensive processing and handling conditions in our local and urban abattoirs, risk these offals to contamination by pathogens such as the Staphylococcus aureus, hence the aim for my study. My study aims to determine the prevalence and antimicrobial susceptibility profile of Staphylococcus aureus isolated from 147 offal samples. Now, from my findings, we can see high contamination of Staphylococcus aureus, high MDR status, and high MRSA or methicillin resistance in the Staphylococcus aureus. At this point, I'm sure some of you or all of you would want to ask, based on my findings, if we should continue eating these offals or we should stop. Now, for an answer to this question, kindly visit my poster, 129, on the last floor. Thank you.
Thanks, Rhoda. Please don't let this get between you and your lunch. I'm sure it's well cooked. Next, we have Ruth Banfo Enin. Thanks, Ruth.
Yeah. Malaria, as we all know, remains a major public health challenge, especially in sub-Saharan Africa. To improve malaria case management, WHO adopted a T3 initiative, which highlighted the need for every suspected malaria case to be confirmed by diagnostics before treatment. In resource-limited settings, malaria RDTs are the most preferred diagnostic tool because they are affordable, easy to use, and require little expertise. Among these RDTs, the HRP2-based RDT is the most widely used due to its sensitivity in detecting the HRP2 antigen that is expressed at all blast stages of the parasite. However, the emergence of HRP2 gene deletion is threatening the effectiveness of these tests, as parasites lacking these genes may escape detection, leading to false negative results. While most studies have focused on HRP2 gene deletions in negative samples, little is known about whether these deletions also occur in positive infections. And this knowledge gap formed the basis of my study. To know more, I invite you to visit poster number 131, where I'll be glad to discuss my findings with you. Thank you.
Thanks, Ruth. Next, we have Nana Ikea Safwa.
Thank you.
So, can yesterday's vaccine-induced immunity protect you against today's variant? This is the question we seek to address in 13 Ghanaian vaccinees that we followed for 30 weeks. So, normally, when we think about vaccine-induced immunity, we think of it as if it's a destination. You get vaccinated, you develop antibodies, and what next? But we don't actually know whether these antibodies persist in our systems and for how long did they persist for. And even after they persist well enough, are they still able to recognize the virus when the virus itself changes? So in this work, we measured nucleocapsid antibodies for these participants, and also we measured neutralizing titers against the wild-type SARS-CoV-2 virus and then the immune-evasive Omicron BQ.1 variant. 3 things stood out. One, we realized that in these participants, there is waning immunity, that their antibody titers are usually high around weeks 2 to 4, and getting to week 30, there is antibody decay. And the participants exhibited higher neutralizing titers to the wild-type compared to the Omicron BQ.1 variant. Finally, prior SARS-CoV-2 exposure enhance your immunity. So to know more about my work, kindly find me at poster number 116. Nanekia Safwa, thank you.
Thanks, Nanekia. Next, we have Frank Ofosu. Thanks, Frank.
Hello, good afternoon. My name is Frank Ofosu from School of Engineering Sciences.
Where's his poster? His slide? Okay, thanks.
Okay.
That's yours, right?
Yes, please.
Okay.
And my work is on anti-biofilm potential of giant African signal collagen chitosan biopolymer.
The mic. You can hear
okay.
you. All right.
Hello. So my work is on anti-biofilm potential of giant African signal collagen and chitosan biopolymer. As we all know, biofilm-associated infection continue to be the global health challenges, and as such, the continual resistance to the antibiotics demand strategic innovative. So as engineers, we try to explore the use of biomaterial to combat these pathogens. So we sort of using collagen, which was actually extracted from our departments as collagen type 1 by Pomeranian et al. 2024, combined with other biopolymer, which is chitosan, to ascertain whether we can control the biofilm formation. And then, as it was mentioned earlier in the preliminary session, biomaterial serving as a vehicle to cure cancer. We also sort of using biomaterial as a vehicle in controlling this biofilm. As such, we introduced 2 antibiotics to see. Please kindly recite poster number 88 to find out more about this cocktail. Thank you.
Thank you. Next, we have Darlington Ekia. Hey, Ekia Aqua.
Hey, my name is Darlington Aqua, and I worked on understanding the mechanism of colistin resistance in multi-drug resistant Klebsiella zitoka. Now, Klebsiella zitoka is an emerging pathogen that is posing a lot of threats to the public. However, multi-drug resistance complicates its treatment, but we don't understand much of the mechanisms that influence its resistance, especially to last-line antibiotic colistin, as compared to its popular cousin, Klebsiella pneumoniae, which most of us would have probably heard of. So this is the gap that we wanted to understand. And what we found quite interesting from our study was that we found heteroresistance, which is an interesting phenotype different from the normal resistance we know. Now, if you want to know more about what heteroresistance is, what it means to our public health, and then the next steps to combat this, please come to my poster number 75. Thank you.
Thanks, Darlington. And we have the final speaker in the person of Jennifer Mtisia. Feel free to pronounce your name again.
Yeah, my name is Jennifer Mtisia.
Oh.
My research is on...
Where's her slide, please?
Yeah, my research is on characterizing HIV2 capsid and capsid spacer peptide 1 for fragment screening and structure-guided drug discovery. So HIV2 is a less prevalent strain of HIV pandemic, and there's a gap in the structural characterization of HIV2 proteins, which limits structure-guided drug discovery. So in this work, I aim at solving structures of 2 drug targets to high resolution with the aim of using these structures to screen new molecules that have efficacy against HIV2. In our lab, we use structural biology methods, which are protein expression and X-ray crystallography to achieve our objectives. And what we've been able to get so far is that we've optimized conditions for the expression of these drug targets to considerable purity. And we've screened an array of buffers, and we've seen some initial crystal hits that we hope to follow up. So for more information about our research, we are at poster number 97. Thank you.
Thanks. Poster number. I think this batch, you've been great at keeping to time. So this is the final speaker, and then we have, before lunch, we have the climate group. Let me look it up again. Climate change. They're going to give a short presentation, and then we'll go out for lunch. Thank you. So is the climate change group here?
Hey. Good afternoon.
Good afternoon.
Yes, so I'm Joseph Kulate from the African Climate Collaborative. I'm the project manager, so I want to give a little...
Can you use the mic, please? Yeah.
Yeah. I want to talk about the African Climate Collaborative and the key milestones we've achieved so far. So...
The mic.
No, this.
Okay.
Yes.
Oh.
So the African Climate Collaborative is an initiative in partnership with the MasterCard Foundation to empower young people within the ages of 18 to 35, particularly women, persons with disability, refugees, and persons from displaced community. We aim at doing this through educational opportunities, cutting-edge research, climate resilience development, circular economic innovations, and green economic growth. This project is for a period of 8 years. Our main focus, again, is 18 to 35. The project is being led by Professor Wandari as the program director, Prof. Chris Gordon as the program deputy director for technical, and Dr. Adelina Mensah, deputy director for operations. Yes. So the African Climate Collaborative at the University of Ghana is it's part of a bigger collaborative where we partner with three other universities within Africa: University of Cambridge, University of Cape Town, and University of Makerere. So how are we doing this? This collaborative is hinged on 4 main pillars: training and capacity building, where we are training the next generation of climate researchers and practitioners across Africa; climate research and resilient research lab, where we are going to transdisciplinary research through the climate future labs and climate action research labs. There's also the networks, policy and learning pillar, where we are strengthening policy engagements and regional networks for climate action. And finally, the innovation and entrepreneurship pillar, where we support climate innovations and green entrepreneurship for African youth. Yeah. So what have we done over the last 2 years? We've supported 52 young Africans by giving them fully sponsored scholarships within the University of Ghana to undertake climate resilience and sustainability related programs. In terms of breakdown, we are supporting 2 postdocs, 11 PhDs, 2 MA/MSc fellows, and 18 MPhil students. Currently, we are in the process of recruiting the next cohort of fellows, and we would at least do a minimum of 27 young Africans. Also, part of the training, we are also doing short courses where we are empowering young people. We are about rolling out a new webinar where we will train young upcoming early career researchers in the various rubrics of publication. So very soon a call will come out, and we expect the young researchers in this room to apply to be trained. Also, we are with partnership with 15 TVET institutions. We are training over 1,000 young students in climate innovations and entrepreneurship. Apart from training, in partnership again with WAGWI, so we enjoy doing partnership. The reason why we are part of this research conference. In partnership with WAGWI, we've upgraded the high-performing computer system, and this system is to strengthen advanced climate research, data analysis, and modeling capacities for the University of Ghana. We've also supported six faculty to promote climate resilience and sustainability research. And also, not only research, we've also modernized and retrofitted certain centers within the university. So the Center for Climate Change and Sustainability Studies and the Center for Disability and Advocacy Studies. So these are two main centers we've retrofitted and modernized with classroom equipment, LED smart televisions, CCTV cameras, and also a board which serves for both C3SS and the New N Block students who visit the New N Block. Also, in partnership with the MP for Ekwapim North, we visited the Senior High Technical School of the Deaf in Mampong, and we established or built a greenhouse facility for them, which is going to serve about 492 persons with disability on sustainable agriculture. So the picture there showing a deba we did where we had the Queen Mother from Mampong gracing the occasion. Yeah, so like I mentioned earlier on, we also do we've just finished a short course for 220 young people where we train. They were in partnership with the geography department. They were trained on climate change, gender equity and social inclusion programs, earth observation for environmental monitoring, smart land use and spatial planning, geointelligence and geographic information systems. So these are programs that we, within the period of time we enroll, so we would also entrust you that you would follow us on our social media handles, and whenever these calls come out, you can also apply, especially the young people, not the old people, please.
Hey.
All young. So as part of this year's World International Women's Day, we also supported some 3 young climate innovators in up to 100,000 cedis in various climate-related entrepreneurship things they were doing. So there's a picture of one of the winners we supported with some seed amounts. Also, we've supported 10 innovation hubs all across the country to train over 500 young innovators on climate innovation, entrepreneurship, and business incubation manuals. So just to enhance their capacity in the various things they are doing all over the country. There is climate change, but the various reporters we have are reporting on politics, football, but there's no reporter on climate reporting. So in partnership with an NGO called CDK and Multimedia, we trained over 200 young people to learn about the rubrics of climate change reporting, and we believe that this would enhance or enable them get some good jobs and do some good climate reporting for us. Apart from all the things, we are also doing strategic partnerships. So we've signed several MOUs. Well, key among them is the MOU we've signed with Navrongo Health Research Center, West African Center for Water Irrigation and Sustainable Agriculture, a dry agriculture college and mechanization center, Kensampo Health Research Center. So we're also doing what WAGBEB is doing. We're also doing something on climate health. So we are here to support. Yes. So please, we want all of you to take up your phones now, and especially the young ones. But the old ones can also do.
God.
But the young ones, we need you more.
What's the cut-off age?
18 to 35.
18 to 35.
Yes.
So if you're above 35, don't.
No, but still follow. You can you can talk to somebody about us.
Oh, who's below 35?
Still do.
Okay.
So please scan, follow us, and when the call comes for the webinar for training on publications, you can join, and we would work with you. Thank you.
Thank you. Prof. Kwashi, do you have any announcement? Lunchtime? And then there'll be a poster session afterwards. Let me look up the program. Okay, so from now we have lunch, and then we have the poster sessions from 2:50 to 3:50, and then we return here at 3:55. Is it okay with everyone? Enjoy your lunch, your offer, all of that. Thank you.
In Awandare. I am an associate professor of biochemistry at the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. I'm also the director of the West African Center for Cell Biology of Infectious Pathogens, which is a center of excellence for higher education. And we are a center that was founded through funding from the World Bank African Centers of Excellence program. And then we have been supported by the Deltas Africa program, which was funded by the Wellcome Trust and coordinated by ISA and the African Academy. So I trained in the US. I did my PhD at the University of Pittsburgh. Then after that, I did a postdoc at the Walter Reed Army Institute of Research in Silver Spring. And in 2010, I packed my bags and I came back home to Ghana to start my independent career here. And basically, the motivation for coming home was that I thought that the skills that I had were much more needed here than where I was. In the US, I was just one of millions of postdocs doing daily routine research. But I felt that if I came home here with the skills that I had, with the ideas that I had, we could set up a system where other students could have the opportunity of the training that I got in the US, but without having to go to the US. So that was really the driving force for me to come home. And when I came, I was fortunate enough to get the opportunity to do what I always wanted to do. The whole vision about the center was really derived out of the dreams of a few young and energetic scientists in the Department of Biochemistry and our collaborators from the American Society for Cell Biology. We used to run some workshops for African scientists on cell biology and molecular biology. And these workshops became so popular that every year we had a lot of students coming from across Africa to participate in these workshops. And the satisfaction that the students drew from these workshops gave us the impetus to try to provide this type of training on a more permanent basis. So that's where this whole idea of having a center came about, and we started talking about it. The problem was the funding. How do you make this type of thing happen? And then the World Bank came along with the African Center of Excellence, and we thought that it was a perfect vehicle for the type of work that we're trying to do. So I organized my colleagues here, and we put together a proposal to establish WACBIP, really. And the idea was to provide world-class training in cell and molecular biology to young scientists. But using this as a tool to do research into infectious diseases, diseases that are common in Africa. What distinguishes us from other research institutes is that we are by taking on students and giving them research projects. So they are getting their research done through the students and the fellowships that come with the students. They're able to use those fellowship resources to buy reagents, to run their labs, and get their career also moving forward. In the bigger picture, we are reducing the brain drain, and in fact, we are achieving some brain repatriation because some of our postdocs trained outside, and they came home because they had the opportunity of a postdoc in WACBIP. They could have gone for a postdoc in the USA or in England or Germany or somewhere else, because these are brilliant young guys. Basically, the environment that we provide it sort of provides the assurance that when they come home, they can get the quality of science that they want to do, that they can have a good wage, a good salary that they can live on, and their careers can go on without all the problems that are usually associated with African science. So that the most important thing is that we are providing them with an environment where they have access to good equipment. They have access to world-class scientists from across the world who come and teach here and interact with them and co-supervise their work. They get funds for buying reagents. Each of them has a budget line. Master's students, PhD students, postdocs, each of them, as part of their fellowship, they have a budget line to buy reagents. So they are assured of the ability to conduct their research. And that is the driving force for most of them to come back. Most African scientists who train abroad, if they get that assurance, they usually want to come home. Everybody likes to be home. So if they get the assurance that there's a position for them, there's an opportunity for them to be able to earn a living whilst achieving their dreams of high quality science, most of those that I've talked to, they are willing to come home. So far, we have brought home four Ghanaian students, or 4 African students, let me say, 4 African scientists who otherwise may have been somewhere else. They finished their PhD, they heard about WACBIP, they applied to WACBIP, and the WACBIP fellowship gave them a landing, sort of a landing spot for them to come and fit back into the system. So, it doesn't get better than that. This is how we stop the brain drain and even start to reverse it.
My name is Dr. Emmanuel Amlabu. I'm a postdoctoral fellow at WACBIP, University of Ghana. I'm from Nigeria.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, is one of the World Bank's African Centers of Excellence and was established in 2014. Led by faculty from the Department of Biochemistry, Cell and Molecular Biology and the Noguchi Memorial Institute for Medical Research at the University of Ghana, the center was chosen by the World Bank as a center of excellence for cell biology with a mandate to provide master's and PhD training.
WACBIP is a place to be as a young, upcoming scientist in Africa because it gives you the enabling environment to undertake collaborative integrative and progressive research directed towards advancing health and science education in Ghana or Africa at large.
WACBIP's research mission is to conduct cutting-edge research to guide the development of new approaches to disease diagnosis, prevention, and control. Having established a world-class scientific research environment with wide-ranging technology platforms, the center is now optimized to conduct high-quality research into an expanded range of infectious and non-communicable diseases.
My home country is Kenya, but I studied in Japan and I wanted to come back to Africa to continue the research in infectious diseases. I specialize in malaria research and we are trying to get a vaccine. So I am fortunate to get an opportunity in WACBIP to continue with my research and join fellow Africans.
WACBIP has positioned itself as a major hub for training young African scientists. Since 2014, the center has provided fellowships for master's, PhD, and postdoctoral training to more than 200 young scientists from more than 10 countries across the African continent. The center also provides opportunities for career development to industry professionals through short courses.
I'm a second-year PhD student at WACBIP. To me, I see WACBIP as a springboard. WACBIP provides basic infrastructure in terms of facilities and training for young African scientists who aspire to achieve great heights in science, with access to facilities that enable us to do good science here in Africa, and a rich research collaboration with a lot of faculty members from all around the world.
With a strong commitment to the principles of impactful scientific citizenship, WACBIP places high premium on public and community engagement with numerous initiatives that seek to bring our science closer to the people we do it for. These initiatives give us the opportunity not only to serve, but also to deliver potentially life-saving information on disease prevention and treatment.
When I joined WACBIP as a graduate student, I expected to be in the lab most of the time, working on my research projects. But then WACBIP turned out to be way more than that, turned out to be much more than that. We had a very fun and exciting and rewarding outreach program where we were able to reach out to children in senior high schools and talk to them about science. And for us to be given the opportunity to make such an impact is something that I will never forget. It's something that I will always hold dear to my heart. WACBIP has also taught me to be tenacious and to never give up, no matter what happens. In science, you may not get the results that you want at that moment, but then WACBIP has taught me that you have to keep on pushing, despite all the odds, despite anything. And at the end of the day, your efforts will be rewarded. I love WACBIP. I mean, it's a place where I can explore my scientific curiosity without boundaries, without being disturbed.
The center also has a mandate to conduct applied research into biology and pathogenesis of tropical diseases and increase research output and innovation by enhancing collaboration among biomedical scientists and industry, private sector leaders in the sub-region.
Je suis à WACBIP depuis l'année passée. C'est vraiment été une expérience vraiment mémorable car j'ai pu rencontrer des professeurs, des amis magnifiques. Ça t'aide, c'est vraiment intéressant car ça te permet de rencontrer beaucoup de personnes, de personnalités. Et en tant que jeune dans le domaine de la science, je trouve que c'est vraiment une très grande opportunité qui s'ouvre pour tous les jeunes scientifiques. Car ça te permet aussi de rencontrer des personnalités d'ailleurs grâce aux conférences qu'ils font ici. Et cela peut vraiment t'aider, ça te guide dans ta recherche, dans ce que tu veux faire pour ton avenir.
WACBIP is further supported by a Wellcome Trust developing excellence in leadership, training and science, Delta's Africa Initiative Grant, to strengthen its research, expand its regional network beyond West Africa, train postdoctoral fellows, and provide additional PhD fellowships.
WACBIP is a life changer It has brought out my hidden potential and exposed me to the global world.
WRC came out of the energy of many young people in the department at the time.
WRC's excellence really stems from teamwork.
We are at the cutting edge of technology. We make excellence the bar for training at WRC.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WRC, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WRC provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WRC was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WRC currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WRC really was a call from the World Bank trying to help higher institutions to set up the BIM. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Theresa, Osborne, Abida. There was another lady called Marian Yaku. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
What first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. and I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than $50 million for training of scientists right here in Africa.
WACBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development, and then to chair WACBIP's advisory board over these last 10 years. A key success of that teaching of WACBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACBIP, but for the fact that WACBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So.
The fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork under very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACBIP because the leadership of WACBIP identifies young talent and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, cruise engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WACBIP, I think is a natural evolution of the development of the center. WACBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACBIP.
Happy 10 years, WACBIP.
Happy 10th anniversary, WACBIP.
Happy 10 years to WACBIP.
Happy 10 years, WACBIP.
Happy, glorious, and illustrious 10th anniversary, WACBIP.
Happy 10th anniversary, WACBIP.
Many congratulations to everybody involved in the WACBIP story.
My name is Gordon Awandare. I am an associate professor of biochemistry at the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. I'm also the director of the West African Center for Cell Biology of Infectious Pathogens, which is a center of excellence for higher education. And we are a center that was founded through funding from the World Bank African Centers of Excellence program. And then we have been supported by the Deltas Africa program, which was funded by the Wellcome Trust and coordinated by ISA and the African Academy. So I trained in the US. I did my PhD at the University of Pittsburgh. Then after that, I did a postdoc at the Walter Reed Army Institute of Research in Silver Spring. And in 2010, I packed my bags and I came back home to Ghana to start my independent career here. And basically, the motivation for coming home was that I thought that the skills that I had were much more needed here than where I was. In the US, I was just one of millions of postdocs doing daily routine research. But I felt that if I came home here with the skills that I had, with the ideas that I had, we could set up a system where other students could have the opportunity of the training that I got in the US, but without having to go to the US. So that was really the driving force for me to come home. And when I came, I was fortunate enough to get the opportunity to do what I always wanted to do. The whole vision about the center was really derived out of the dreams of a few young and energetic scientists in the Department of Biochemistry and our collaborators from the American Society for Cell Biology. We used to run some workshops for African scientists on cell biology and molecular biology. And these workshops became so popular that every year we had a lot of students coming from across Africa to participate in these workshops. And the satisfaction that the students drew from these workshops gave us the impetus to try to provide this type of training on a more permanent basis. So that's where this whole idea of having a center came about, and we started talking about it. The problem was the funding. How do you make this type of thing happen? And then, the World Bank came along with the African Center of Excellence, and we thought that it was a perfect vehicle for the type of work that we're trying to do. So I organized my colleagues here, and we put together a proposal to establish WACBIP, really. And the idea was to provide, world-class training in cell and molecular biology to young scientists. But using this as a tool to do research into infectious diseases, diseases that are common in Africa. What distinguishes us from other research institutes is that we are in the business of using research as a vehicle to train, and not just doing research alone, but we're using it as a vehicle to train. So we have a degree program we have a graduate degree program, which awards master's and PhD degrees. And we also have a postdoc program. And we drive all this through the research. So we have a research program which supports various faculty to do various research projects. So we have a wide spectrum of research. So we started off doing infectious diseases mainly. So we're doing a lot of pathogen biology, looking at viruses, rotaviruses, HIV virus, looking at malaria parasites, trypanosomes, looking at mycobacteria. These are the bacteria that cause TB and borreliosis and things like that. So that's what we started with doing a lot of. Then with the Deltas project, we got the opportunity to start to introduce human genetics, but all this at the molecular level. Our focus is always doing work at the molecular level. So we started doing human genetics, trying to look at what are some of the genetic markers of the risk of disease, both infectious disease and non-communicable diseases. So we have people even looking at diseases as different from malaria, as autism, or hearing impairment. For me as the leader, it has given me the opportunity to affect both the careers of my colleagues as well as the students. So for the students, they get an education out of it. So far, we've given fellowships to 110 African students for master's, PhD, and postdoc. All these people, there's no guarantee that they would have had a master's or a PhD or a postdoc position without the opportunities that WorkBib gave. So 110 students, 3 years into our life, have already had fellowships through WorkBib. And they're working here in the building as well as in our partner institutions. And what that has done is that we're creating a pool of scientists who have high quality training, who have the exposure and the connections that they need to then proceed to have an independent career and then also impact people around their surroundings. On the side of the faculty, there are many colleagues here who have been able to access funding by taking on students and giving them research projects. So they are getting their research done through the students and the fellowships that come with the students. They're able to use those fellowship resources to buy reagents, to run their labs, and get their career also moving forward. In the bigger picture, we are reducing the brain drain. And in fact, we are achieving some brain repatriation because some of our postdocs trained outside and they came home because they had the opportunity of a postdoc in WorkBib. They could have gone for a postdoc in the USA or in England or Germany or somewhere else because these are brilliant young guys. Basically, the environment that we provide is, it sort of provides the assurance that when they come home, they can get the quality of science that they want to do, that they can have a good wage, a good salary that they can live on, and their careers can go on without all the problems that are usually associated with African science. So that the most important thing is that we are providing them with an environment where they have access to good equipment. They have access to world-class scientists from across the world who come and teach here and interact with them and co-supervise their work. They get funds for buying reagents. Each of them has a budget line. Master's students, PhD students, postdocs, each of them, as part of their fellowship, they have a budget line to buy reagents. So they are assured of the ability to conduct their research. And that is the driving force for most of them to come back. Most African scientists who train abroad, if they get that assurance, they usually want to come home. Everybody likes to be home. So if they get the assurance that there's a position for them, there's an opportunity for them to be able to earn a living whilst achieving their dreams of high quality science, most of those that I've talked to, they are willing to come home. So far, we have brought home four Ghanaian students or 4 African students, let me say, 4 African scientists who otherwise may have been somewhere else. They finished their PhD, they heard about WACBIP, they applied to WACBIP, and the WACBIP fellowship gave them a landing, sort of a landing spot for them to come and fit back into the system. So, it doesn't get better than that. This is how we stop the brain drain and even start to reverse it.
My name is Dr. Emmanuel Amlabu. I'm a postdoctoral fellow at WACBIP, University of Ghana. I'm from Nigeria.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, is one of the World Bank's African Centers of Excellence and was established in 2014. Led by faculty from the Department of Biochemistry, Cell and Molecular Biology, and the Noguchi Memorial Institute for Medical Research at the University of Ghana, the center was chosen by the World Bank as a center of excellence for cell biology with a mandate to provide master's and PhD training.
WACBIP is a place to be as a young, upcoming scientist because it gives you the enabling environment to undertake collaborative, integrative, and progressive research directed towards advancing health and science education in Ghana or Africa at large.
WACBIP's research mission is to conduct cutting-edge research to guide the development of new approaches to disease diagnosis, prevention, and control. Having established a world-class scientific research environment with wide-ranging technology platforms, the center is now optimized to conduct high-quality research into an expanded range of infectious and non-communicable diseases.
My home country is Kenya, but I studied in Japan and I wanted to come back to Africa to continue the research in infectious diseases. I specialize in malaria research and we are trying to get a vaccine. So I am fortunate to get an opportunity in WACBIP to continue with my research and join fellow Africans.
WACBIP has positioned itself as a major hub for training young African scientists. Since 2014, the center has provided fellowships for master's, PhD, and postdoctoral training to more than 200 young scientists from more than 10 countries across the African continent. The center also provides opportunities for career development to industry professionals through short courses.
I'm a second-year PhD student at WACBIP. To me, I see WACBIP as a springboard. WACBIP provides basic infrastructure in terms of facilities and training for young African scientists who aspire to achieve great heights in science, with access to facilities that enable us to do good science here in Africa, and a rich research collaboration with a lot of faculty members from all around the world.
With a strong commitment to the principles of impactful scientific citizenship, WACBIP places high premium on public and community engagement with numerous initiatives that seek to bring our science closer to the people we do it for. These initiatives give us the opportunity not only to serve, but also to deliver potentially life-saving information on disease prevention and treatment.
When I joined WACBIP as a graduate intern, I expected to be in the lab most of the time, working on my research projects, but then WACBIP turned out to be way more than that, turned out to be much more than that. We had a very fun and exciting and rewarding outreach program where we were able to reach out to children in senior high schools and talk to them about science. And for us to be given the opportunity to make such an impact is something that I'll never forget. It's something that I will always hold dear to my heart. WACBIP has also taught me to be tenacious and to never give up, no matter what happens. In science, you may not get the results that you want at that moment, but then WACBIP has taught me that you have to keep on pushing despite all the odds, despite anything, and at the end of the day, your efforts will be rewarded. I love WACBIP. I mean, it's a place where I can explore my scientific curiosity without boundaries without being disturbed.
The center also has a mandate to conduct applied research into biology and pathogenesis of tropical diseases and increase research output and innovation by enhancing collaboration among biomedical scientists and industry, private sector leaders in the sub-region.
Je suis à WACBIP depuis l'année passée. C'est vraiment été une expérience vraiment mémorable car j'ai pu rencontrer des professeurs, des amis magnifiques. Ça t'aide, c'est vraiment intéressant car ça te permet de rencontrer beaucoup de personnes, de personnalités. Et en tant que jeune dans le domaine de la science, je trouve que c'est vraiment une très grande opportunité qui s'ouvre pour tous les jeunes scientifiques. Car ça te permet aussi de rencontrer des personnalités d'ailleurs grâce aux conférences qu'ils font ici. Et cela peut vraiment t'aider, ça te guide dans ta recherche, dans ce que tu veux faire pour ton avenir.
WACBIP is further supported by a Wellcome Trust developing excellence in leadership, training and science, Delta's Africa Initiative Grant, to strengthen its research, expand its regional network beyond West Africa, train postdoctoral fellows, and provide additional PhD fellowships.
WACBIP is a life changer. It has brought out my hidden potential and exposed me to the global world.
WACBIP came out of the energy of many young people in the department at the time.
WACBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. my application was not successful. But Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACBIB really was a call from the World Bank trying to help higher institutions to set up the thing. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, you know, Patrick, Lydia, Teresa, Osborne, Abida. There was another lady called Marian Yako. The Vice Chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
What first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACBIB to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACBIB because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WACBIB's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honour for me and a privilege to help in some small way with that early development, and then to chair WACBIB's advisory board over these last 10 years. A key success of that teaching of WACBIB in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established. As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
through my collaboration with WorkBib, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WorkBib is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WorkBib has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WorkBib has more than prepared me, and I wouldn't say this because I've just stayed in WorkBib, but for the fact that WorkBib has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WorkBib was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WorkBib had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WorkBib did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WorkBib did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
we have a tradition that we say in the center known as the WorkBib machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork and very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WorkBib because the leadership of WorkBib identifies young talent and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere. We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, schools engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WRC has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WRC has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and, in fact, most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to research that is being built at centers like WRC, I think is a natural evolution of the development of the center. WRC's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WRC.
Happy 10 years, WRC.
Happy 10th anniversary, WRC.
Happy 10 years to WRC.
Happy 10 years, WRC.
Happy, glorious, and illustrious 10th anniversary, WRC.
Happy 10th anniversary, WRC.
Many congratulations to everybody involved in the WRC story.
Our world is home to lots of tiny things that we cannot see. And one of these tiny things is this very naughty virus called SARS-CoV-2. This is what caused the COVID-19 pandemic. When scientists found out about the virus, they started to learn everything they could about it to find solutions and measures against it. The way to detect this virus is by identifying tiny pieces of its genome through a method known as real-time polymerase chain reaction, popularly shortened to PCR during the pandemic. PCR allows scientists to recognize pieces of the virus's genome or alphabet, which will only be present in an infected individual. Our body's immune system defends us from infection, and this was the case in COVID-19. But SARS-CoV-2 used the immune responses to its advantage. People with extremely strong erratic responses became sick, and those with a more moderate immune response had only mild infection. But SARS-CoV-2 was smart. It started to change itself. It created more versions of itself to make it difficult for scientists to fully understand its nature and weaknesses. With each new version, the solutions the immune system always had to adapt, and the tests were not always effective against it. These changes helped the virus spread from person to person more easily and made it harder for our body to fight the infection. That's why some variants of concern made people more sick and were sometimes difficult to detect. Our hard-working scientists are like detectives and use special tools to study the changes in the virus. They use techniques like sequencing, where they look really close, closer than ever, and read the virus's alphabet. They also develop vaccines to fight against the virus and use the sequences to develop new versions of the vaccines. Understanding the virus and its variants makes it possible to develop better vaccines and safety advice, even as the virus tries new tricks. Even as scientists, and health workers continue to do their part, we all must also continue to do our part. Whether it's keeping clean, getting vaccinated, or following the latest health advice, each step you take makes a big difference. By staying informed and working together, we make our world a top place for SARS-CoV-2. We can win the battle against COVID-19.
Hello, people. Do I even need an intro? SARS-CoV-2, my name. I'm world famous. Oh, and I'm here to give you all the tea about my journey so far. From when I arrived in my glory, how I made your lives miserable, and how all of you banded together to take me on. Let's rewind to my beginnings, shall we? When I came to life in 2019, and fast forward, I had the whole world on its knees. My grand entrance was anything but quiet. Imagine bringing the whole world to a standstill. Oh, the power. I started active work in March 2020, with many parts of the world reporting their first cases. It was showtime. As I unleashed my anger, you quickly rolled up your sleeves. I was unstoppable. I invaded homes, workplaces, even into your grand celebrations. Oh, was I ruthless. I filled your hospitals with sick people. I stretched your resources thin and made your hearts heavy with the losses of loved ones. The economic slowdown, I had a hand in that too. I contributed massively to the loss of jobs, shattered businesses, and the fall of the global market. But then you hit me with your big counter, the lockdown. Oh, the resilience. The world didn't just sit tight. You rallied your warriors, all health workers. You also embraced new norms. Virtual classes on Zoom, digital markets, and work from home. You adapted marvelously. And your innovation took me by surprise. From contact tracing apps, drone delivery for test samples, and even local productions of personal protective equipment like nose masks and sanitizers. You turned what I thought were weaknesses into superpowers. But your real game changer? The vaccine. You launched the vaccination campaigns, and many people around the world armed yourselves against me. But it wasn't all smooth sailing, eh? Just when you thought you had me, I started my tricks and started changing my style, turning into new variants of myself, Alpha, Beta, and Delta. My big hits. Together, we packed a punch. Even then, you countered with boosters, fortifying yourselves against me and my tactics. So, hats off to all of you, but don't start celebrating just yet. The war is far from over. I'm here, lurking, waiting, evolving. I just need you to let your guard down. I'm counting on those who've skipped the vaccines or the boosters. I also hope that you forget to wash your hands with soap, and I encourage you to also cough or sneeze in open air without nose masks. If you do all these, then I'll pounce. Together, we've weathered the storm, and together, we will continue to fight. Stay vigilant, get the jobs, and keep on practicing the safety protocols to keep ourselves, family, community, and nation completely COVID-free.
You won't believe what happened yesterday at the market. This man was trying to persuade me to buy this enormously fat chicken, claiming it had been fed nothing but fresh corn. But I know the truth. It's all those injections and concoctions they've been adding to their feed. That's why all their chickens are looking big like that. And speaking of injections, can you believe people are still going on about getting COVID-19 vaccines? And now they're saying we need to take some boosters too.
Really? Wait, but I thought once you get the vaccines, then that's the end of it. But why the need for more?
Exactly my point. They are saying COVID-19 has been changing itself into new versions. So we need boosters to keep up with the changes. Ha! What should they think of next? That our water will turn to wine unless we put some magic tablets inside?
I also heard those vaccines and boosters are making people impotent. My cousin even claims his PP doesn't work like it used to.
Good morning, everyone. Your interesting discussion caught my attention when I was passing. May I provide some clarity on the COVID-19 vaccines and boosters?
Dr. Fatima, always nice to see you. Please do. We are all here wondering why there's talk of more jabs when this COVID-19 business seems over. Enlighten us. We are all ears.
Thank you. If I may, I would like to tell you a story. Let's think of our body as this fortress. When we were first hit by the COVID-19 virus, it was an invader unknown to our fortress defenses. So, we needed reinforcement to strengthen our walls from the destruction of the invader. That is why we administered the initial vaccines to protect ourselves from the virus. These walls kept us safe. Yet, much like any stubborn enemy, the virus changed its tactics, coming in different forms of itself in a bid to breach our walls yet again. So what do we do in response? We again sharpen our spears and call for reinforcement to make the walls stronger. This is where the booster shots come in. They give our bodies more strength and protection to fend off the virus and its variants. It doesn't mean that the vaccines administered first were ineffective.
But what about side effects, Doc? I heard stories.
Another important question. Imagine you're training for a great battle. The training might leave you sore or tired, and usually these are temporary. Similarly, most side effects from vaccines or boosters are mild and temporary. It's our body's way of preparing to fight the virus. As for those stories saying men becoming sterile or impotent, there's no evidence of the vaccines or boosters causing anything of the sort. They are well tested and safe.
So, in essence, getting the vaccines and booster is
like sharpening our spears, reinforcing our shields, and ensuring we are ready for whatever COVID-19 throws at us. It's how we keep ourselves and our community safe and thriving.
Then what am I waiting for? If it means extra protection, off I go to get this booster. My husband needs me in top form.
Let's remember, our strength lies in our unity and our ability to adapt. By embracing vaccines and boosters, we safeguard ourselves, our loved ones, and our community from the COVID-19 virus.
My name is Gordon Awandare. I am an associate professor of biochemistry at the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. I'm also the director of the West African Center for Cell Biology of Infectious Pathogens, which is a center of excellence for higher education. And we are a center that was founded through funding from the World Bank African Centers of Excellence program. And then we have been supported by the Deltas Africa program, which was funded by the Wellcome Trust and coordinated by ISA and the African Academy. So I trained in the US. I did my PhD at the University of Pittsburgh. Then after that, I did a postdoc at the Walter Reed Army Institute of Research in Silver Spring. And in 2010, I packed my bags and I came back home to Ghana to start my independent career here. And basically, the motivation for coming home was that I thought that the skills that I had were much more needed here than where I was. In the US, I was just one of millions of postdocs doing daily routine research. But I felt that if I came home here with the skills that I had, with the ideas that I had, we could set up a system where other students could have the opportunity of the training that I got in the US, but without having to go to the US. So that was really the driving force for me to come home. And when I came, I was fortunate enough to get the opportunity to do what I always wanted to do. The whole vision about the center was really derived out of the dreams of a few young and energetic scientists in the Department of Biochemistry and our collaborators from the American Society for Cell Biology. We used to run some workshops for African scientists on cell biology and molecular biology. And these workshops became so popular that every year we had a lot of students coming from across Africa to participate in these workshops. And the satisfaction that the students drew from these workshops gave us the impetus to try to provide this type of training on a more permanent basis. So that's where this whole idea of having a center came about, and we started talking about it. The problem was the funding. How do you make this type of thing happen? And then the World Bank came along with the African Center of Excellence, and we thought that it was a perfect vehicle for the type of work that we're trying to do. So I organized my colleagues here, and we put together a proposal to establish WRC, really. And the idea was to provide world-class training in cell and molecular biology to young scientists. But using this as a tool to do research into infectious diseases, diseases that are common in Africa. What distinguishes us from other research institutes is that we are in the business of using research as a vehicle to train, and not just doing research alone, but we're using it as a vehicle to train. So we have a degree program, we have a graduate degree program, which awards master's and PhD degrees, and we also have a postdoc program. And we drive all this through the research. So we have a research program which supports various faculty to do various research projects. So we have a wide spectrum of research. So we started off doing infectious diseases mainly. So we're doing a lot of pathogen biology, looking at viruses, rotaviruses, HIV virus, looking at malaria parasites, trypanosomes, looking at mycobacteria. These are the bacteria that cause TB and borreliosis and things like that. So that's what we started with doing a lot of. Then with the Deltas project, we got the opportunity to start to introduce human genetics. But all this at the molecular level. Our focus is always doing work at the molecular level. So started doing human genetics, trying to look at what are some of the genetic markers of the risk of disease, both infectious disease and non-communicable diseases. So we have people even looking at diseases as different from malaria, as autism, or hearing impairment. For me as the leader, it has given me the opportunity to affect both the careers of my colleagues as well as the students. So for the students, they get an education out of it. So far, we've given fellowships to 110 African students for master's, PhD, and postdoc. All these people, there's no guarantee that they would have had a master's or a PhD or a postdoc position without the opportunities that WACBIP gave. So 110 students, 3 years into our life, have already had fellowships through WACBIP. And they are working here in the building as well as in our partner institutions. And what that has done is that we're creating a pool of scientists who have high quality training, who have the exposure and the connections that they need to then proceed to have an independent career and then also impact people around their surroundings. On the side of the faculty, there are many colleagues here who have been able to access funding by taking on students and giving them research projects. So they are getting their research done through the students and the fellowships that come with the students. They're able to use those fellowship resources to buy reagents, to run their labs, and get their career also moving forward. In the bigger picture, we are reducing the brain drain, and in fact, we are achieving some brain repatriation because some of our postdocs trained outside, and they came home because they had the opportunity of a postdoc in WACBIP. They could have gone for a postdoc in the USA or in England or Germany or somewhere else because these are brilliant young guys. Basically, the environment that we provide is, it sort of provides the assurance that when they come home, they can get the quality of science that they want to do, that they can have a good wage, a good salary that they can live on, and their careers can go on without all the problems that are usually associated with African science. So that the most important thing is that we are providing them with an environment where they have access to good equipment. They have access to world-class scientists from across the world who come and teach here and interact with them and co-supervise their work. They get funds for buying reagents. Each of them has a budget line. Master's students, PhD students, postdocs, each of them, as part of their fellowship, they have a budget line to buy reagents. So they are assured of the ability to conduct their research. And that is the driving force for most of them to come back. Most African scientists who train abroad, if they get that assurance, they usually want to come home. Everybody likes to be home. So if they get the assurance that there's a position for them, there's an opportunity for them to be able to earn a living whilst achieving their dreams of high quality science, most of those that I've talked to, they are willing to come home. So far, we have brought home four Ghanaian students, or 4 African students, let me say, 4 African scientists who otherwise may have been somewhere else. They finished their PhD, they heard about WACBIP, they applied to WACBIP, and the WACBIP fellowship gave them a landing, sort of a landing spot for them to come and fit back into the system. So, it doesn't get better than that. This is how we stop the brain drain and even start to reverse it.
My name is Dr. Emmanuel Amlabu. I'm a postdoctoral fellow at WACBIP, University of Ghana. I'm from Nigeria.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, is one of the World Bank's African Centers of Excellence and was established in 2014. Led by faculty from the Department of Biochemistry, Cell and Molecular Biology, and the Noguchi Memorial Institute for Medical Research at the University of Ghana, the center was chosen by the World Bank as a center of excellence for cell biology with a mandate to provide master's and PhD training.
WACBIP is a place to be as a young, upcoming scientist in Africa because it gives you the enabling environment to undertake collaborative, integrative, and progressive research directed towards advancing health and science education in Ghana or Africa at large.
WACBIP's research mission is to conduct cutting-edge research to guide the development of new approaches to disease diagnosis, prevention, and control. Having established a world-class scientific research environment with wide-ranging technology platforms, the center is now optimized to conduct high-quality research into an expanded range of infectious and non-communicable diseases.
My home country is Kenya, but I studied in Japan and I wanted to come back to Africa to continue the research in infectious diseases. I specialize in malaria research, and we are trying to get the vaccine. So I am fortunate to get an opportunity in WRC to continue with my research and join fellow Africans.
WRC has positioned itself as a major hub for training young African scientists. Since 2014, the center has provided fellowships for master's, PhD, and postdoctoral training to more than 200 young scientists from more than 10 countries across the African continent. The center also provides opportunities for career development to industry professionals through short courses.
I'm a second-year PhD student at WRC. To me, I see WRC as a springboard. WRC provides basic infrastructure in terms of facilities and training for young African scientists who aspire to achieve great heights in science, with access to facilities that enable us to do good science here in Africa, and a rich research collaboration with a lot of faculty members from all around the world.
With a strong commitment to the principles of impactful scientific citizenship, WRC places high premium on public and community engagement with numerous initiatives that seek to bring our science closer to the people we do it for. These initiatives give us the opportunity not only to serve, but also to deliver potentially life-saving information on disease prevention and treatment.
When I joined WRC as a graduate intern, I expected to be in the lab most of the time, working on my research projects, but then WRC turned out to be way more than that, turned out to be much more than that. We had a very fun and exciting and rewarding outreach program where we're able to reach out to children in senior high schools and talk to them about science. And for us to be given the opportunity to make such an impact is something that I will never forget. It's something that I will always hold dear to my heart. WRC has also taught me to be tenacious and to never give up, no matter what happens. In science, you may not get the results that you want at that moment, but then WRC has taught me that you have to keep on pushing, despite all the odds, despite anything. And at the end of the day, your efforts will be rewarded. I love WRC. I mean, it's a place where I can explore my scientific curiosity without boundaries, without being disturbed.
The center also has a mandate to conduct applied research into biology and pathogenesis of tropical diseases and increase research output and innovation by enhancing collaboration among biomedical scientists and industry, private sector leaders in the sub-region.
Je suis à WRC depuis l'année passée. C'est vraiment été une expérience vraiment mémorable car j'ai pu rencontrer des professeurs, des amis magnifiques. Ça t'aide, c'est vraiment intéressant car ça te permet de rencontrer beaucoup de personnes, de personnalités. Et en tant que jeune dans le domaine de la science, je trouve que c'est vraiment une très grande opportunité qui s'ouvre pour tous les jeunes scientifiques. Car ça te permet aussi de rencontrer des personnalités d'ailleurs grâce aux conférences qu'ils font ici. Et cela peut vraiment t'aider, ça te guide dans ta recherche, dans ce que tu veux faire pour ton avenir.
WRC is further supported by a Wellcome Trust developing excellence in leadership, training and science, Delta's Africa Initiative Grant, to strengthen its research, expand its regional network beyond West Africa, train postdoctoral fellows, and provide additional PhD fellowships.
WRC is a life changer. It has brought out my hidden potential and exposed me to the global world.
WRC came out of the energy of many young people in the department at the time.
WRC's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make
excellence the bar for training at WACBIP?
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACBIP really was a call from the World Bank trying to help higher institutions to set up the ring. So I called Gordon and said, Gordon, I think you should host this.
So basically I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Teresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WRC's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development, and then to chair WACBIP's advisory board over these last 10 years. A key success of that teaching of WACBIP in the early years was the contribution of early career faculty, both from the North and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the continents.
WACBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACBIP, but for the fact that WACBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WABIC did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WABIC did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WABIC machinery. And that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork under very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WABIC because the leadership of WABIC identifies young talent and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, cruise engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WABIC has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WABIC has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WABIC, I think is a natural evolution of the development of the center. WABIC's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WABIC.
Happy 10 years, WABIC.
Happy 10th anniversary, WABIC.
Happy 10 years to WABIC.
Happy 10 years, WABIC.
Happy glorious and illustrious 10th anniversary, WRC.
Happy 10th anniversary, WRC.
Many congratulations to everybody involved in the WRC story.
Our world is home to lots of tiny things that we cannot see. And one of these tiny things is this very naughty virus called SARS-CoV-2. This is what caused the COVID-19 pandemic. When scientists found out about the virus, they started to learn everything they could about it to find solutions and measures against it. The way to detect this virus is by identifying tiny pieces of its genome through a method known as real-time polymerase chain reaction, popularly shortened to PCR during the pandemic. PCR allows scientists to recognize pieces of the virus's genome or alphabet, which will only be present in an infected individual. Our body's immune system defends us from infection, and this was the case in COVID-19. But SARS-CoV-2 used the immune responses to its advantage. People with extremely strong erratic responses became sick, and those with a more moderate immune response had only mild infection. But SARS-CoV-2 was smart. It started to change itself. It created more versions of itself to make it difficult for scientists to fully understand its nature and weaknesses. With each new version, the solutions the immune system always had to adapt, and the tests were not always effective against it. These changes helped the virus spread from person to person more easily and made it harder for our body to fight the infection. That's why some variants of concern made people more sick and were sometimes difficult to detect. Our hard-working scientists are like detectives and use special tools to study the changes in the virus. They use techniques like sequencing, where they look really closer than ever, and read the virus's alphabet. They also develop vaccines to fight against the virus and use the sequences to develop new versions of the vaccines. Understanding the virus and its variants makes it possible to develop better vaccines and safety advice, even as the virus tries new tricks. Even as scientists and health workers continue to do their part, we all must also continue to do our part. Whether it's keeping clean, getting vaccinated, or following the latest health advice, each step you take makes a big difference. By staying informed and working together, we make our world a tough place for SARS-CoV-2. We can win the battle against COVID-19.
Hello, people. Do I even need an intro? SARS-CoV-2, my name. I'm world famous. Oh, and I'm here to give you all the tea about my journey so far. From when I arrived in my glory, how I made your lives miserable, and how all of you banded together to take me on. Let's rewind to my beginnings, shall we? When I came to life in 2019, and fast forward, I had the whole world on its knees. My grand entrance was anything but quiet. Imagine bringing the whole world to a standstill. Oh, the power. I started active work in March 2020, with many parts of the world reporting their first cases. It was showtime. As I unleashed my anger, you quickly rolled up your sleeves. I was unstoppable. I invaded homes, workplaces, even into your grand celebrations. Oh, was I ruthless. I filled your hospitals with sick people. I stretched your resources thin. and made your hearts heavy with the losses of loved ones. The economic slowdown, I had a hand in that too. I contributed massively to the loss of jobs, shattered businesses, and the fall of the global market. But then you hit me with your big counter, the lockdown. Oh, the resilience. The world didn't just sit tight. You rallied your warriors, all health workers. You also embraced new norms. Virtual classes on Zoom, digital markets, and work from home. You adapted marvelously. And your innovation took me by surprise. From contact tracing apps, drone delivery for test samples, and even local productions of personal protective equipment like nose masks and sanitizers. You turned what I thought were weaknesses into superpowers. But your real game changer? The vaccine. You launched the vaccination campaigns, and many people around the world armed yourselves against me. But it wasn't all smooth sailing, eh? Just when you thought you had me, I started my tricks and started changing my style, turning into new variants of myself, Alpha, Beta, and Delta. My big hits. Together, we packed a punch. Even then, you countered with boosters, fortifying yourselves against me and my tactics. So, hats off to all of you, but don't start celebrating just yet. The war is far from over. I'm here, lurking, waiting, evolving. I just need you to let your guard down. I'm counting on those who skipped the vaccines or the boosters. I also hope that you forget to wash your hands with soap, and I encourage you to also cough or sneeze in open air without nose masks. If you do all these, then I'll pounce.
Together, we've weathered the storm, and together, we will continue to fight. Stay vigilant, get the jobs, and keep on practicing the safety protocols to keep ourselves, family, community, and nation completely COVID-free.
Believe what happened yesterday at the market. This man was trying to persuade me to buy this enormously fat chicken, claiming it had been fed nothing but fresh corn. But I know the truth. It's all those injections and concoctions they've been adding to their feed. That's why all their chickens are looking big like that. And speaking of injections, can you believe people are still going on about getting COVID-19 vaccines? And now they're saying we need to take some boosters too.
Really? Wait, but I thought once you get the vaccines, then that's the end of it. But why the need for more?
Exactly my point. They are saying COVID-19 has been changing itself into new versions. So we need boosters to keep up with the changes. What should they think of next? That our water will turn to wine unless we put some magic tablet inside?
I also heard those vaccines and boosters are making people impotent. My cousin even claims his PP doesn't work like he used to.
Good morning, everyone. Your interesting discussion caught my attention when I was passing. May I provide some clarity on the COVID-19 vaccines and boosters?
Dr. Fatima, always nice to see you. Please do. We are all here wondering why there's talk of more jabs when this COVID-19 business seems over. Enlighten us. We are all ears.
Thank you. If I may, I would like to tell you a story. Let's think of our body as this fortress. When we were first hit by the COVID-19 virus, it was an invader unknown to our fortress defenses. So, we needed reinforcement to strengthen our walls from the destruction of the invader. That is why we administered the initial vaccines to protect ourselves from the virus. These walls kept us safe. Yet, much like any stubborn enemy, the virus changed its tactics, coming in different forms of itself in a bid to breach our walls yet again. So what do we do in response? We again sharpen our spears and call for reinforcement to make the walls stronger. This is where the booster shots come in. They give our bodies more strength and protection to fend off the virus and its variants. It doesn't mean that the vaccines administered first were ineffective.
But what about side effects, Doc? I heard stories.
Another important question. Imagine you're training for a great battle. The training might leave you sore or tired, and usually these are temporary. Similarly, most side effects from vaccines or boosters are mild and temporary. It's our body's way of preparing to fight the virus. As for those stories saying men becoming sterile or impotent, there's no evidence of the vaccines or boosters causing anything of the sort. They are well tested and safe.
So, in essence, getting the vaccines and booster is
like sharpening our spears, reinforcing our shields, and ensuring we are ready for whatever COVID-19 throws at us. It's how we keep ourselves and our community safe and thriving.
Then what am I waiting for? If it means extra protection, or if I go to get this booster, my husband needs me in top form.
Let's remember, our strength lies in our unity and our ability to adapt. By embracing vaccines and boosters, we safeguard ourselves, our loved ones, and our community from the COVID-19 virus.
My name is Gordon Awandare. I am an associate professor of biochemistry at the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. I'm also the director of the West African Center for Cell Biology of Infectious Pathogens, which is a center of excellence for higher education. And we are a center that was founded through funding from the World Bank African Centers of Excellence program. And then we have been supported by the Deltas Africa program, which was funded by the Wellcome Trust and coordinated by ISA and the African Academy. So I trained in the US. I did my PhD at the University of Pittsburgh. Then after that, I did a postdoc at the Walter Reed Army Institute of Research in Silver Spring. And in 2010, I packed my bags and I came back home to Ghana to start my independent career here. And basically, the motivation for coming home was that I thought that the skills that I had were much more needed here than where I was. In the US, I was just one of millions of postdocs doing daily routine research. But I felt that if I came home here with the skills that I had, with the ideas that I had, we could set up a system where other students could have the opportunity of the training that I got in the US, but without having to go to the US. So that was really the driving force for me to come home. And when I came, I was fortunate enough to get the opportunity to do what I always wanted to do. The whole vision about the center was really derived out of the dreams of a few young and energetic scientists in the Department of Biochemistry and our collaborators from the American Society for Cell Biology. We used to run some workshops for African scientists on cell biology and molecular biology. And these workshops became so popular that every year we had a lot of students coming from across Africa to participate in these workshops. And the satisfaction that the students drew from these workshops gave us the impetus to try to provide this type of training on a more permanent basis. So that's where this whole idea of having a center came about. and we started talking about it. The problem was the funding. How do you make this type of thing happen? And then, the World Bank came along with the African Center of Excellence, and we thought that it was a perfect vehicle for the type of work that we're trying to do. So I organized my colleagues here, and we put together a proposal to establish WACBIP, really. And the idea was to provide, world-class training in cell and molecular biology to young scientists. But using this as a tool to do research into infectious diseases, diseases that are common in Africa. What distinguishes us from other research institutes is that we are in the business of using research as a vehicle to train, and not just doing research alone, but we're using it as a vehicle to train. So we have a degree program, we have a graduate degree program, which awards master's and PhD degrees, and we also have a postdoc program. And we drive all this through the research. So we have a research program which supports various faculty to do various research projects. So we have a wide spectrum of research. So we started off doing infectious diseases mainly. So we're doing a lot of pathogen biology, looking at viruses, rotaviruses, HIV virus, looking at malaria parasites, trypanosomes, looking at mycobacteria. These are the bacteria that cause TB and borreliosis and things like that. So that's what we started with doing a lot of. Then with the Deltas project, we got the opportunity to start to introduce human genetics. But all this at the molecular level. Our focus is always doing work at the molecular level. So we started doing human genetics, trying to look at what are some of the genetic markers of the risk of disease, both infectious disease and non-communicable diseases. So we have people even looking at diseases as different from malaria, as autism, or hearing impairment. For me as the leader, it has given me the opportunity to affect both the careers of my colleagues as well as the students. So for the students, they get an education out of it. So far, we've given fellowships to 110 African students for master's, PhD, and postdoc. All these people, there's no guarantee that they would have had a master's or a PhD or a postdoc position without the opportunities that WACBIP gave. So 110 students, 3 years into our life, have already had fellowships through WACBIP. And they are working here in the building as well as in our partner institutions. And what that has done is that we're creating a pool of scientists who have high quality training, who have the exposure and the connections that they need to then proceed to have an independent career and then also impact people around their surroundings. On the side of the faculty, there are many colleagues here who have been able to access funding by taking on students and giving them research projects. So they are getting their research done through the students and the fellowships that come with the students. They're able to use those fellowship resources to buy reagents, to run their labs, and get their career also moving forward. In the bigger picture, we are reducing the brain drain, and in fact, we are achieving some brain repatriation because some of our postdocs trained outside, and they came home because they had the opportunity of a postdoc in WACBIP. They could have gone for a postdoc in the USA or in England or Germany or somewhere else because these are brilliant young guys. Basically, the environment that we provide is, it sort of provides the assurance that when they come home, they can get the quality of science that they want to do, that they can have a good wage, a good salary that they can live on, and their careers can go on without all the problems that are usually associated with African science. So that the most important thing is that we are providing them with an environment where they have access to good equipment. They have access to world-class scientists from across the world who come and teach here and interact with them and co-supervise their work. They get funds for buying reagents. Each of them has a budget line. Master's students, PhD students, postdocs, each of them, as part of their fellowship, they have a budget line to buy reagents. So they are assured of the ability to conduct their research. And that is the driving force for most of them to come back. Most African scientists who train abroad, if they get that assurance, they usually want to come home. Everybody likes to be home. So if they get the assurance that there's a position for them, there's an opportunity for them to be able to earn a living whilst achieving their dreams of high-quality science, most of those that I've talked to, they are willing to come home. So far, we have brought home four Ghanaian students, or 4 African students, let me say, 4 African scientists who otherwise may have been somewhere else. They finished their PhD, they heard about WACBIP, they applied to WACBIP, and the WACBIP fellowship gave them a landing, sort of a landing spot for them to come and fit back into the system. So, it doesn't get better than that. This is how we stop the brain drain and even start to reverse it.
My name is Dr. Emmanuel Amlavu. I'm a postdoctoral fellow at WACBIP, University of Ghana. I'm from Nigeria.
The West African Center for Cell Biology of Infectious Pathogens, WACBIP, is one of the World Bank's African Centers of Excellence and was established in 2014. Led by faculty from the Department of Biochemistry, Cell and Molecular Biology and the Noguchi Memorial Institute for Medical Research at the University of Ghana, the center was chosen by the World Bank as a center of excellence for cell biology with a mandate to provide master's and PhD training.
WACBIP is a place to be as a young, upcoming scientist in Africa because it gives you the enabling environment to undertake collaborative, integrative and progressive research directed towards advancing health and science education in Ghana or Africa at large.
WACBIP's research mission is to conduct cutting-edge research to guide the development of new approaches to disease diagnosis, prevention and control. Having established a world-class scientific research environment with wide-ranging technology platforms, the center is now optimized to conduct high-quality research into an expanded range of infectious and non-communicable diseases.
My home country is Kenya, but I studied in Japan and I wanted to come back to Africa to continue the research in infectious diseases. I specialize in malaria research and we are trying to get a vaccine. So I am fortunate to get an opportunity in WACBIP to continue with my research and join fellow Africans.
WACBIP has positioned itself as a major hub for training young African scientists. Since 2014, the center has provided fellowships for master's, PhD, and postdoctoral training to more than 200 young scientists from more than 10 countries across the African continent. The center also provides opportunities for career development to industry professionals through short courses.
I'm a second-year PhD student at WACBIP. To me, I see WACBIP as a springboard. WACBIP provides basic infrastructure in terms of facilities and training for young African scientists who aspire to achieve great heights in science, with access to facilities that enable us to do good science here in Africa, and a rich research collaboration with a lot of faculty members from all around the world.
With a strong commitment to the principles of impactful scientific citizenship, WACBIP places high premium on public and community engagement with numerous initiatives that seek to bring our science closer to the people we do it for. These initiatives give us the opportunity not only to serve, but also to deliver potentially life-saving information on disease prevention and treatment.
Hello, good afternoon. It's finally time for the final session of the WACBIP Research Conference, Day 3, session 3, and this session is on drug discovery, therapeutic targets, and resistance. And for the session chairs, we would have Jesli Chirarua and Hadija Tuembe, if they can kindly take their seats up here to begin the session.
It's something that I'll WRC has also taught me to be tenacious and to never give up, no matter what happens. In science, you may not get the results that you want at that moment, but then WRC has taught me that you have to keep on pushing despite all the odds, despite anything. And at the end of the day, your efforts will be rewarded. I love WRC. I mean,
hello. Hello. Yes. Yeah, so welcome to the last session. I know we are so much late, about more than 15 minutes late. And yeah, I think we'll just get straight ahead. So we'll have about 3 talks. One plenary talk from Dr. Stephen Ahato, and then we'll have 2 fellow sessions. So if you're ready, I don't know if... Okay. Yeah, so it's 15 minutes. I don't know if I have to go over your topic or you here.
Yeah.
Do I have to go over your topic?
I think we're already behind time, so we
okay.
can start. Yeah.
All right. Thank you.
Okay. Yeah. Thanks so much for the opportunity to present. My name is Stephen Della Ahato, and today I'll be presenting a platform which I use for studying host microbe interactions. So I'm a researcher at the Arctic University of Norway, where I kind of develop nanoparticles or lipid nanoparticles for delivery of biologics, such as proteins, microRNAs, as well as peptides. And also, I'm involved in other groups where we develop kind of machine learning platforms, as well as also bioinformatics pipelines for understanding kind of the evolution of bacteria. And in WRC, I'm kind of using these machine learning platforms as well as bioinformatics to be able to understand the evolutionary trajectory of most escape pathogens, particularly the gram-negative ones. Things not working so well.
Point it to the back.
Oh, I need to point to the back. Oh, okay. Yeah, sure. Yeah. So my main research focus is combining or integrating multiple kind of omics platforms to be able to understand how pathogens interact with their host. And one method I'm using is the chemoproteomics platform. Aside that too, we are using this platform to be able to identify new targets druggable targets for novel antimicrobial identification. And also using this platform also, we are kind of studying the metabolic pathways of bacteria in a quest for the ability to be able to target them and also metabolically reprogram most of the persistent cells to be able to treat them. And as well as some of my research in AMR evolution and adaptation, where we use these machine learning platforms to be able to kind of understand how bacteria evolves and also at the same time assess some of the conserved proteins or targets which we can directly target for new treatments of these bacteria. So most of my focus is understanding how bacteria evolves resistance, at the same time to understanding how they adapt to the host environment. Now, prior traditional kind of omics platforms have been very good in helping us understand which kind of pathways are very important, but one kind of limitation of these proteomics platforms, as well as even transcriptomic genomic platforms, is that they are able to identify the most abundant proteins. And as we know, the most abundant proteins are not always the most active ones in bacteria. So with a chemoproteomics platform that I'm using to be able to study these kind of host pathogen interactions. It involves activity-based probes, which are mostly composed of a reactive group, which binds covalently to the active sites of the enzyme. So by binding to the active sites of the enzymes, they kind of act as inhibitors of the enzyme. So these enzymes are not available to be able to bind to their main targets. And in such a way, we're able to get a snapshot of the active enzymes in the bacteria under the specific conditions we are testing. And connecting these reactive groups to the tags is a linker which can be modified to allow for reduction of steric hindrances, as well as also development of clickable probes, where you can actually enhance the penetration of the probes into live cells, allowing you to study the active enzymes as in the active biological systems. Now, one thing good about these probes is that with the tag, which is mostly linked to the active reactive group, these tags can be either tagged with a fluorophore, which allows you to directly visualize the active enzymes. At the same time, too, we can actually tag them with kind of an enrichment tag, such as a testibiotin or any other affinity tag, to allow us to enrich the active enzymes and then pull down and then identify these enzymes. Now, the reason why I use these platforms is that it's able to give us a real functional kind of readout of the enzymes which are active in the bacteria without requiring further kind of assays to be able to confirm them. And aside that too, we are able to develop these probes which allow for direct penetration of the cells, allowing us to directly image most of the active enzymes as well as their functional readouts in intact biological systems. Then also with studying host pathogen interaction, one kind of limitation is during infection of bacteria in the host cell, most of the host proteins usually dominate. So if you rely on the abundance-based methods, you are going to end up losing information on the bacterial proteins in that sense, and that will require a larger volume of culture to be able to profile the bacterial proteins in that sense. By using these activity-based probes, because we are relying on the active enzymes, and also we are able to enrich these active enzymes using the affinity tags, we are able to directly profile both the host proteins which are active as well as the bacterial proteins, allowing us to be able to map their metabolic output or their activity. So in one of our studies, we used this probe, which is the ATP probe, to study the interactions between bacteria during infection in Hackett cells as well as THP1 cells. Now, in the Hackett cells, these are human keratinocytes, and then the THP1 cells are human-derived macrophages. Now, using the system, we grew the bacteria, we grew the cells, infected them with the bacteria, and then profiled them using the desdiabitin ATP probe. So these ATP probes have a desdiabitin group, which allows us to enrich them and then pull them down to be able to use mass spec analysis to identify the proteins which bind directly to the ATP. And using this system, we're able to directly identify both active bacterial enzymes as well as the host enzymes. And through that, we're able to must be something. I think probably the battery is down or something. I don't think it's responding so well.
Pointed directly there.
This is kind of like back. Sounds good. Oh, yeah. Yeah. Okay. So, okay. I hope it works this time. Okay. Good. Yeah. So, using this system, we're able to also map the metabolic profile of the bacteria. So, we're able to identify specific enzymes which are activated in the bacteria during infection in the THP1 cells as well as in the HACCT cells. And in the HACCT cells, what we were able to observe from our assays was that we saw that the bacteria kind of adopts a kind of a persister phenotype when infecting the HACCT cells. But in the THP1 cells, we're able to map out the metabolic profile and identify that the bacteria kind of switches to a more active kind of metabolic pathway where it's able to switch on pathways involved in oxidative stress resistance as well as nutrient scavenging, transport systems, fatty acid metabolism, and amino acid metabolism. So we're able to use the system to be able to identify map out the direct kind of metabolic profiles of the bacteria in the system. At the same time, too, we're able to use the system to map the metabolic profile of the host cell also. So in a sense, we're able to look at both how the bacteria responds to the cell and how the cell also responds to the bacteria. And what we saw with this was that the THP1 cells, which are macrophages, were kind of prioritizing a more inflammatory or autophagy or bacteria killing kind of pathway. But in the HACCT cells, we saw that most of the pathways which were activated were mostly involved in maintaining the epithelial barrier as well as activating of the innate immune system of the cell. Now, together with one of my PhD students also, we used the system to understand how Enterococcus faecium adapts to different growth conditions. So in the system, we grew the bacteria in BHI, brain heart infusion medium, supplemented with human serum, as well as organoid extracts from colon. And through this profiling, we were able to identify unique, activated, and repressed kind of bacterial proteins under the various conditions. So we're able to test this system in different bacterial systems as well as different media conditions, and we're able to identify really good kind of pathways. We're also able to identify some unique proteins or proteins such as the ABC transporter, as well as this two-component system, which was very vital for the survival of the bacteria or the growth of the bacteria under these conditions. So furthermore, with this kind of system, although it can work as a standalone system, it's actually more informative when you integrate this kind of system with other omics platform. And that's what we did in this study, where we studied how S. aureus adapts to different growth conditions. So we have the artificial nasal medium, human plasma growth in human plasma, and then the normal TSP media, and then the RPMI media, which is used to grow the THP1 cells. And then we looked at also the extracellular bacteria, as well as the intracellular bacteria, internalized bacteria by the THP1 cells. And in this study, we used two probes. We used the fluorophosphonate probe, which actually binds to most hydrolases, as well as the ATP probe, which binds to most ATP interacting proteins. Now, the reason why we integrated this system with the RNA sequencing is to be able to identify the possibility of post-transcriptional control during the growth in the various conditions, where we can see the correlation between the RNA sequencing as well as the activity-based profiling. Because the RNA-seq gives us the gene readouts, whilst the activity-based protein gives us more of the activity readouts. So that's why we integrated these two systems, and also to be able to identify some of the kind of survival or condition-specific kind of adaptation of the bacteria in the various media conditions, as well as how they're able to adapt to the various conditions we are testing. But then, as we expected, the correlation between the RNA sequencing and activity-based profiling was very low when we looked at both the ATP activity-based probe as well as the fluorophosphonate probe. And we saw very low correlation between the two. We also classified all the proteins and genes based on their core category and look at the correlation between them. And most of them showed very low correlation, and some even showing kind of a negative correlation between the activity of the protein and the gene expression. We further looked at clustering based on the concordant or discordantly expressed or activated proteins in the bacteria. And with this, we use the UMAP to be able to map out the distribution of these proteins. And with those proteins which are marked in red, those are either all kind of down-regulated or repressed under all conditions, or either activated or expressed in all the conditions tested. And using these clusters, we're able to also map out the enrichment based on the core categories. And as expected, for most bacterial proteins, they have not been annotated, so most of them classified as unknown functions, with a few of them classifying under the category J and also in the category E. So at least this also gives us the chance to be able to map out these kind of pathways in which the bacteria kind of adopt during the various conditions we are testing. At the same time, too, we're able to identify kind of condition-specific as well as probe-specific kind of conditions where we have some proteins which are expressed. Let's say, for example, this protein which showed activity with the hydrolysis as well as expression with the RNA-seq. But we have some proteins which were showing kind of differential expression based on the condition we are testing. And further analysis also showed that these were kind of responding to the various conditions based on the media and also the host cell we're using to test them. At the same time, further validation also showed the presence of post-translational and post-transcriptional kind of control of most of these proteins as well. Okay. Okay, I'll just wrap up. Yeah, so we're able to also map out the various kind of survival strategies where we have the bacteria adopting a nutrient shift, energy conservation shift, as well as immune evasion due to the various proteins and also proteins and genes which were activated in the system during testing. Then furthermore, aside studying host pathogen interaction with our group in Stanford, we're able to also use the system to identify kind of novel kind of drug targets and also use the chemotypes based on the fluorophosphonate protein to be able to screen a large-scale kind of compound library to identify various compounds which show differential kind of activity or inhibition of the bacteria. Then also further testing also showed that these kind of proteins were very active in other gram-negative bacteria, gram-positive bacteria as well. Then currently with the work I'm doing with WRC, which is sponsored by the Grand Challenges. We are kind of combining the chemoproteomics platform with AI and then whole genome sequencing with molecular biology to be able to kind of identify new kind of antimicrobials against Klebsiella. So this is my team of researchers in WACBIP, as well as the collaborators from Norway, UK, as well as New Zealand, and then in WACBIP. Thank you.
Thank you so much. That was a very exciting talk. May I take one or two questions? Okay.
Thanks very much for such a wonderful presentation. And what
I don't know if you can hear it. Yeah, is it?
So, what I wanted to understand was the proteome profile, and I think you mentioned something about an enzyme being able to effectively bind to its protein targets. But then I wanted to ascertain how this was quantitatively determined, because if there is no binding affinity markers to actually target to check these binding, I don't know if you'll be able to quantify this binding as maybe it was effective or not. So I wanted to check how this was quantified.
Yeah. So with these probes, the direct interaction with the protein targets actually allows us to be able to put them down and identify the proteins using mass spec analysis. One way we are also able to overcome the challenges of non-specific binding is that for each probe we have a competitive inhibitor, which we use to also test to be able to rule out non-specific binding. And for each probe that we use, we have those inhibitors, in-house inhibitors, which we use for testing that. Yeah.
Okay. Yes.
Yeah, thank you. For COVID-19 vaccine, one challenge basically, should I say, happens to be that a vaccines, should I say, getting to the cells, and a proposal has suggested that nanoparticles can address that. So how can nanoparticles help basically with these solutions in drug discovery and target identification?
Yeah. So with regards to using nanoparticles for delivery of these targets, for example, one of the work we are doing is to be able to incorporate these kind of microRNAs as well as other kind of biologics into nanoparticles to be able to target cells. But then you also have to consider the kind of stability of the vaccine, whichever, if it's a biologic or whichever kind of system you are using, as well as also consider the kind of nanoparticle you are using. Because you have to consider if you are delivering it to maybe through inhalation, they have to look at the stability, whether you are using a nebulizer or you are kind of just using a dry powdered form, the stability of the kind of cargo you are going to deliver as well. So there are multiple factors that go into kind of delivering this those kind of particles for the targets you want in that sense. Yeah.
Okay. All right. I guess, thank you so much, Stephen, for a great talk. And before we move to the next presenter, I was told to remind all those who were selected to score the posters that you have to do it now. So if you haven't done it, I'm told to remind you to do the scoring now. Okay, so.
Okay, our next talk will be from Silas Kansa Yeboah, sorry if I'm saying that wrong, who will take us through genomic signatures of lumefantrine selection and tolerance in Plasmodium falciparum clinical isolates.
Thank you for the introduction. Once again, my name is Silas, and I'm going to be sharing with you findings from my MPhil work titled Genomic Signatures of Lumefantrine Selection and Tolerance in Plasmodium falciparum Clinical Isolates in Ghana. Currently, global anti-malarial drug surveillance is heavily focused on artemisinin partial resistance, which originated first in Southeast Asia and has since emerged independently in parts of East Africa. Now, while this partial resistance may spread or even emerge independently in westward across the continent, we in West Africa, specifically Ghana, already face a different paradox. Despite little to no evidence on artemisinin partial resistance within the country, there are already reports indicating reduced parasite susceptibility and field efficacy to artemether-lumefantrine, which is one of the most widely used ACT regimens in the country. Now, this is thought to be driven largely by emerging tolerance to the partner drug component, lumefantrine, and this has been reported in other West African countries such as the Gambia, Senegal, and Burkina Faso. Now, zooming in on Ghana, drug resistance, drug susceptibility studies conducted within the region suggest that for partner drugs, piperacin and amodiaquine, parasites generally remain susceptible to these partner drugs. However, when we look at chloroquine, which is a former frontline anti-malarial and has since been withdrawn from clinical use due to the emergence of widespread resistance, we see a downward trend which signifies that chloroquine is gradually regaining its efficacy. Now, this has been reported elsewhere, and in stark contrast, this is my drug of interest, lumefantrine, where we see an upward trend signifying that lumefantrine efficacy is waning with time. To further exacerbate this problem, there are no known reliable markers to track this emerging phenotype. Hence, the malaria community relies on existing markers such as the PFMDR1 NFD haplotype, which may not fully be able to explain these subtle changes we are seeing, highlighting a paucity on genomic regions that may influence this emerging phenotype. And hence, I address this critical gap by integrating ex vivo susceptibility testing and whole genome sequencing. Toward this, 84 malaria-infected individuals were recruited for this study, from whom blood samples were collected at the Lekma Hospital in Accra. These samples were then transported to the lab where they were used for our ex vivo susceptibility testing. And we adapted to short-term in vitro culture a portion of these samples to increase parasitemia to boost DNA yield for subsequent whole genome sequencing. From this, we were able to sequence about 29 genomes, and using bioinformatics approaches and population genetics analysis, we were able to highlight regions within the genome under recent selective pressure. Now, the variants obtained thereof were then integrated with my phenotypic IC50 data to determine associations with this altered response. Now, first of all, we set out to determine the ex vivo susceptibility profiles of these clinical isolates. And from our ex vivo susceptibility testing, we were able to take a snapshot of parasite response to ACT partner drugs, piperacin, lumefantrine, and amodiaquine. Now, the red line represents the IC50 thresholds for these drugs, and the number of successful assays set up for these drugs are indicated above. Now, at a glance, for piperacin and amodiaquine, we see that parasites remain generally susceptible, which corroborates the trend shown earlier, with few parasites crossing this threshold. Now, for lumefantrine, we see that there are a number of parasites that cross this threshold, indicating that there is emerging tolerance within the population. Now, next, using the 150 nanomolar cutoff for lumefantrine, we're able to group our parasites into two groups, the tolerance group and the sensitive group. Now, generally, we see that parasites have varied response to this drug as compared to a 3D7 reference, which clusters tightly at the bottom. And our tolerance population recorded high median IC50s and geometric mean IC50s as compared to the sensitive group and the 3D7 control. Now, for uniformity, I would like to clarify that this tolerance I'm talking about indicates reduced susceptibility to lumefantrine. Now, from a public health perspective, we sought to understand how these phenotypic groups behave or fare when challenged with the other partner drugs. And we realized a characteristic trend in the tolerance population where there is decreased IC50 values as lumefantrine IC50 values stay high. Now, when we move on to the lumefantrine sensitive group, we see a much more confused pattern, but once we're able to drown out certain noise and focus on the parasites at the top of this group, we realize that we begin to see some directionality as compared to our tolerance phenotypes. Now, this is exciting for the National Malaria Elimination Program because it supports the rationale of multiple first-line treatments and drug rotation. Now, having determined this emerging tolerance and having established the responses of our clinical isolates to these drugs, we went on to query the genomes that we sequenced to be able to identify regions within the genome that were responding to this pressure. And for the analysis I'll be presenting after this slide, we utilized our 29 samples we were able to sequence. Now, before zooming into the discovery phase of the project, we zoomed in on PFMDR1 and PFCRC genes, both of which have haplotypes, the NFD haplotypes and the CVMNK haplotypes, which have been linked to reduce susceptibility to lumefantrine. But interestingly, when we look at these phenotypic, these genetic stratifications, we realize that we have a mixture of parasites where both sensitive parasites and tolerant parasites retain the same haplotype that has been linked to reduce susceptibility. Hence, we say that this genetic stratification does not phenotypically distinguish these parasites, which calls for an unbiased whole genome discovery to identify the novel genetic drivers of this field-observed lumefantrine tolerance. Now that brings me to the discovery phase, and here we utilize two major measures of positive selection, the integrated haplotype homozygosity score and the ratio of site-specific homozygosity, and I show here the latter, which allowed me to compare signals of selection in both comparing my sensitive and tolerant cohorts. Now, the striking observation here for me is the identification of signals in the phosphoinositide binding protein, PX1, which our keynote speaker on Wednesday touched on. Now, this is exciting because this gene has recently gained traction within the malaria community with regards to finding markers for lumefantrine, and toward this, a haplotype designated PIN, PIN, has been linked to reduce susceptibility to lumefantrine. However, what remains unclear is the potential impact of these individual SNPs on the tolerance phenotype, and the fact that this study was conducted with an Eastern Ugandan population suggests that this may be site-specific. Hence, we need to track across other sites as well. Now, from my scans, I was able to generate 9 SNPs in total, which I integrated with my ICVT values, as I mentioned earlier, using a logistic regression model to determine associations with altered lumefantrine response. Now, out of the 9 SNPs obtained, 3 out of the 9 fell within the PX1 gene, with the M1701I being the only SNP that was common to the PIN haplotype I described earlier. Now, for this plot, an odds ratio greater than 1 indicates an association with tolerance, and less than 1 means sensitivity. We were able to obtain some directional trends here. However, none of them were able to attain statistical significance, mainly due to the small sample size. And to test for the true nature of these directional trends, we used a simulation analysis, right, which helped us to increase the sample numbers to 100. And by so doing, we realized that some of the biological trends we observed previously reached statistical significance. In total, we were able to select 5 SNPs, 3 of which confer or associate with tolerance and 2 associating with sensitivity. 2 out of 3 of the tolerance-associated SNPs were found within the PX1 gene, and altogether, this generates hypothesis for further functional validation and testing of these markers with regards to reduced lumefantrine response. Now, in essence, my study has been able to show that parasites within the country are generally susceptible to partner drugs with emerging tolerance to lumefantrine. PX1 SNPs, as I mentioned earlier, are also significantly associated with this tolerance in the study cohort, and together this provides significant critical evidence for the National Malaria Elimination Program in terms of drug susceptibility and surveillance. I'd like to acknowledge my supervisors, Professor Lucas Amenge Tego, Dr. Yawaniwe, the following people, the Cell Biology and Immunology Lab here at WACBIP and Lekma Hospital. I'm also grateful to the Gates Foundation for my MPhil fellowship under the IMPALS project, Malaria Gen for the sequencing support, and the Department of Biochemistry and WACBIP for the opportunity and platform. Thank you for your audience.
Thank you, Silas. Any questions for him?
Yeah, thank you very much for the presentation. For your definition of tolerance, you said reduce sensitivity to the drug, and I want you to clarify. Does it have any temporal condition, or is it based on time that they become either sensitive again or they become fully resistant to the drug?
No, so in parasite evolution, when we talk about sensitivity and resistance, there is an intermediate level that is tolerance, where we've not yet seen fully blown resistance, but then these parasites demonstrate reduced susceptibility to the drug in question by means of the IC50 values. Yes, so it's possible that with time, when whatever pressure that is driving these parasites to tolerance might definitely drive them to resistance with time. Thank you.
Yeah, I have two questions. So the first is the meta-analysis you did.
Yes.
What are the confidence intervals? Because you're looking at samples collected across different years, and I don't know if those samples were coming from the same region. There's an issue about lumefantrine and its solubility, so which creates a lot of variability across labs, even between batches of analysis. So I think you might need to think about the confidence intervals around those, and how reliable those differences, those trends that you're seeing might be. Secondly, in your haplotype, cross-haplotype analysis, RSB or XPEHH.
Yeah.
There were other regions of the genome, like I could see chromosome 13 or chromosome 12, that had higher values, but you biasly picked PX1 because there was already a publication on it. In that publication, there were those SNPs that were in the pin haplotype were very rare in West Africa.
Yes.
And wouldn't you think it's maybe better to actually analyze those other regions that you have in the genome? Secondly, you might want to think about also the confidence, because I could see that your values are a bit low. Considering that you had only 20-something genomes, I think those that you are comparing, the groups don't give you probably enough power to actually get the right frequencies for haplotypes that may be segregating based on the phenotypic background of the parasite. So you may want to think about that.
Yes, please. Thank you so much.
Any further questions?
Thank you. Thanks for your very clear presentation. There was just one thing I didn't understand at the end when you were looking at the associations. You had a sample size of 29, and then on the right you showed a right-hand panel with a sample size of 100. And I think you said it quite quickly, so I didn't fully understand where the 100 sample came from, and the validity of the statistical analysis.
My apologies for that. So the 29 samples are actually my original data set, but then because none of our biological trends attained statistical significance, mainly due to the sample size of 29, we used a simulation to help us increase the numbers to 100 and test whether these associations were true.
Then I can say it's not valid. It's not statistically valid. You can't expand a sample size by simulation.
Not just increase the sample sizes. Not at all. But then it's just supposed to tell us that, all right, so maybe if we could have increased these sample numbers to 100, what could have been happening or what could be happening?
So it could guide you in sample size calculation for a future study, but that's a different thing from making any inference about the statistical significance of your existing data. So I think it's important to separate.
Sure.
Yeah.
Okay.
Thank you very much.
Thank you too.
Thank you very much, Silas.
Thank you.
Well, it's simulation. Anyway, okay. Yeah, thank you so much, Silas. So we'll now have our last presenter, who is Lukman Bati. I don't know. Yeah. He's going to be talking on extracellular vesicles derived from Lactobacillus fermentum. CECT5716 attenuates tumor development and inflammation in mouse model. Okay. I thought he was one. Lukman, is that you? Oh, okay. So he's there. Lukman, yes, you have 12 minutes.
Which one? Okay. Okay, this one? Okay, this one, okay. Okay. Good afternoon, everyone. I am Lukman Bati. I'm PhD student at University of Granada, Spain. First of all, I would like to thank the organizer to give me the opportunity to share my work with the title Extracellular Vesicle from the Probiotic Lactobacillus fermentum Attenuate Tumor Development and Inflammation in Mouse Model. Now this is the outline of my presentation. So colorectal cancer is the third most common cancer diagnosis worldwide and the second leading cause of cancer-related death. So every year we have approximately 1.9 million new cases, and which lead to 900,000 deaths every year. So its development results from a complex and multifactorial interaction between genetical alteration, environmental factor, chronic inflammation, gut microbiota dysbiosis, and alteration in the tumor micro environment. So another important concern is the increasing of the colorectal cancer in young adults. So although the highest incidence rates in Europe, North America, and Eastern Asia, we have a lot of mortality in many low and middle income countries due to the delayed diagnostic and limited access to treatment. So we have major risk factors include obesity, chronic inflammation, genetical mutation, smoking, and consume of alcohol. So the current therapeutic management of colorectal cancer remain on surgery, chemotherapy, targeted therapy, and immunotherapy. But several major challenges remain, including drug resistance, tumor recurrence, and metastatic progression. So it's very urgent to look for another approach therapeutic to reduce and treat the colorectal cancer, such as probiotics, postbiotic, and extracellular vesicle, which are the focus of my study. So we started working on these probiotics many years ago. Recently, evidence has shown that with immunocompromised people, the alive probiotic can cause some damage. So we started to look at the metabolites produced by the probiotic. So my latest work showed that the bioactive low fraction of the probiotic attenuates intestinal inflammation and dysbiosis in DSS treated mice. So now we want to look out of the vesicle produced by the probiotic. So extracellular vesicles, they are nano-sized membrane-bound particles realized by almost all cell types, including bacteria. So they can transport bioactive molecules such as protein, lipid, DNA, mRNA, and microRNA. And they participate in a lot of biological processes, which are cell-to-cell communication, immune modulation. So our aim is evaluate the effect of these vesicles in inflammation associated to colorectal cancer using in vivo experimental model. So first of all, before evaluated the biological activity, we isolated the extracellular vesicle. And these isolated extracellular vesicles are characterized by several complementary methods. We use Bradford protein assay to determine the concentration of protein in our extract, and the transmission electron microscopy to visualize our vesicles dynamic light scattering to see the size distribution of the vesicle, and at the end we use LCMS metabolomics approach to characterize the metabolites present in our vesicles. So after that, we use the well-established method azoxymethan DSS, which consists to combine the azoxymethan which provoke the DNA mutation and generate cancer. We combine it with the DSS, which provoke the chronic inflammation. So in every cycle of DSS, we look for weight loss, stool consistency, and rectal bleeding to see the disease activity index. So at the end of our experiment, we collect colon sample and fecal sample to analyze by histology, qPCR, ELISA, and fluorescence technique. So we use, to further understand, we use another model, which is xenograft CMT96 model. So this consists to inject the cancer cell to the subcutaneous injection of the CMT96 mice cells, and we treat with the vesicles during two weeks. So every three days, 4 days, we see the tumor volume, the body weight, and at the end, we collect the tumor sample to analyze more. So now I will present you some finding of my work. So the transmission electron microscopy show us vesicle. The one who is in blue, they are the vesicles, and the other one we don't know for now what is it, but we are still looking for that. So the characterization show us an heterogeneous size distribution from 108 to 265 nanometer. LCMS show us 17 metabolites from different chemical class. The most represented are monoacyl glycerols, peptide, and sphingolipid. So this slide summarize our macroscopy finding. So the administration of our extracellular vesicle reduce the tumor burden, including the decreasing tumor number, volume, and size, while they improve macroscopic disease indicators such as colon morphology and disease activity index evolution. So histological examination. Histological examination confirmed that our extract reduced tissue damage and inflammation in the treated mice. Ki65 staining show that the vesicle attenuates cell proliferation in the model, and so that they can contribute to protective effects against tumor development. So in this model, we further analyze some genes, some pro-inflammatory cytokine who are increasing in colorectal cancer, because we are doing the association of chronic inflammation and colorectal cancer in this model. So we see that our treatment significantly reduce the expression of these inflammatory mediators. So we can say that our treatment may attenuate the pro-tumor inflammation microenvironment, contributing to the reduction of colorectal cancer development. So the analysis of gout microbiota show that our vesicles restore gout microbiota balance and diversity, contributing to their protective effect against tumor progression in azoxymethan DSS model. So in this model, we observe that the treatment with the vesicle significantly inhibited tumor growth and decreased tumor weight in this model when we compared it to the control. So to further understand the mechanism, we went and see some gene expression like P55, which are cancer suppressor genes, that our vesicles, the gene expression analysis show that our vesicle increase tumor suppressor gene expression and reduce the proliferation marker supporting that cycle cell arrest tumor growth inhibition. So we can associate the activity of our extract to some metabolites identified, which are the phosphatidylglycerol, sphingolipid, fatty acidamide, because they have been reported that these components can attenuate chronic inflammation by suppressing some signaling pathway and reducing the production of pro-inflammatory cytokine. The anti-tumor activity can be associated to the bioactive lipid mediator and peptide, which have been reported that they can regulate cycle cell progression and inflammatory signaling pathway. So in conclusion, we can say that our treatment may inhibit tumor progression by activating P53 signaling, decreasing inflammation by down-regulated pro-inflammatory cytokine, restoring gut microbiota homeostasis, highlighting their therapeutic potential. So we continue the analysis. Now we are running in vitro model in cancer cells, and we are comparing it with the normal epithelial cells. We are still doing Western blot analysis to see the mechanism of the action of our vesicles. So we are doing more experiment to better understand the mechanism. So I would like to thank my team, all the institutions who have found my work.
Right. Thank you so much, Lukman. Are there any questions? Okay.
Thank you for that presentation. That was very clear. You mentioned some side effects with the current therapeutics, but I'm wondering with the extracellular vesicles, if you noticed any side effects in the mice with that treatment.
Say he didn't hear you.
Oh, really? Sorry. Can you hear me now? Can you hear me now?
What?
Can you hear me now?
Yes.
Yeah, so I'm wondering if you noticed any side effects with the extracellular vesicle, like that treatment.
That treatment?
Like the side effects, are there any like downside? Like extracellular vesicles, when you use them, you're seeing positive outcome, but are there negative outcomes as well? Oh, I don't know if that's what you mean.
Yeah, that's my question, yeah.
Okay, if the extracellular vesicle...
So you've seen that it's reducing the tumor? Yeah, but are there any adverse effects? Are they...
Okay, we evaluate the cytotoxicity of the extracellular vesicles. So we don't see any adverse toxicity in the cells we use for the assay. Well, we started using extracellular vesicle or postbiotic because of the toxicity of probiotics. So they should be more safer than the use of the probiotics.
Okay. Pardon? Yes, that's what he says. Are there any additional questions? I think time is fast spent. Okay. All right. Thank you so much, Lukman. And that was our last talk.
This way. Let's thank Jesli and Hadijatu for closing us out well. So, did we even finish on time? I'm just trying to see if we finished. Yes, we finished roughly on time. So, Prof. Awandari is at Mike is at farewell function for outgoing Pro VC raid, and he's running late. So we'll start off the feedback session, and hopefully he's going to join us before we start the party. So I think, do you guys have the feedback? Yes. So while we're talking, please scan the QR code and give feedback for the conference, and we'll get some feedback from the room. Okay, it's fine. All right. So I think it's been an eventful 3 days. It's been, for the first time ever, our visitors discovered that the power goes off in Ghana. You know, even though we did manage to recover a little bit, but also that when the power goes off, the internet becomes also problematic. I hope it didn't have too much disruption, but maybe let's get some feedback around the room. We'll start with Kwipilat. I think this is your first time here in person, right?
Okay, thank you very much. So I want to take this opportunity to thank WRC for giving me this opportunity to, first of all, to not only present my work, to also learn from the best. So I think I've learned a lot from the first day when we attended the course that had to do with the grant writing. And we've had conversation from different domains of science. And I believe I'm going back with the message that WRC conference is one of the best. And I hopefully by next year,
Wait, when
I'll
you
have
say something like that, people have to clap.
So hopefully you guys will give me the opportunity to attend another conference like this. Thank you very much.
All right. So it's not just that we want the good only. We want the good, the bad. So maybe you give the high points, what you thought was the high point of the conference, what you thought was the low point of the conference. I'd like to give our keynote of the day a chance to maybe give us his high and low. Oh, okay. You want to finish?
Yes, I won't call it the low point though, but I believe, in the next session, you guys should bring more of vector biologists, because I think for all the abstracts that I saw up there was nothing that had to do with vectors. So I didn't quite relate.
Okay.
So, but in the next conference, you guys should bring more vector biologists. Thank you.
I can see some people saying, what, you didn't see any vector biology? Okay. Yeah, but that's a good point, well taken. I think we don't deliberately restrict what you actually see in terms of the allocation is based on the number of submissions that are vector biology. And the number of, so the sessions that have a lot of submissions end up having a lot of presentations and a lot of accepted abstracts. So that's really how it ends up being. But Professor Alapulinsa, yes, let's get his feedback.
Hello. Yeah, overall, I think it's an excellent program. It exceeded my expectations. So before I came here, I was going to YouTube, watching the past conference just to have a fair idea of how it is like.
The volume seems to be a bit low.
Hello?
Yes.
All right. So before I came here, I was always going to YouTube and trying to watch videos to have a fair idea of how the conference is like. So I think it has exceeded my expectations. And the part that I like the most is the student presentations. And if there's anything that I can recommend, would be to even expand it and give them more opportunities, create the awareness in other institutions like KUST. I mean, for me as an undergrad in Ghana here, I wish I had the opportunity to meet other students from UG and other places to present. And like I said in my talk, I had zero experience about how to analyze data and all of those things. And even when I started graduate school, I had never presented in front of people, so it took me a while to know how to present before people. So you would always see me very quiet when I'm trying to make a presentation, because I was learning the first time to stand in front of people and present. So if there's anything that could be done to improve this more, maybe to give the trainees more opportunity to present, maybe not even the flash talks alone, but to even present a few of them to not those who are doing PhD, and like even the undergraduate students. But I think overall it exceeded my I didn't know there's so much work going on in Ghana. And then the other thing that could be done to improve, I think most of the talks have been on malaria or antimicrobial resistance. So, I mean, and so it can become like everybody is doing the same thing, the same artisanal resistance. So like it's too much overlap. So maybe most people are not aware that they could present on other topics. I know a lot of people do this, like there are people who are investigating these medicinal plants that are used, look in folkloric medicine, and then they are testing them and those things. Yeah. So some of those things could also be presented here. But for me, overall, I intend to actually encourage my lab members. When I was coming, I had this undergrad saying, I've never been to Ghana. He was just trying to tell me that, oh, I would have loved to go with you to Ghana. And in my lab, if you have an abstract accepted at a conference, we will normally sponsor you, we'll pay for your accommodation and all of those things. So I'm hoping that maybe people in my lab will be able to attend future conferences. Yeah.
Yes, thank you for those remarks. In fact, we want as many students as possible to present. This is why we were trying to do the experiment of the parallel session to see if we can expand it, to do it well. The challenge is not just space, it's also technology. And that's one of the reasons why we have to make sure that we can effect it very well. We tried 5-day conference, and that was crazy. I don't know, maybe we'll do 4 days or something, because 5 days there, it's unless if, personally, we could go to 9 every day. I don't mind. I don't know what others feel like, but we'll keep looking at it. Actually, before we continue, do you have the slide of the committees that were involved in setting up this? Okay, we'll continue when they find the slide. It was supposed to be projected at the beginning of the conference. And that's just to also give us for us to appreciate everybody behind the scenes who puts together the conference. I think I'm the only one who stands here most of the time, but there's a whole team, there's a whole machinery behind, and we need to appreciate them. So, Hadijatu, would you like to give us your highs and your lows? Or you can give it to the gentleman sitting next to you. Yes. All right. Okay.
Okay, go ahead. I'll think about it.
So how we plan the conference is that we have 2 committees, which is how it's been for the last 5 years or so. And this year, we had the scientific committee, which was chaired by myself. Then you have we have Prof. Niels Kwashi. Andrew, this is unforgivable. It's unforgivable. You misspelled his name. Yeah? No. We have the same Kwashi. Eh? All right. We have Prof. Oh, so please, when I mention your name, even though I know that's not your name, show of hands so that everybody sees. So Prof. Niels Kwashi. Prof. Samuel Dodu. Prof. Lucas Amengetigo. Dr. Henrietta Mensah Brown is in her office. Dr. Elma Ametepe. Dr. Jesli, that one too is misspelled. Jesli Chirarua. Miss Lady Baba Donko. I think she's out there in the back. And then Miss Priscilla Ewa. And then on top of this, for the abstract review, actually a lot of the abstract review, we use all our fellows, our faculty, postdocs. So please just put up your hands if you're in the room and you reviewed abstracts. And then the logistics committee is chaired by Ms. Sika Menka. Sika is our grants manager and pretty much in charge of everything. Andrew, Mr. Andrew Nantoma, MFA Jaju, Priscilla. So unfortunately, because of logistics, I think we'll repeat this later because the logistics people are doing logistics right now for the party. But basically, the idea is to understand that it's not just myself and Prof. Wandari that put this together. It's a whole machinery, as he always says, and it's important to recognize that every single person is equally important to put this together. So those in the room that will recognize Mr. Alfred Kasarisam at the back, Mr. Theophilus Duga. Theo? And is there anyone else in the room? Okay, all right. Let's go back to our feedback session. I think our keynote of day one has been looking like he wants to talk. So please. Oh, Hadi, do you want to go? Hadijatu.
Put you on the spot.
Okay, then from there, your boss.
So I've attended the WACBIP conference before that, but that was a long time ago. And coming back really, and well, basically seeing the quality of presentations, the research that everyone's doing, it's awesome. It's given me a good opportunity to network with so many here, and I've already started talking about collaborations, which is good. One thing though I would have loved is for us to have better internet connection. But overall, and thank you for the opportunity for me chairing a session. It's nice being on the other side of the room. Was that okay?
What is the downside, though? You have to have an app and a...
The internet connection.
Okay.
Yeah, Peter, I don't know what I'm supposed to say because I come here. I've been here every year for 12 years or 13 years. I don't know. Yeah, but the conference I've been attending every time, and I think it's always been an improvement. And I enjoyed the theme of this year, which obviously had the angle of environment, given that the funding streams are flowing environmental now. They're facing climatic change as well. So it was really interesting. So that for me, that was the high. But the real high was the second keynote speaker, and of course David's presentation. For the second keynote speaker, obviously the whole idea about sovereignty, and I think this goes beyond just the presentation in this hall because it takes it at a higher level. So WACBIP is not only talking about molecules and science and cells and AMR, but actually looking at the interface of policy at that level, not only nationally but transnationally and continentally, which is really, really important for us. I think science in isolation is no longer something that we want to continue to do. We need to actually increase our collaboration across the continent. There is no real low side, but if I may recommend, I think it will be important. There are several other institutions which are actually centers of excellence within the University of Ghana, and I think across Africa as well, there are a few others, especially those that are new and upcoming. Beyond the Nigerian team that has been here, I think there may be an effort as well to cross-fertilize by inviting members of other institutions to show up at some point, at least sending out their invitations. Some may be able to actually pay for themselves and come. And that may actually increase the dynamics of this continental collaboration that we want to continue to encourage. And lastly as well, as David had said, encouraging more presentations from younger scientists. Not just lower students. I think really having more aspiring scientists having the opportunity to make a pitch for what they want to present, what they are doing, and what they hope to do in their future would be fantastic. Thank you very much.
Thank you very much. No, I think that's actually a very good idea for us to invite people from different centers to come and present. I mean, we do that, but mostly the directors. But and then we do advertise, and people submit their abstracts. It's really the challenge is the space and the time. The time, unfortunately, is finite. And I was thinking about it this time, whether we should try have one extra day again. I don't know. I'm seeing people shaking their heads. 3 days is optimal. Yes. So we'll just have to figure it out, and we'll see the feedback on parallel sessions, whether this is something that we can do. And to do it well, that means that we'll have to find a way to do streaming from multiple rooms, which is where the challenge comes. Yeah, but we'll figure it out. Another comment, and then I just wanted to ask again. So you said up-and-coming scientists. Is that pre-university?
Oh, no, I mean the more early career development scientists.
Okay. I feel like... Okay, Bolahan? Yes, so if you feel inspired and you want to share a testimony, feel free to put up your hand.
Okay, thank you so much. I think it's been a wonderful conference, in my own opinion. And probably as a suggestion, I don't know, I think it was raised last year. I'm just trying to recollect in terms of having a more formal way of networking the students with senior, especially from those from the diaspora, because especially during lunch, that dichotomy is there. So if there's a way, even if, yes, they are taking different meals, but you can still find a way to make it more formal, to integrate so that the younger scientists can have time to ask questions from the mentors, especially those coming from outside. I think it would be a beautiful way for the students to also develop themselves in networking too. Thank you.
Thank you. Yes, you're right. That was raised last year, and we did debate it, but I guess we didn't do a good enough job of prioritizing that. So it's a good point to make for that. Laura seems to have something on her mind. Oh, okay, you have the mic, and then.
Yeah, thank you very much. This was a nice conference, and a whole lot of ideas was presented. I'm very impressed. This is my second time attending a conference, and I have some few things. I think this is more a scientific space, so maybe the next time we can invite the politicians, maybe one or two of them, to come and spend time listening to some of...
Politicians are always invited.
Yes, maybe they might not get the time, but if they listen to one or two presentations, especially with the climate, environment, health, it might click something in their mind, and then they will, our policies will be well addressed and listened to in the political space as well. So we shouldn't make it only a scientific...
They're always invited.
Yeah.
I'm handing over.
I don't know the question, I just came, but if it's about inviting politicians, we always invite them. We have a standard list of people we invite. We invite all the relevant people. Ghana Health Service, all the directors, the minister, everybody, we invite them. Most of the time, they don't even acknowledge the letters. You know, but we keep doing what we're doing, right? We're not deterred by that, so we invite them. Yeah.
Thank
Peter,
you.
Here's your microphone. Why are you giving it to me? Oh, you're done. Are you done?
But
do the evaluation. You go ahead.
Okay, so I thought I could hand this over.
Yes. Hello.
Yes.
In the case of the politicians, although they don't come, I've had an experience where at another conference where one came and she was like, we were talking so much science. So she didn't really understand what they were talking about. So she tried emphasizing and communicating so that they could also understand. And I think that is what mostly discourages them from coming to some of our programs. So it's mostly about the communication. We are mostly talking science, using technical terms. So, yeah.
All right. I mean, it's a scientific conference. So actually what I can take from that is that we can, maybe when we do our report, we can find a way to synthesize it and share with the politicians. I think next week or so, I'm going to go on TV to talk about the feedback of the conference. So hopefully we try and use simplified language, and it's not something we've done in the past, I don't think. To talk about some of the key messages coming out of the conference, and maybe the politicians might appreciate it. But the truth is, would they even pay attention? They might not even read it. In fact, they probably will not read it, but we can try. You know, they always come, they send people to come for the open day, the first day. And I don't know if they read the reports those people give them, right? Those people are typically told at the last minute. All right. Any other feedback, highs and lows? Anna? You look like you have lots of things going through your head.
I didn't tell you I had lots of things going through my head. Okay, maybe just to add to the point of the politicians. So it would be maybe they don't come for these kinds of events. Maybe is it possible to organize a special one for them, like a policy dialogue event? So where you break down all your science into just bullet points or something, and then you invite them, or even go to the government house or Ministry of Health or something. You can organize something with them. So you know where I work. That's some of the things that we do. We do policy dialogue events for the Ministry of Health to the government. Will something come out of it? Maybe not immediately, but at least they've heard what you are talking about. So, okay, great. Thank you, Prof.
What's the high? What's the high?
I've been attending all our big events. So I always learn a lot. I think I network and I meet a lot of people. Maybe the low for me will be the continuous next slide. What is it called? The pointer is not always working. I think every year it repeats itself. Maybe we can work on that. That would be great.
Thank you very much. Every feedback is important. I hope while we're doing this, can you keep the thing on the QR code so that people can be doing the feedback? Yes.
I was just going to say, I was looking for souvenirs to buy. I didn't find any work-based souvenirs, except for the 2 shares that Sika gave me. It wasn't for free. She's going to deduct for songs. But I was looking for smaller souvenirs to buy, like bands or something like that. I think that would be good just for people to take away.
Okay, I think Andrew wants to say something to that.
We have, apart from the shirts, we have the Wagbe marks. I'm sure the guys who manned our exhibition stand should have told you that. We also have the brooch for sale. It's, the mark goes for 50 Ghana cedis. You can use it for your coffee. And we also have the brooch, which goes for 30 Ghana cedis. So if you are still interested, during the dinner, we'll come around with them. Then we'll set up a special table. Yeah, we'll set up a special table. Those of you who want the Wagbe, who want to go away with something Wagbe, you can get the mug, you can get the brooch. Yeah, thank you.
You mean the pin? Yes. When you say brooch, I don't know how many of us. All right. Professor Kwe, do you want to say something? Okay.
If there's nothing more to say, let's move on.
I was going to give it to our visitors. I was going to give it to our visitors to speak. Okay, go ahead. So, Prof, I was wondering if you had some feedback for us about this meeting.
Well, I must say that I was very, very pleased with the level of presentations, the diversification of the work that's going on in various studies, in various areas, and then the presentation. The slides were very, very impressive, especially coming from some of the junior, staff, the fellows. And then, of course, the results, were very impressive as well. looking at various components and areas of malaria. And then, of course, this morning we also had something on dengue and a number of areas that really, are very critical to public health, and looking at the genomic levels of these. And then with regards to organization, I said that's also very, very impressive. And then I think one of the reasons why we're here really is to continue to see how we can work with, the center. And we hope that Nigeria will be able to work with your center. I mean, the Kano Center that's, we were here in 2024. And I think I want to encourage, this kind of continuous, conferencing, bringing, scientists together, the very senior and the junior, so that they can learn and everybody will grow. And then most importantly is to be able to harness all the presentations, come up with a report, and see how this will impact to the whole public health, public health investment in Ghana as well as the region. Thank you so much. And I want to also thank the director for inviting us and our very close friend, Professor Mpufu. Thank you so much.
Thank you. All right. So I think now people just want to have their beer. So let's stop the talking and let's go have some beer. So we've come to the end of another very interesting conference. I believe that it was worth your time. We thank you all for making the time to be here with us, to spend these 3 days with us. Those of you who traveled, we give you special mention for taking the time to travel. We don't take it lightly that you made the time to travel. These days, traveling is such an inconvenience. So not only have you committed your time, but you have to go through the inconvenience of traveling to get here. I think those from the Gambia, for example, their travel is more difficult than those from London. What's Alfred? So we really appreciate the efforts that you've made to be here. I want to thank my team for the hard work as usual. One of the things that we pride ourselves of is that we have a machine that works. Okay, so even when I'm not here, things are still running because everybody knows their job. Everybody does their job. I don't need to whip them to do their job, and that is part of the infrastructure that we put in place, is to have really talented people playing different roles, so that the entire center works like a machine, and things move very smoothly. So I want to thank my team once again for all the hard work. Yeah. I don't know if you projected the committees. You did? Okay, so committees have already been thanked. So we normally have two major committees, the logistics committee and then the scientific committee. So Peter chaired the scientific committee. They are responsible for the content of the meeting. And then we have the logistics committee that's responsible for everything else, from arranging flights to hotels to everything, cleaning the room, setting up everything else is logistics. And you can imagine that a lot goes on. They barely sleep. When you leave here, then they have to prepare for the next day. They have to clean the place and set up, and everything has to be run through again for the next day. And once you get here, everybody's running around making sure that we can have a pleasant experience. So I'm grateful to them for all that they've done. So thank you all for coming. Hope to see you next year. Let's go have some beer.